little sense of fun, but like, it actually can be responsible as often in air conditioning. I don't know, they're just cool enough though that like somehow, I don't know, I think also every, like, I have bad memories, every drive to like Indonesia, I've felt bad that I've received it.
Okay, I got it working.
Thank you.
We are now streaming.
Do you?
I'll send out the new link.
Oh, there's like 2. Oh, you mean the new one, right?
Yeah.
Oh, there's 3. Sorry.
Yeah, the third is live.
Okay,
That's
cool.
us.
Awesome. I'm gonna give it a like.
Yay.
Thank you for getting it to work.
It's not for kids. Save. Share. Now I gotta send out the new link.
Okay, thank you. I don't think I'm doing anything in the morning, right?
Yeah, I don't think so.
All my stuff is in the afternoon. Cool. Just wanted to chat. I'm just on chat. That's fair. But yeah, sit here. Hello. Thanks for coming.
Are you excited the rest of the lab is coming? I heard from Mark.
Yeah,
The last 2 are coming in July.
Reed and Teacher are coming July 5th, and then Emily is coming back for a visit, and she might come in July 11th, I don't know.
Okay.
Yeah, and then.
That works now with her laptop.
Yeah.
You need to unplug it and plug it in.
Yeah.
Okay, great. I'm gonna pick up some breakfast really quickly. You guys want anything?
It really is. Like, I actually spoke earlier when I met up with a friend, and like, doesn't live to work here. And so we were figuring out, it was just so odd, like, honestly. You didn't enjoy it?
I'm good. Actually, a coffee would be fantastic. Thank you.
Yeah, the same room as lunchroom. We should have hot water, coffee, and
Do-do-do.
some like breakfast.
I guess the only thing on the stream that I wanted. Oh no, it's on your screen too.
Yeah.
There's a square like bar.
Yeah. Oh.
Oh. Oh.
That's interesting. Oh, that might be a window that's just overlapping it.
Oh, okay. It's not an issue. Just wanted to let you know in case.
Yeah.
Okay, I will sit down then.
Okay. don't think so.
Do you want me to monitor like a YouTube or Zoom or some questions?
Yeah, I guess all of those. I'll be watching them too. We can take turns asking.
Okay. Oh, I was just gonna ask you if I see a question, how do I tell you that you're Zoom? I'll just be like, oh, yeah, this is on YouTube or something.
Yeah, you can just raise your hand and ask it.
Oh, okay, that works too.
I have too many windows open.
Oh, there's 5 people watching it.
Yep. Hello, 5 people. And the next door workshop is singing. Yeah.
There should be, there's one right there.
What? How did you just see one so easily?
I don't know.
Oh, I think it's not on your back though. Let's see.
There's one right there. There should be one over here somewhere. No, this is the door. Never mind.
It's your superpower, spotting plugs.
It's great at airports. That's enough.
Yes.
Hello.
Hi. I think so, yeah.
You know where the vendor booths are?
Oh, there's one like downstairs.
Downstairs.
And like kind of that way.
That way.
Oh, it's not like a booth though.
Yeah.
It's more like a, or you're trying to get like fancy coffee.
No, no. See if some friends are setting up stuff.
Oh, I guess it's like multiple booths and stuff. For the one guy I heard from,
Okay.
downstairs.
All right. Yes.
I'm kind of like having.
Okay. All
It's
right.
like past the teacher though.
Ask the teacher.
Because I was sitting there yesterday and there was like a door.
Okay.
But I don't know if that's what they did. You might want to oh, better.
Yeah, like the companies that are coming.
Oh, that's completely different. I think I like a coffee vendor.
No.
You might want to ask me more.
Okay.
Yeah.
Hopefully, thanks.
was my phone vibrating the podium. Hi, are you Ed?
Yes, I'm there.
Hello.
See you. Yes, you see. Sorry, are we not allowed to shake hands?
It's a weird time in our ego too.
I just slapped you. We have a range of boards
Okay.
for poster session. I don't know where they are. Oh, they're apparently they're passive dining. Oh, I don't know if you walked around.
Well, several times, but not to the right places.
So that you figure out where like lunch is.
Yes, although I'm not sure I signed up early enough yet.
Okay, but that's where the poster session is, kind of near the lunch.
Okay. Okay.
Um,
Put that up. No, sorry.
Yeah, you can put it up. And there's like there are a couple breaks in your lunchtime too. If you get that.
Okay. Pins.
It should be.
Oh, I think they have pins. The other person I talked to, he said he used like 8 pins.
Okay.
You have a 4x8 poster, right?
No, it's too small.
Oh no, then you probably need less than 8. Yeah, I know, it's
Yeah.
really big.
Yeah. And expensive.
Are you? I wonder if we should start on time or like 5 minutes after. I don't know if you're doing.
How long is your thing?
I can adjust it to make it longer, sure.
Maybe we can start a few minutes late and still end on time.
Mm-hmm. Yep.
Go to the next and you come back.
Don't know what that was.
Is it not coming from you?
Oh. Hello.
You should see that my notes.
Yes.
Okay.
Oh, I have it pulled up here. Oh,
There we go,
hello.
okay.
I'm gonna,
I don't know if you can hear me. Oh, hello.
we can hear you, yeah.
Okay, great.
I'm gonna shrink it back down to just,
Do you want to test out her spot, her screen?
let's see, you're after the coffee break. We'll test out your screen share during the coffee break. If that's okay. I have not.
Again, I'm not sure if you're talking to me, but checking my slides during the coffee break for my talk sounds great.
Okay. Yeah, I'm talking to you. Sorry.
All right. Sorry again, I cannot be there in person, but I'm really excited to participate remotely and
Yeah, no worries at all.
really looking forward to all the talks.
Hmm.
Yeah, we're still trying to figure out the audio for virtual people because we have one microphone up here by the podium, and so I don't know how much you can hear the room or not. Yeah, so we'll find out.
Yeah, I guess we will.
Someone has to worry. I'm already working.
Let's see.
Yeah, I got charged with tea price. Well, I just was working at it, so it's fine.
Okay, I'm gonna go ahead and get us started so we can keep on schedule. Welcome all to the second CVPR workshop on multi-agent behavior. Welcome also to the folks who are joining us virtually and on YouTube. We'll be keeping an eye out for your comments and questions. So a little bit of an introduction, and then we'll launch into our talks for the morning. If I can advance my slides. Our goal in this workshop is to understand scenarios of multi-agent interactions. So there are many different flavors of multi-agent behavior in machine learning and computer vision. One of the examples you probably saw a lot of in the advertising banners outside is self-driving cars. So the quintessential challenge in self-driving cars is to interpret and predict the actions of the cars and pedestrians in your environment. It's something you do naturally and subconsciously while you're driving, but involves many levels of mental modeling. You need to react to your immediate environment, prepare upcoming actions, adjust your driving style depending on the weather and the city you're in. So it has structure on multiple timescales, incorporating sensory cues as well as context information. Another scenario where we might encounter interacting agents is in swarm robotics, where many simple agents can interact to achieve complex collective behavior. So by understanding how swarming organisms behave in nature, we can devise new approaches to building robots that can respond to diverse challenges in their environment. Behavior analysis and forecasting has also increasingly become an aspect of sports analytics, where computer vision startups have partnered with professional sports teams to model the split-second decisions that players make on the field and consult with them to improve their strategy. Hang on. Yes, everything will be recorded. Sorry for that. In response to the chat. Multi-agent behavior is also an area of keen interest in biology. So many species have evolved complex social behavior that, like this example of swarming in bees, this may help organisms protect themselves from predators and shelter their young. Social structures also allow animal groups to efficiently process information and carry out collective decision-making. So they're integrating evidence from individual agents in the environment and as a collective acting in a more effective means to ensure survival of the group. In neuroscience, we're also interested in how the brain processes social information to make decisions about how to react to another organism. We aim to understand how we form social connections with each other, how we mentally model the actions or states of another person or animal, and how the brain has evolved to shape survival behaviors and social interactions that are flexible and adaptive. And finally, we can study how cells interact to form complex emergent structures. By examining the biochemical pathways that cells use to communicate, we can gain insight into how our bodies function as a cooperative effort of many different specialized cell types. Investigating these topics may lead us to understanding how the first multicellular life on Earth began to emerge from independent single-celled organisms. So multi-agent behavior is something that really spans many areas of scientific research and engineering, and all of these fields have faced a suite of common problems with understanding the actions of agents and how they're interacting with their environment. So the objective of this workshop is to bring together domain experts from different specific applications of multi-agent behavior analysis and share different fields' insights into the ways that they model multi-agent behavior, the different goals of this kind of modeling, and how we understand or what we're trying to achieve by studying multi-agent behavior, and where the challenges are for the field. What emerging methods are developing? Where do we still face barriers to progress to understanding the structure of multi-agent behavior? So we put together a couple of ideas of the goals of multi-agent behavior, which I'll go over briefly here. I'll use a model organism system that we're mostly all familiar with, of interacting agents in Pac-Man. So when you're talking about studying multi-agent behavior, this can mean a couple different things. So the first thing you could do is just produce a description of what's happening, produce a summary of the agent's behaviors while ignoring useless details. So you don't care where a particular behavior was performed in an arena, but you want to be able to distill out that chasing is occurring between your two agents regardless of where they're interacting in space. Ideally, in scientific applications, you want this to be a human unbiased description of behavior as much as possible, so you're not relying on human judgments of actions, but rather you're using structure in the agent's movements themselves to identify agents' actions. We also want to be able to describe the actions of interacting agents across multiple timescales. So from moment-to-moment actions like chasing and turning to more long timescale behaviors as the animals or agents move through different behavioral regimes. Another problem that we face in analysis of multi-agent behavior is prediction. So forecasting and telling us the expected trajectory of our agents and what we plan for them to do next. This could be over the time scale of milliseconds, like when you're forecasting for a self-driving car or trying to predict the action of an animal in an environment. It might extend out to seconds or minutes. So a question that's open in the field is on what time scale we can forecast animals or agents' actions. What happens when animals or agents are initiating an action, and what is the probability that agents perform different actions? Is this something that we can capture in a generative model to predict how agents are going to perform? Another theme and goal of multi-agent behavior analysis is understanding causality. So what are the environmental signals that drive agents to produce specific actions? How do we identify the cues in an agent's environment that they're reacting to generate behavior? And how do these cues change over time depending on the state of the agent? And how do we distinguish between a correlation between an environmental signal and the agent's movements and actual causation, actual variables that are affecting the agent behavior? And in neuroscience, we're also interested in what are the brain mechanisms that are controlling behavior and what are the sensory cues that animals are processing from moment to moment to make their decisions about their actions. In addition to causality, there's this idea of motivational state, which is inferring unobserved aspects of an agent's state, such as their level of stress or their recent history and motivational state, and figure out how these inferred signals influence the agent's actions. We might want to determine what objective an agent is trying to achieve on multiple timescales. We might want to know what cues in the environment the agent is paying attention to, or what the emotional or internal motivational state of the agent is. We might also want to understand how to generate realistic-looking agent trajectories. This is something that you might see in a sports video game where you're trying to generate players that move realistically and perform reasonable-looking actions. We might also want to understand what algorithms are consistent with the actions that agents are performing, and how an agent might modify their actions if we introduce a new challenge or a new objective into their environment. And then finally, overall, we want to understand interaction between agents. We want to know what stimuli agents are detecting from each other, what roles agents are adapting as they're interacting, and whether agents are acting in a cooperative way or a competitive way or in an agnostic way with respect to each other. So some of the computational challenges that are currently faced in the study of multi-agent behavior are understanding our capacity for generalization. We don't really know if there are fundamental computational building blocks or tools that can be used to study behavior across diverse domains. We don't know to what extent we can build behavioral models that generalize across a specific setting for a particular agent. Another goal is simply accuracy. A challenge is determining what description or representation of agent behavior best captures important information for downstream analysis. Do we need to describe everything about an agent's state in order to forecast what it's going to do next? Or can we distill out information about the agent's behaviors and abstract away some pieces of information from the raw data? Can we build a model that is producing consistent descriptions of the agent behavior, that is producing consistent forecasts and consistent predictions about how the agents are going to behave in their environment? Another computational challenge that we might face is interpretability. So producing models that help researchers answer questions about the structure of behavior and provide semantically meaningful descriptions of actions for domain experts. And we want ways of encoding expert knowledge. Conversely, we want to be able to take input from domain experts about what kinds of actions agents are performing and incorporate those into our models effectively. So at this workshop, we're going to be hearing from a range of speakers studying multi-agent behavior in simulated agents, in self-driving cars, and in biological systems, and trying to identify some of these common themes of how agents interact. Before we get started, I wanted to thank some of the folks who made this workshop possible, including our sponsors, HHMI, Google, Amazon, and Simons Foundation, all contributed to the multi-agent behavior challenge that you'll be hearing about in the second part of the day, and to providing travel grants for some of our poster presenters to come to the workshop. The challenge that you'll hear about this afternoon was hosted by AI Crowd, who provided a lot of technical support for hosting the data sets and getting teams up and running on the competition. We'd like to thank our advisory committee, Yisong Yu, Pietro Perona, and Ting Lu, for helping us to organize this workshop and reach out to sponsors, and also to our additional reviewers who reviewed your submitted abstracts for the poster session. We also owe our thanks to the City of New Orleans for hosting this conference. This year, June 19th and 20th marks Juneteenth, which is a US holiday commemorating the end of slavery in the United States. It's a holiday of special significance in the US South, and this is the first year that it's gained recognition as a federal holiday. And the organizers of CVPR encourage attendees while we're here in New Orleans to learn more about Juneteenth and its historical context, to join the city in celebrating the holiday. We're really in a city right now where the historical context of Juneteenth is all around us. So I'll go on and review the schedule today. We have a series of invited talks in the morning and interrupted partway through by a coffee break. And in the afternoon, we'll have a series of submitted talks from presenters at our poster session, as well as talks by 3 of the winners of our multi-agent behavior competition. So with that, I'd like to thank you all for attending, people who are here in person and people who are joining us online, and move us along to Kristen, who is going to introduce our first virtual invited speaker. Yay. Thank you. So, let's see.
So our first speaker is Nicholas Heath, a researcher at DeepMind, and he'll be telling us about embodied artificial agents trained to compete and collaborate in simulated environments. So Nicholas, hopefully you can share your screen.
Yes, I'm just trying to figure out how to do that. 1 second.
Okay, let's.
Do you see my screen now?
We do. Let me get you over to the right monitor.
Okay, I hope you also now see my slides, but not.
Yep.
Do you want my email?
We see your slides, not your email, and it looks like they're going to YouTube also.
Wonderful. Okay, should I just get started then?
Yeah, go for it.
So yeah, thank you very much for inviting me. I'm very sorry that I'm seeing the person I really don't want to be there. I'm excited about the workshop. I think this is a very, very interesting topic. So my plan for today is that my talk consists of 2 parts. We'll see how far we get, and maybe I'll just stop after the first part, and we can have a little more time for questions. By the way, I can't see the Now, what I want to do today is I want to briefly discuss two case studies that focus on soccer. I'm actually not a soccer coach, but it's an interesting test case because it's a very rich task and poses a number of challenges that include sophisticated short-term motor control, long horizon and goal-directed behavior, and importantly for this work. What I find particularly exciting about soccer, obviously also in the other scenarios, is that these levels of decision making are intimately coupled and can't easily be separated. So it's very interesting this with the problem of playing chess with a robot arm. Although this is a problem that involves motor control and high level decision making, in this case, two levels needed to be solved independently and as a result of scientific. That's not true for soccer and many other real world problems. So the first part of the talk, I'll focus on some work that is about learning or training artificial agents to play soccer in simulation. And then in the second part of my talk, I'll briefly touch on a related project that focuses more on modeling and understanding human soccer. Okay, let me get started with the first part. So a significant chunk of your work has been focused on RL algorithms from body control systems. In these days, I don't need an introduction anymore, but for the purpose of this workshop, it's useful to remind ourselves why RL is exciting. And I think one of the main reasons why RL is exciting is that it allows us to specify the goal of the learning process rather than the solution. This is very different from supervised learning. In RL, it effectively avoids trial and error.
And ultimately, it allows the algorithm to discover solutions that we couldn't come up with ourselves. Now, while this has been shown in a number of cases in non-embodied domains, in previous work we've shown that this applies also to the embodied setting. For instance, we have found that, let's place it in order, that in the multi-agent setting, cooperative behavior can arise from simple shaping rewards, for having to play against a diverse pool of opponents, and from the autocurriculum that arises from self-play. Incentivizing such cooperative behavior would be manually very difficult, but from population-based self-play, this arises naturally. And similarly, if we place Walker into environments with diverse obstacles and incentivize them with a simple reward to move forward and don't fall, and also give the environment some curriculum structure, they can come up with sophisticated movement strategies that are functional and robust, would be hard to handle. Take this out for a second. So, unfortunately, while this is true in some cases, the strategy is not always successful, and more often than not, it is difficult to specify the desired learning outcome for an embodied control problem in a way such that learning can succeed. We're often in situations where agents maximize the reward but do not necessarily behave as expected. So this is shown here on the left, where the agent moves forward at the right velocity, but the behavior is clearly awkward and unnatural, and certainly wouldn't work on a robot. Alternatively, agents may struggle to learn at all. For instance, on the right-hand side, where the task is to put the object into the box in the right corner, on the right-hand side of the image, but using only a sparse reward and in combination with naive exploration will basically make learning not succeed at all. And this situation is usually even worse in a multi-agent setting. So to test that we can scale reinforcement learning to considerably more complex scenarios, both in terms of the embodiment and in terms of the task complexity, we've created a simple artificial soccer environment with realistically simulated physics. Compared to real soccer, the environment has simplified rules. For instance, there are no fouls, and the ball cannot leave the pitch. And for our experiments, we instantiated with the environment with only 2 players, mostly for computation reasons. Now, the environment was designed to encourage competent behavior at least 3 different levels of abstraction. So there is robust and dynamic low-level motor control. There are mid-level goal-directed behaviors such as dribbling and shooting. And finally, there's hyper-planning, such as positioning yourself on the pitch, coordinating with teammates, and paying attention to opponents. And in the context of this talk, then, the interesting question is how we can facilitate learning of well-regularized behavior, discovering the right structure at these different levels of abstraction. And the solution strategy that we developed for this work consisted of multiple components. So firstly, we relied on gradual skill acquisition, moving from low-level movement skills to mid-level soccer skills such as running, turning, dribbling, and kicking to goal-directed coordinated soccer gameplay. We use different learning signals to make these different levels of skills succeed. We use imitation learning for the low-level movement skills, reinforcement learning with well-defined rewards for mid-level skills, and multi-agent RL and self-play for the discovery of complex strategies. And we use two different types of skill representations to transfer the behavioral structure from the low-level skills and the mid-level skills into the multi-agent training problem. And finally, we created a curriculum over team size using an invariant perceptual representation to allow training with teams of more than 2 players. So this slide shows an overview of the solution strategy. You don't need to parse it in full, but can you see my mouse pointer? So broadly, the training pipeline proceeds from low-level skills via motion capture indication to mid-level skills trained via RL, which are then these two types of skills are then injected into multi-agent reinforcement learning to train the full game of soccer using population-based self. I'll walk through each of the stages now in turn. To obtain general-purpose low-level movement skills, we used motion capture data from human free-form soccer play. We trained a policy to track diverse motion capture reference clips. The policy consisted of 2 components. The first is an encoder that takes the future poses and translates it or encodes it into a latent embedding or motor command. The second is a decoder or low-level controller that translates the desired future pose encoded in the motor command and the current pose of the body and translates it into an action that is likely to achieve this future pose. The fully trained system can be seen as a general-purpose imitation policy that can reproduce arbitrary motion capture clips sufficiently close to its training distribution, of course. And most importantly, the low-level controller or the decoder can be used as a general-purpose motor module that can be controlled via the embedding space or the motor command here as part of a policy that can solve specific tasks. They've used this idea in the past. After training motor modules for other purposes, we can train a task-specific policy that chooses actions in the motor command space. This is shown here schematically. Then we reuse the low-level motor module to execute or translate the motor command into low-level actions. In the top left corner, we see an example where we had used a similar motor module to learn complex visual motor tasks that depend on memory. The agent is placed in an environment with different colored orbs, and in every different episode, a different orb is digestible, and the agent needs to identify which color is digestible and then collect all of the orbs of that color. And the second example is a manipulation task where the agent needs to learn to pick up and carry boxes around and capture balls. In both these cases, the agent gets egocentric vision and can learn these rather complicated behavior using the low-level motor module. So while this is a powerful idea, as we found in the past, to learn soccer, the skill module itself was not sufficient. In particular, the skill module induces naturalistic low-level behavior, but does not directly induce or incentivize goal-directed behavior like interacting with the ball. So to achieve this, we created a set of mid-level skills. And to this end, we set up a number of training drills relevant for soccer and trade policies to solve them via RL using the previously trained low-level controller as part of the expert policy for each drill. So we had 4 different drills following a target, dribbling, kicking, and shooting, kicking to a target and shooting at the goal. The video shows the module before training, and then after some training for the dribble policy. The expert policy acts in the motor control space for the low-level controller. And here are some of the other drills, kicking to a target, and following. So after training, we then transformed these drill experts into mid-level skills. For this, an extra step was required. All the drill experts are dependent on a drill-specific context, like the target, for instance, for the dribble policy, which is sort of instructed to follow a target. To make them useful as skills, we therefore distilled the expert policies into mid-level skill priors via distillation of information asymmetry. For instance, for the drill prior, so the objective is shown down here, it's effectively supervised learning. For the dribble prior, we executed the expert while it was following the target, and we then copied the behavior that the expert executed into the drill prior, but did not give the drill prior access to the target position that the expert policy was trying to follow. And the consequence of this was that the drill prior effectively represents dribbling towards a mixture of different goals and dribbles in different directions, randomly every so often. This is what you see here, this behavior of the dribble prior. You see it doesn't follow the target, and it turns randomly, stops, accelerates, and slows down. So you can see that, think of this as a pluripotent dribble policy that abstractly represents the skill of dribbling. Okay, so with all these components at hand, they provided the basis for the multi-agent training phase. To ultimately get the complete soccer behavior, we relied on population self-play because it allows the discovery of complex solutions. And what we found in previous work, high-level strategies such as cooperative behavior are hard to handcraft or specify by a rule. Population self-play exploits the fact that A, you have an ensemble of different agents that play against each other, and because you have to play against many different opponents, you automatically become robust. But also because these agents learn sort of simultaneously and all start at a low level, then improve, you get a form of autocurriculum which helps to go from a very, very simple behavior to very sophisticated, or can get to very sophisticated behavior. Okay. As I was saying before, in the case of soccer with humanoids, population self-play was not sufficient. So we therefore combined it with these 3 shaping strategies that are shown here: the low-level skills, the mid-level skills, and also additional shaping rewards. More specifically, we used the low-level controller as part of the agent policies. So the agent policies would take actions in the motor command space of the low-level controller, which would facilitate exploration and impose a constraint on the behavior that the soccer agent would learn to stay roughly in the space of naturalistic, human-like soccer play. very similar to the expert policies. Secondly, we then regularized the agent behavior towards the mid-level skills. Since the skills are quite specific, and we want the agent to be able to deviate from these mid-level skills, we didn't use another level of hierarchy, which may seem natural, but instead we implemented the influence of the skills via regularization. What this means is that we augment the RL objective with a term that encourages the policy to stay similar to the skill prior, expressed by penalizing the KL divergence between the policy and the prior. And then, since we have multiple skills, we need to give the agent an opportunity to choose among the different skills in different situations as appropriate. So in particular, we want to avoid a situation where the agent always follows just one of the different priors, like always the follow prior or always the dribble prior. And to achieve this, we construct a mixture policy of the different mid-level skill priors and then regularize against that. So this is shown at the bottom here, where the different mixture components correspond to dribbling, kicking, shooting, and so on. To understand the effect of this regularization strategy, it's helpful to think about the effect of the KL regularized objective via this simple diagram here. So if you think of the whole plane here as the space of possible solutions to an RL problem, then the RL objective shown up here without the KL term tries to find a solution that corresponds to the region of high rewards, this ellipse here. Now we can think of the prior, and the prior expresses a preference for a particular type of behavior, again in the space of solutions, in the space of trajectories that the agent produces, and this is indicated by the red region. And the KL term encourages the policy to stay similar to the prior behavior, to stay within this red region. If you now combine the reward term here with the KL term in the objective, we obtain an overall objective, the KL regularized objective, that favors solutions that effectively lie in the intersection of the behaviors favored by the prior and by the reward, so this area here. So since the prior and the reward combine in this interesting way, we can use the prior to bias learning towards soccer-like behavior, while the reward, for instance, only incentivizes the scoring a goal. Now, for multiple priors, we can now think the situation is a bit more complicated. If we regularize against all priors simultaneously, we would end up effectively with a preferred behavior that lies somewhere in between all priors. This is sort of indicated here by the gray region. So think of this as the dribble prior, the kick prior, the follow prior. We regularize against all of them simultaneously, we would sort of end up with a sort of average behavior, which is not desired. Instead, by regularizing against the mixture, due to the particular nature of the KL, which is asymmetric, we then get an effect where the agent is effectively just regularized against the best matching mixture component. That is, the mixture component that is best aligned with the reward at a particular point in time, but is allowed to ignore all the others. So by regularizing the agent behavior against this mixture, we allow the agent to switch between different skills effectively at different points in time. So while the skill module and the mid-level skills get us pretty far to make learning happen, some simple shaping rewards still help. So these shaping rewards are very simple, and in particular do not operate at the level of the population. Instead, advise the player to go to the ball or to kick the ball towards the goal. And this sort of puts together the whole pipeline. And I'm now going to show a few results that emerge from training with this pipeline. What you can see here is, although one might not want to call this a behavior human-like, it is rather realistic, rather naturalistic. This is always lies a little bit in the eye of the beholder. And in particular, the agents have learned very sophisticated movement strategies. They've become very robust from the multi-agent training. We've seen the jostling just before. And they've also developed reasonably sophisticated sort of interaction strategies where, for instance, as soon as one of the players has reached the ball, the other one turns away and runs into the space, or they run into space to block opposing players. Just take this play for a little while. Okay, let me move on. You can watch more on YouTube. So we also performed various analysis on the behavior of the players. That was a big part of the work. For instance, we analyzed at what time during training different behaviors would emerge. And we found that simple movement skills like getting up would emerge early in training. So the x-axis here shows training time on a log scale, and the y-axis performance on a particular type of behavior. Whereas like a division of labor between two teammates, where only one of the players goes to the ball, would emerge much later in training. Secondly, we looked at the internal representation of the agents, and we found that they would re-represent the observation of the game in a way that sort of clusters them according to high-level properties of the game situation. So this is a piece in the embedding that is color-coded according to different game states of the raw observation that's fed into an agent, and this is the same observation, but processed by the agent's perceptual encoder, and the clusters are much more pronounced here. We also looked at whether players' performance is correlated with whether they can predict the future of the game, which is a feature that was found in professional sports men and women. So the x-axis here shows agent performance, and the y-axis shows the performance of the agent on a prediction task, where they were asked to predict particular futures. From the internal representation of the player, we tried to predict different aspects of the future of the game, and you can see a clear correlation. And finally, we looked at whether agents can adaptively make use of different mid-level skills. So I was explaining this a couple of minutes ago, how this regularization strategy should incentivize adaptive switching between different skills, should make it possible to adaptively switch between different skills. And what you find here is the difference of an agent's behavior relative to before different skill priors as a function of the time from a kick. So this is the time of a kick, and you can see here that, for instance, the behavior becomes less similar to the follow prior and more similar to the kick prior as the agent as the time approaches the time of the kick executed by the agent. And very last, we also sort of looked at whether we could train larger team sizes through a team size-based curriculum. So training teams larger than 2 versus 2 from scratch becomes computationally very intensive, but because of a particular representation that is sort of to some extent player number invariant, we can first start by training with in 2 versus 2 games, and then gradually increase the team size.
Nicholas, just to let you know, you're at about 26 minutes, so if you can start wrapping up.
Okay, so maybe I'll just stop after the first part of my talk then, and we can have some time for questions if you prefer.
think that would be great, yeah.
Okay, cool. So, and this is actually basically just the end of the first part of the talk. It was a bit slower than expected. So I've introduced soccer as a reinforcement learning challenge. Obviously, we're not the first ones to do that, but there's a long history of RoboCup here. Our goal was in particular to look at basically multi-scale behavior in a multi-agent setting. And the solution strategy that we developed for this was a reuse of low-level skill module, a behavior prize for mid-level skills, and multi-agent training for high-level strategy and robustness. And this has led, and so as you've just seen, the outcome of this was emergent behavior with tightly coupled low-level, mid-level, and high-level skills. Maybe sort of just to say what I was going to talk about is some recent work, mostly done by colleagues of mine, on models of human behavior in soccer play, where they've developed a model that does trajectory imputation to fill in gaps from a camera when the camera comes across the soccer pitch and doesn't always show all players. And I think this sort of nicely links with the previous work that I have presented, because it's sort of it's basically about modeling human players directly, and in particular modeling the high-level play. And we can think about various ways how we can combine this with RL learning strategies. Okay, let me stop here, and then I can take some questions.
All right, thank you. So do we have any questions in person? Oh, okay. Do you want to control your computer? Sorry, I don't know how to control it.
I'll try and find my Zoom client again.
Okay, we have one question in the chat, if I can find it. Oh, it's down here. Okay. Doop.
Right. I can read the question.
Z1 asks if there are any difficulties when doing reward shaping, and how many terms are in your reward.
Right. So the whole point of what I presented was actually to reduce the need for reward shaping, and reward shaping is indeed quite difficult in the setting that we are in. So the ultimate number of rewards is relatively low. I think we have. So you should probably look at the paper if you want to have the exact number, but it's probably about 4 or 5 different reward terms. The whole point is that basically the skill modules that I introduced, the low-level skill modules, sort of ensures that we have reasonably naturalistic behavior without having to work with many different shaping terms, as for instance needed in sim-to-real work for robots. And then the mid-level skills avoid sort of slightly more abstract shaping reward terms, which would incentivize the agent to interact with the ball or kick towards the goal. So basically, the remaining shaping reward terms are then really very simple. And they're also, which I didn't talk about, automatically adjusted by sort of a meta optimizer that runs sort of as an outer loop to the RL algorithm.
So I think we're unfortunately out of time, and we have to move on to the next speaker. But I don't know, Nicholas, if you can answer questions also in the Slack channel potentially, if there are more questions there, or on Zoom. There's so many different ways that we can communicate. And then we also have a panel scheduled for later in the afternoon, and I hope you can join us for that panel too. But we should move on to the next speaker, who is Alessandro Zanardi. I don't know if Alessandro, you're there and you can share your screen.
Yeah, we'll be at the panel.
You might want to do this. Sorry.
Okay.
Can you hear me?
Yeah, let's find
Okay, perfect. All right. Now I hope you can also see my slides.
We can see your slides. Let me pin you.
If you go to gallery view, maybe then you can see everyone. And
Yeah. Okay.
Okay, great. So our next talk is from Alessandro Zanardi, a PhD student at ETH Zurich with Professors Frazzoli and Dorfler.
My name is Frazzoli, and today my talk is going to be about games and preferences, in particular for autonomous vehicles. I want to start off with a bit of motivation and why we are looking at games theoretical aspects of these interactions. And the main claim is that the standard motion planning techniques lead to passive road users. What do we mean by that? If we look at the algorithmic pipeline, or it's oversimplified, but more or less, we have sensors getting, we have a perception module gets the sensor data, builds a representation of the world. We have a prediction module that predicts what the other agents around us are going to do. And all these informations are fed to the planning module that it takes care of computing a trajectory that minimizes certain cost function while avoiding collisions and so on and so forth. So in this sense, we say that the resulting behaviors tend to be overly cautious and passive because we are always subject to what the others are about to do. And clearly, at the same time, we cannot neglect the predictions because otherwise our behavior would become hazardous. And I bet most of you have seen on Twitter or a lot of catchy titles around the web of video, for example, showing some Waymo cars or other manufacturers that have difficulty in merging onto the highway because it's they miss a bit this interactive aspect of the players. So the main claim is that we need to explicitly consider others' decision making when we do planning, and we go from something where we predict what the others are about to do to predict what the others are about to do, conditioned on our actions. And this is really what, at the core, what motivates our interest towards game theoretical aspects. We know that historically, game theory has been comes from economics, has been used to model strategic decision making. But little has been explored for embodied agents. And most of the assumptions, if you take a textbook and start reading it, seem very idealized or very unrealistic for, let's say, the real urban scenarios. Nevertheless, I want to remark that I think you should really see this as a generalization of standard planning, in the sense that you don't miss anything. If you, for example, consider let's say, non-reactive model for the other agents, then you would retrieve exactly what we are already doing for standard planning. So this being said, I want to characterize a bit more precisely what kind of games and what are the peculiarities of this, what we call the urban driving games. So first of all, we have objectives that can be personal and communal, and usually they don't have the same importance. So here you see, for example, they're organized lexicographically. We have collision in the first place, then we want to respect traffic rules, and only later we want to optimize for personal comfort and similar. At the same time, this problem is multi-stage, so we don't want to plan just the immediate next action, but we want to plan a trajectory. I guess most of you are familiar with model predictive control, where you plan a trajectory over a certain horizon for the next n steps. what are other difficulties? So first of all, there's uncertainty, and we think that the most important uncertainty is uncertainty on others' intents. That we don't know where others want to go, and we don't know exactly what are their intents. And finally, we have to deal with partial information, in the sense that oftentimes we don't have a perfect representation of the world. There might be occlusions, there might be information that we don't know, and all this needs to be integrated into the decision making. For today's talk, I'm gonna just speak about the first two, so this hierarchy of objectives and the multi-stage aspect, and let's say delegate the other two to future works. So let me motivate a bit further why we believe that objectives or outcomes are not scalar. So it's very hard, by not scalar, I mean that it's very hard to compress into a single real number the complexity of the decision-making problem that we are dealing with. So first of all, we have multiple objectives that are often conflicting. You know, you want to make progress towards your goal, at the same time you want to stay safe, so have clearance from the other cars and things like this. Clearly, you cannot formulate it just as a scalar objective that you want to optimize, plus some hard constraints. Because you are in an environment where you don't control everyone and everything. In particular, you cannot control others' behaviors. There might be failures, there might be unforeseen circumstances. So there will be eventually a long tail of events, of things that are bound to happen. And when you find yourself in that situation, you don't want your policy or your controller to tell you, sorry, the problem is infeasible. At the same time, you want your behaviors to be explainable because you're dealing also with liability issues. And all this, it's very hard to do it, to explain your behavior if your cost function or what you're trying to optimize is completely a black box, or it's something that is a weighted sum of many objectives. You know, it's hard to retune, it's hard to interpret. So this being said, the idea, we borrow from the idea of minimum violation planning, and the idea is the following. You can organize your objectives or rules, or you can also call them constraints, rewards, in a hierarchical way. And you will have the most important at the top and the least important at the bottom. And you want to find the solution, your decision, your trajectory that minimally violates this. So given that if you are forced to violate one of these, you want to find the one that minimally violates this. And note that also, you know, this doesn't exclude using things like learning or similar approaches, which are black box. They can just be caged into this structure where they propose or suggest a trajectory that then gets scored as other trajectories. Slightly more formally, for the ones of you that are familiar with this, when we extended this to multi-agent systems, then we were asking ourselves, okay, what happens when we extend this to multi-agent systems? And we have that each player can express a preference over the possible outcomes, and this preference, it's a binary relation. And we have these rules, these metrics, objectives that are just functions that given the state and the action, or you can think of it as the prediction of what the other players are going to do, they return a real number. So in this case, very toy example, you have a vehicle with 4 possible alternatives, A, B, C, D, and you have the objectives, this hierarchical preference, which says, okay, at first we have collision avoidance objectives, then we want to minimize the violation of the double lane crossing, and then we want to keep clearance from this obstruction, this obstacle. And this hierarchy would induce an order over the trajectories. So for example, we have that trajectory B and C are going to be preferred over A, which leads to a collision. And we have the same holds for D, which is going to be preferred to A. And now if here we don't further specify what's the relation between area of elevation and clearance, then we would de facto have a Pareto front between the free trajectories that don't collide. So we instantiated this and looked at the structure of these games I want to show you two examples. First one is this cube and drawing games with lexicographic preferences, where we observe that it's sufficient to have a structure in which everyone puts as a first desire not to collide, and then optimize for personal objectives. And this already returns a potential structure. So what you obtain is a potential game, which has socially efficient solutions, meaning that there exist Nash equilibria which are going to be the minimum of the social cost. And this is interesting because it's not the case, for example, for congestion games where you can have things that go arbitrarily bad if you think about the Bragg's paradox and similar. So here in this instantiation, you see, for example, that also these two vehicles in the solution, they are forced to go on the other lane to avoid this car. So in this sense, here they are forced to minimally violate the traffic rules in favor of not colliding. Then we extended this work to more exotic preference structures. In particular, you can build a partially ordered set of these preferences. And what we found out is that as you refine these preferences, meaning that, and refining, I mean that these preferences are you're gonna specify what's the relation between some objectives that at first you consider as incomparable. So for example, you do a weighted sum of these two. It's called an aggregation operation, or you specify an order between two objectives. Long story short, you make these preferences more akin to a total order. And this it reflects on the solutions of the games, where given a set of alternatives options for each player, this refinement of preference induces also a refinement on the equilibria of the game. And this is interesting in particular for the fact that for the, let's say, works that we are doing at the moment, where we show that you can also play games specifying only a partial preference for the other agents. So you're not very confident about what is exactly what is the other agent optimizing for, but you have some good assumption, for example, that he's not suicidal, so he also doesn't want to collide. And this way of formulating the game allows you to do that in a very modular way. So, in general, when you're dealing with these orders, arbitrary ordering, it's very hard to solve the minimization problem. So as you might have seen from the slide before, how do you do it? Well, you generate a lot of ideas and you throw away the bad ones, in the sense that you do the scoring of all the possible trajectories. In this sense, if you look inside let's say the mind of one of the planners, you have a trajectory proposers that generates a lot of candidates, and these candidates get scored according to your order, and you get the best plan, or at least the plan that minimally violates your objectives. Now, what is the problem when you move from this to the multi-agent setting? The bad news is that the joint actions explode exponentially in the number of players. So in order to deal with this problem, I'm going to present now a bit more in-depth recent work that we have under review, where we took the general formalization of these games, which I don't know how many of you are familiar, but I'll go quickly through it. I guess most of you are at least familiar with dynamic programming. So if you take dynamic programming and you expand it to the multi-agent setting, you get, let's say, similar equations, just that you have multiple agents. So you have the dynamics of the game that evolves according to a certain function. You have the joint state of you have the state of the game that in this case is going to be the joint state of all the players. You have the input actions from the players. You have policies or strategies that are maps from the state to the input. You have a certain incremental cost. And what you would like to do is find a solution. Now here, I report for the Nash equilibrium as a solution concept, but clearly there are other solution concepts, for example, different types of Nash equilibria or security strategies and so on. But for now, I will keep it, let's say, modular up to the solution concept that you specified. How do you solve it? Usually, if you look at the textbook, okay, you solve it via backward induction. So if you have discretized a bit your state and your inputs, you build a game graph, and you solve it backwards. The problem is that indeed this explodes exponentially. So if M is the cardinality of your action space, you're going to have a joint action space that is going to be M to the power of N, where N is the players. While ideally you would like at least to have something that scales linearly, let's say like as if you were to solve one N players games, then you would have M times N. So how do people deal with this? The thing is that most of these works try to, let's say, overcome this complexity by solving local games. So you solve an agent, just the game with respect to the neighbors, and then clearly you get a net of all the games that are entangled to each other. What we proposed is something that we call factorization of games over spatio-temporal resources. What's the trick? First of all, this factorization process is something that allows you to build a game graph whose solutions are also solutions of the original game. So let's consider this very simple case where you have two agents, 3 states each. And then you build the joint game, you build the game graph. So you start from the state 1A, so the car is here, the pedestrian is here, and you have all these possible transitions. Like it's basically the equivalent of the multi-agent MDP. Now, what if I were to, and then you have to solve it via backward induction. What if I were to tell you that you can actually retrieve the solutions of this game by solving something like this one on the bottom, where you actually have, for certain transitions, players that get decoupled and go into game nodes where they are completely independent of each other. So what you need to do is rewrite a bit everything as function of the game nodes. So now you allow these game nodes to just have a subset of players, and then everything else basically stays the same. You just substitute the state of all the players with the state of the players in your game node. And you allow dynamics of the type of this type. So where you go from a joint state to possibly you continue the joint game, or you go to decoupled games, and so on and so forth. So this is what would happen as the game proceeds. It could be that you transition from a 4 players games to playing 2 separate games, where one is a 3 players game and one is a single player game. So what's the trick on when are you allowed to do this? The idea is that you want to find some games that are independent at equilibrium, so for which the solution of the original game can be built as the product of 2 separate subgames. And this is, I want to stress that this is not the same as strategic independence. So for example, if you think about the state here, let's say 2A. So if the pedestrian moves forward, the car stays there, assuming that they just only have these 2 actions. from this state, they're not decoupled, the players, but their optimal solution is decoupled, because you know that from here you can move, the pedestrian is going to move forward and the car again is going to move forward. So in this sense, when you actually build the graph, you want to, you're checking if the new state can be factorized. So when you expand these nodes as you proceed, you want to know if you can decouple some players. And the problem is that, yeah, it seems that there's a catch in the sense that to actually factorize, according to what I told you here, you need to know the solutions of the two subgames, because you will want to check if their product is a solution also for the original game. So it turns out that actually you can use, let's say, spatial-temporal resources to overcome this issue. In particular, you can solve the individual games and compute the resources that each player is going to use. So the reachable resources, the reachable set, and the resources that are associated to its optimal policy. And now you can check via intersection of these resources whether or not two players are independent. So very briefly, the difference is that if you solve this in the naive way, you would build a game graph and solve it via backward induction, where build the game graph would be the full game graph of the game. What we did is simply, okay, what if you first solve the individual games? So basically you solve each dynamic programming problem as if the other players were not there. And you can associate to each of these solutions the optimal resources that you would use. And then using these checks among the resources, you can build a factorized game graph and solve this factorized game graph via backward induction. So I will briefly skip this, but well, very briefly, here we propose two algorithms. One that just relies on the fact that empty intersection of resources imply strategic independence of players. And then we have what we call the fact2 algorithm that also uses the fact that you know the optimal resources for the single player games that you solved here at the beginning. It turns out that these have a real impact, especially so these are some examples when we instantiated this on common world scenarios. Well, as you increase the number of players, here you have the number of game nodes and the time to solve it. This is not really optimized, but the relative margins are where it actually what you should pay attention to. You see this huge increase in performance, because basically instead of solving 500,000 nodes, you're solving just 7,000 nodes for this simple game. And here you can appreciate this graphically. For example, if you have this is a visualization of the game graph. If you don't do factorization and you see this dense black area with all the edges versus the game graph that results when you build this in a factorized way. And you can see here, if you plot the number of nodes that you can reach at each stage, you have this, which is at least 2 orders of magnitude less if you factorize properly the game graph. So to conclude, I think some final thoughts on what I learned from this is that, first of all, is that when you have these applications at hand, there's often a lot of structure that you can exploit. For example, in the first two works that we have seen, there's the structure of communal and personal objectives that often don't have the same importance. And in particular for robotics, when it's outside factories, so when for things like autonomous vehicles, you have these very complex decision problems which are in an uncontrolled environment. So you want to have something which is well-defined, and make sense in any possible situation, because there will be always a long tail of events that something happened. Finally, I think that it's important to consider something that we haven't done yet, but the fact that in the real world you have bounded sensing and computation, so what is called bounded rationality for agents. And you want to learn or integrate the uncertainty about others' intents in these decision-making models. And we're still looking forward to find a satisfactory answer to what is the right way of doing it. So with this, I conclude. I think hopefully I didn't go too much over time.
Thank you.
And I'm happy to take questions.
So do we have any questions?
Thank you for the talk. I have a question about how a prediction would fit into this structure of solving game as decision making. Like currently, as you mentioned, is very passive, where your ego motion is not taking into account for other agents' decision making. And if we say we want to solve this game, like how would structure or how would imagine the output of like agent prediction to be, and how would that different from the passive prediction that we currently have?
So there's yes, thanks for the question. That's a great question. I think I've seen already around some work from I think it was published. I don't remember if this year or last year from Waymo, something called conditional predictions, where you can actually query, you want from your perception module or your prediction module to tell you, okay, what if I do this, how do you expect the other to react? Which is different from just asking, okay, what is the other about to do? So you basically condition these predictions on your decisions.
All right, thank you. Maybe one more quick question if anyone has it. Go ahead. If you want to come up here to...
Hello, thanks for the great talk. I have a question about the source of your trajectories, and I was wondering if you can apply your method to select the best trajectories to avoid collision, for example. In the case you have trajectories generated by a neural network. So if you have trajectories for each agent generated by a neural network, and you can choose between different trajectories, can you apply your model to select?
Yes. So indeed here you see that there's complete decoupling between the part where you generate or propose trajectories and the part where you actually do the scoring of these trajectories. So in principle, you can use any method to generate trajectories. The important thing for this to work well in practice is that you generate trajectories that have a good coverage of all the possibilities. So in some sense, they are heterogeneous, right? Because clearly, if this module were to propose just one trajectory, well, there's very little to score, and your behaviors are going to be very poor. But instead, you want to have something which is, to some extent, a component which is unbiased, which covers all the possible behaviors. And you can also merge it with learning schemes that actually throw in some suggested trajectories that you learn from data, right? If you have a module that just from raw data propose, okay, I think this is a good candidate trajectory, And in this way, even if that model works 99% of the times, the time that it predicts or gives you a bad suggestion, you're still covered because you're going to have the other trajectories that are going to be scored or preferred to the one that has been proposed.
All right, I think let's thank Alessandro again, and our next speaker can set up.
Thank you.
Okay, so
And then
you're on the Zoom.
I am on Zoom.
Just need to plug this thing into that, right?
Well, I'm not sure. We might need to keep it connected because of the stream.
That's fine. If I can just put this here, like, isn't going to share whatever is being
Okay,
shared.
yeah, that'll
We're
work,
good.
yeah.
Cool. I'm just going to.
Okay.
Scoot it a little bit. Fantastic. And then I can turn on my video. If I take, is it this microphone?
Both microphones are
every
useful.
microphone.
Okay, great. I'll just take my mask off and it'll be okay.
Okay.
Yeah, I think we can point it.
I'm currently muted also.
Optimize your video, not your sound.
Do you want to be muted?
Okay, that's right, good call. Actually, my mouth is actually muted, so I always have extra.
But all the mutes.
All the mutes.
Okay, that looks good.
You're seeing screen? Oh, hang on. Yeah.
And there's just a lag.
Okay. Yes.
Great.
Okay. Okay, so we're now going to move from artificial to organic agents, and our next talk will be from Alison Scrow, a group leader at the Howard Hughes Medical Institute's Danelia Research Campus, and she's going to tell us about the mechanisms by which independently behaving cells can create collective behaviors.
Great. Thank you for the lovely introduction. I want to really change gears here for a second and give a different kind of talk than the previous two. So as Kristen said, I'm really interested in in cells. So we're focusing on a totally different kind of agent, a biological agent, and I want to understand computations in there. And I also want to give a different kind of talk in that there are no answers in this talk. We have solved one set of problems in terms of how do we even see these behaviors. And I'm really excited to have good discussions about how we can think about the next steps in terms of analyzing them. So just to sort of help you think about the way that I see these kinds of problems and get into the world of cellular computation, cellular behaviors, I want you to imagine that a cell is an agent, right, and it's performing computations molecularly, right? Inside cells, there are chemicals, right, and these chemicals change concentrations in time, right? These can be chemicals maybe you've heard of like calcium in the brain, basically X, Y, Z, doesn't matter what it's called, but there's some chemical that changes its concentration, and the dynamics of those changes encode information. Right, and I'm really interested in what those dynamical motifs are that encode information, because single cells do a lot of work with those dynamics. If I'm a cell, I take in information about the environment. It can be mechanical, I can be pushed on, I can receive a biochemical signal from another cell, maybe I can receive an electrical signal. I encode information about those signals in the dynamics of those molecules inside of me, and I use those dynamics to make decisions about what to do next. Those decisions can be things like gene expression, right? What's gene expression? Right? I'm making a new protein or thing. I'm making something to do something else. I can change what kind of cell I am by changing my gene expression. I can change the behaviors I can do. Maybe I can move faster. Maybe I can become a type that contracts better. I can secrete and send signals to other cells. And I can also use those dynamics to tell me to move. Right? So I have a lot of different things I can do as a single cellular agent. And if I have a bunch of different cells, we can build up multicellular behaviors. I can relay a signal across a long distance as cells, right? If I am able to pass along a signal that I secrete, I could even move as a group. And it's those multicellular behaviors that build up what we think of as emergent behaviors in biological systems, right? Things you might think of as wound healing, right? When you cut yourself, a bunch of cells need to decide how do we work together, right? And over time, really tightly integrate a bunch of different behaviors in order to heal that wound. I'm going to focus on a microbial problem today, microbial development and aggregation. Right? How do microbes deal with stress in their environment and integrate information about that environment and decide what to do in order to face and deal with adversity? So the system I'm going to be speaking about today is Stichiostelium, the social amoeba. This is a cellular slime mold. It lives in the dirt. And when it runs, it eats bacteria in the soil. All right, and once out of food, it makes this structure you see here on this screen. So this is a piece of mulch. These are a few millimeters high. So if you're a big hiker, you might have maybe seen these out in the wild. I want to show you how this process happens. All right, so these exist as individual cells, and they only make this structure when they run out of food. And this is a very dynamic process. So think back maybe if you've ever been in a biology lab, or even if you just have a very messy kitchen. Maybe you've got a surface that grows a bunch of bacteria or fuzzy stuff. All right, this side of the screen here on the right, that's fuzzy stuff. That's just bacteria on a plate. This is what they love to eat, so we've given them a ton of it. So we're now on a nice 2D surface. What you're going to see here, and I'll play this video in a second, this is a feeding front, a ridge. This is where the slime mold themselves, you can't see any of the individuals, you're just going to see group behavior, are gorging themselves on that food. And then back here is the starving cells, and I want to start playing this video for you. Right, so this feeding front is moving forward, and at the ridge they're interacting with the bacteria, eating it and gorging themselves. But at the back of the ridge, they start to starve. And when they start to starve, they turn the machinery to make a small molecule signal. I'm only going to talk about one molecule today. It's called cyclic AMP, but that thing they make, when they receive that signal from their cells, they round up, and that's how you see these waves. Right, and these waves are coherent, and they will chemotax up these waves and move to this chemical signal. So they say like, oh, hey, the wave's coming from there, I want to go up that. And that's how they find each other in these groups. All right, and when they get into these groups, they need to make a decision. Some of them are going to become the cells that make that stalk that hold everybody up. Those cells actually make a commitment to death. They die to hold everyone else up, those spores, so they can go to an area with more food. All right, so this is an incredibly important process, life or death, that's evolved in order to allow them to, basically sustain life when they run out of food and go to new areas with more food. So I want to take a moment to go ahead and eavesdrop on how they're thinking about this process and show you a little bit more about what we know about how they behave as individuals and what these groups look like up close. So I told you that cells can sense and secrete signals to one another. So we can both see how they talk, and we also have an understanding of how, we can see how they think, and we can also have an understanding of how they talk. So something I want to emphasize here is what you're seeing and what you're going to see in these videos is how cells are processing information about the signals they receive. You can't see the signals themselves. But what cells do is they sense this environmental chemical that they send to one another. They respond with their own internal spike. They're excitable cells. For those of you who do neuroscience, they actually can be modeled very similarly. They release that signal to the environment so that they can tell the next cell, hey, I got a signal. And then they'll actually elongate and chemotax. They'll move toward what they think that signal is from. All right, and so this drives their movement behavior at the single cell level. And so what you're going to see here is, again, each little sort of individual thing here is a cell. Many of them are overlapping, but they're about 20 microns in diameter, so you have about 10 of them in this length here, just to give you a sense of scale. And what you're going to be seeing again is their intracellular thoughts. So this yellow amount is low, and this red amount is a high level of the molecule. And so you're seeing how they're able to process that signal that they're going to spike and then release and send on to another cell. All right, so this process starts to happen only when you remove food. All right, so they'll stochastically begin to generate spikes of this molecule. And so initially you'll see those small little areas where it's coming out and beginning to propagate. And over time what happens is the waves will travel longer and longer distances as they're able to send more and more signal. All right, and the cells will start to move toward that signal. All right, and stream up those streams. All right, so you can see they even change shape and you get this elongated shape and it draws many of them in. And again, this is how you're going to be able to go in and these centers pull these cells into this group. And it's inside, again, this group that they're making that decision of what kind of cell to become. And so we know a lot about this. I've been able to show you that we can even go ahead and eavesdrop on how they think about how to do this. We know how this process drives motility in single cells. We can watch single cells crawl in a dish. We know how they make decisions, how they shape their space. But what we don't understand is how this works really in natural environments. So everything we know about this process comes from a 2D environment, right? I've shown you these waves. Here we go. I've shown you these waves, and you can see them, and they form this beautiful coherent pattern on a dish. Right? But that's what happens when you spread them out in one dimension. And they will, of course, recapitulate this process and make those beautiful fruiting bodies. Right? But what actually happens in the soil where they evolved? Right? This is a system that's evolved over millions of years to be able to cope with adversity in a three-dimensional environment. Right? They have to come together, find each other in the soil, crawl out of the soil, and generate these fruiting bodies up above where an animal can carry them somewhere with more food. right? So how does this work? We know it happens, right? You can find these if you go out hiking. There we go. But how does this actually happen in nature? How do you see this, right? How do you get a video camera in the soil and look at this? So we wanted to find a way to see how environments like these 3D environments shape multicellular behaviors. All right, not just this sort of 2D, perfectly synchronized flat surface. But I want to sort of outline the challenge to you. Like, why haven't people done this before? Why haven't you seen this happen? So if you think about it, these environments are heterogeneous. You need to capture a lot of different complexity that's out there in the environment. They're three-dimensional, so you need to be able to build up layers upon layers of this environment, so it's not just I can like put one pattern down or a gel. I need to be able to get extra dimensionality here. I need them to be transparent in some way that I can see them. It's nice if it's visually transparent, where you can get light to go through. These cells aren't super warm, but maybe we could sense heat if it were, thermally transparent. But again, you need to have some way to see through it. We need to have some means to sense what's going on. We need some way to see what the environment looks like, so visualize the particles in the environment and see that separately from space, and also see that separately from the cells themselves. So we have multiple things we need to see and identify as separate, so that we can understand how they interact and what kinds of behaviors happen with certain objects or without certain objects. Ideally, we'd also like to be able to visualize signaling behaviors, how are cells thinking about the behaviors they're going to perform. One of the challenges here with all of this is that every interface, particularly the interfaces with where particles are, scatter light, and also the cells. So we have challenges of how do we sort of account for what interfaces are there, and is there a way to minimize those interfaces? We also need a system that's survivable for life. If you think about it, you probably have a lot of demands in order to live, right? You would like things to be humid, you need access to water, you'd like things to be a certain pH, you need some oxygen. Cells are the same way, very demanding. And we'd also like a system where cells can actually grow and move naturally and behave in a way that we think mimics how they behave, right? You as a human don't like to exist alone, you need to exist with other agents, you need to be able to find food, feed at certain times. Cells have the same kinds of demands. We want to, again, recapitulate this entire natural system. So the way we're able to actually do this is by taking a natural mineral that's known as an ice rock. If you have some friends who are into creating good vibes and energy, cryolite is something that you can find on Etsy if you'd like to buy. It will really improve, apparently, the vibes of the area you're in. But it also is fantastic in that if you grind it up, you're going to get these small particles that have a refractive index very similar to water. And what that means is that you minimize the interface that light bounces off of, and so that you can easily see through this mineral and see how things are behaving on the other side of it. Right? So it's a trick to basically have almost a soil-like thing made out of glass. Right? It has really desirable pH and other properties, so it doesn't make the environment toxic to cells. So they can live there, they can have that access to that oxygen, cells can grow. All right? But it's, again, like we've been able to create a transparent soil that we can just see through. And so I want to show you what are some of the cool behaviors they do in these environments so we can start to think about how to categorize them. So again, I want to remind you, so we've got this sensor where we can see how cells think. We are now also able to visualize the extracellular space, right? So what you're going to see here is that the media, the liquid they're in, is going to be blue. We put in a dye, and that the cryolite, the particles, the soil are going to be dark. Right, so you've got now three colors, low and high signal, but the cells are all going to be red or yellow, and then we can also see again the space they're crawling in, and the particles will be dark. And so what I'm showing you here is a sort of top-down view. The joys of Wi-Fi, where everything is slightly asynchronous. There we go. This is a top-down view where you can see again these particles, and these are maximum intensity projections of z-planes. You can see that they're going to be moving in up and down in Z on the sides, and then you can see how they move in X and Y in the big square. And you can see that single cells, this is over even just a few minutes, you can watch cells crawl around, move, and they actually take advantage of all three-dimensional space. All right, so I've shown you that we can get cells crawling around a little bit in a few cells for maybe 15 minutes. But I put together this challenge, like what are the behaviors that happen naturally over days? How do you survive? So can we even look at that? So imagine you've got one of these microcosms, right? You can make them relatively large. This is now millimeters across. This is a 500 millimeter scale bar, so you can see those chunks. What you can do is you can start and seed some cells and bacteria in there. So these cells, again, are these yellow particles. And the yellow particles are going to then eat bacteria and multiply over the next day. So we can get many more cells growing, right? Here just the main behavior is replication. They'll crawl around, look very similar to what I showed you in the previous video. They're not really working together, right? Again, these are things that only act as basically collective agents when they don't have access to food. They turn on this program to work together. They can turn this program on in this system, right? They eventually do run out of food at about between 30 and 35 hours here, and they start to send each other those waves, right? And those waves can actually be huge, coordinated across the whole ecosystem. And those waves will drive aggregation into those kinds of structures we saw before, right? And so we see that across the whole system, you'll start to get aggregation centers where they work together and come together here, here, and here. They actually also love to crawl out. They do have a program where they're basically designed to try and find where's the most oxygen and get up. So they will eventually escape our eavesdropping. And this is totally reproducible. You can change the particle size and even see interesting different behaviors here. So now if we scale up to sort of larger soil, you can again recapitulate this environment where they'll start to grow, eat food, replicate. They're now starve and are turning on their collective program, and you can see that they'll generate waves to communicate with one another and aggregate and start to stream together into those groups that I showed you in 2 dimensions. This still happens in 3D. So one of the big questions I have is that the main thing I focus on is, again, information processing. What are the dynamics of that process? What are the sort of solutions that you have in 2 versus 3 dimensions to how you can transmit information? And so we really want to understand, what do those streams look like? What does the information transmission look like? So let's go ahead and zoom in and see if we can see what this looks like up close. So we can get we can zoom in a little bit here just to show you, I want you to be able to interpret these videos a little bit more. So in all the videos I'm going to show you now, again, yellow and red are going to be looking at the cyclic AMP concentrations. So this is again, their information processing going on. The blue is going to be the media. And so when you see these overlaid, you're going to see sort of these dark-ish areas or sometimes redder areas are going to be the ones of high levels of cyclic AMP, and then the lower ones are going to appear as yellows, just to sort of orient you to the data you're going to see here. All right, so what I'm showing you now is a video that's going to loop, and we're going to look over tens of minutes, and it's going to go deeper and deeper into a microcosm. So you can see the same field of view, but I'm showing you what it looks like at different depths. And what I think is really exciting about this particular video is that we've got 2 different kinds of behaviors going on. Over here in this area, we've got feeding. Right, so these cells are still eating, they're vegetative, they're crawling around, behaving like single agents, yet over here they've run out of food very close by and are starting to work together. And so you can see this stream go along, and actually as you watch at depth, it's going up and around and over different things, and it's basically weaving along, right? It doesn't get, when it runs into an obstacle, it goes up and over a little bit. All right, and again, it's still able to transmit information back on the stream and pull along cells with it. But actually, it doesn't transmit information very far, right? These cells over here that are still feeding don't receive that signal. All right, I just want to sort of zoom in for a moment so you can see that interesting 3D branch structure, how they move around different obstacles, right? You can see, again, there's a particle here that's sort of pointed out, and there's actually a branch that comes together, we think. All right. So something we also thought was really interesting in this system is that in 2D, you often get these very large aggregation regions. Cells want to actually band together in as big a group as possible. You can have up to 100,000 cells in one of those structures. If you think about it, there's advantages to that. You can move faster together as a group. You can also make a larger structure that gives you greater height above the soil. And so there's a huge advantage to that. But here we actually find and we see a lot of localized aggregations. So again, we're going to step through, as it gets darker, you're just getting farther into the microcosm, and we've chosen to preserve that loss of light just so you can have a sense of depth. But we actually end up with 2 aggregation centers here that are pulling cells along from 2 different rocks. There seems to be something really interesting about having a large rock in an area that makes it very attractive to cells. All right, so you can also just sort of look at an area where things are just moving along. And initially, this video looked like maybe, there we go, synchronizing now. The joys of the lag, there we go. Initially, it looked like there was not a whole lot going on here, right? There's just some cells moving along, but this video is actually exceptionally rich. So I want to point out a few different things. So we're going to zoom in on one area of this video. So the first one is over here. So we've got this rock, and this is the same rock in multiple dimensions. Right? These cells want to move along and interact with the rock. So they sort of crawl along them, will often sort of step off and interact with the different things in their space. Right? So they can actually be taken away from a stream or potentially join a stream from where they're interacting with a particle. The other thing I think is really neat in here is that you actually have branches that come together in these videos. So we have 2 different branches you can see here that come together and crawl along this rock, and then you can see how they stream up and over this particle right here. One of the last things I thought was really cool that we were able to see that people had no idea happened is that cells actually remodel their environment. Right, so again, all the work done to date has been in an environment where cells are fixed on a 2D surface. Sometimes people do put obstacles in, right? But if you're trying to have a really uniform substrate, you really don't have anything for things to interact with. We don't put in obstacles. We don't put in things that they can remodel themselves. But it turns out if you give them a naturalistic soil environment, they will actually remodel it. So check up here. There is a piece of soil, and this group works together to drag it away. Right, so this is like we have cells almost being like ants or some other, small animal that's able to collectively carry this chunk of soil with them. So this was not something we expected at all. That we were very used to the kinds of collective motility behaviors I talked about, the idea that they stream together, that they'll differentiate into different cell types, the idea that they work together to remodel their environment as part of this process. We have no idea what function that serves, if it does serve a function, how often does this happen. But this was a very exciting thing for us to see, and it really opened our idea to the idea that there's new behaviors out there that we should be thinking about a sort of behavior agnostic approach to thinking about this data. So part of what I love about workshops is that they're great for having discussions, and so I want to put you sort of where things are, and then what are things I'm excited to talk to all of you about, sort of out there today. So in terms of experiments, we have a lot of ability to control this kind of system. So we have the ability to control things like cell density. And I mean that in several different ways. We can change the number of cells we initially put in. We can also change the amount of food we put in so that we can titrate how long they'll grow for, how uniform the growth conditions are, and how many cells you have when you starve. Right? So that's already known to be a parameter that controls how big a group you can get and how dense it is. These cells like to be close together, and so you can draw out the whole process and create more heterogeneity by having less cells, because you'll be relying on the cells themselves to create a uniform area. And they don't need to be super uniform, but they need themselves to be relatively dense, and so they'll work to create that if you give them less cells to work with. We also have the ability to control the environment itself and particle size and introduce heterogeneity here, and I'll talk a little bit more about how we think about that as a really key potential modulator of how groups work together. And we also have the ability to control a lot of cell properties. So something I think is really amazing about this system is that we can actually control how the cells think. So I showed you some natural cells and how they're thinking about their environment, but we can use mutants that we can turn on with light to talk to one another and send signals to other cells. We can also use mutants that can't receive signals from other cells, and we can mix all these cells together so we can control basically how are the cells thinking and communicating with one another in this system to test predictions about what's important to drive behaviors. We also have access to a huge library of natural isolates, naturally isolated slime mold that have very different signaling features. So all of these cells are going to make those waves, but they're going to make them at different frequencies, and they're going to make them over different time scales, and coordinate this process differently, and they're known to work together. So we can actually tune heterogeneity of cells in the environment to control, again, their thinking and communication. We are able to do some modeling of these cells, and these are actually, there's a lot of nice mathematical techniques to do this. I sort of hinted at everyone earlier. I know there's some class of neuroscience in the audience, but when I sort of expand on this idea, single cells are excitable. What that means is that I can stimulate them with a signal from another cell, and they'll respond with their own signal. They'll also propagate that signal along. So there's a lot of nice mathematical techniques to think about how to model how these cells think and propagate information. Those techniques do work here. I'm not going to go into a ton of any detail unless you'd like to chat afterward. But basically, established models that can recapitulate excitable systems can recapitulate those waves. And we are able to start to build up basic simulations of how those waves would work in an environment with different particles in them. Just to sort of briefly walk you through how you can sort of think about this idea that I keep bringing up particles in an environment. If you think about the world from the point of view of a cell, right, you can have particles that are similar to you. And if you have particles that are similar to you, you can sort of model the soil as like a defect, all right, in the way you're propagating information transmission and where you're able to crawl. It's like you had a grid, you deleted a square, so you can't signal into that square or move into that square. You can think about the soil that way if it's roughly similar to your size. If you start to scale the soil up and have intermediate-sized particles, these soils create channels, right, that cells can move through and signal through. So you now have an interesting topology of sort of, almost tubes that cells have to work through and communicate through. So that sort of opens up a new regime for how do you think about modeling this. And finally, if you scale up to particles that are much larger than a cell size, the particles themselves actually just create large irregular boundary regions where you can actually have sort of segregated areas where the behaviors you're used to happen in terms of how do cells work together and how do they transmit information, but you've just sort of cordoned them off from one another and given it an irregular shape. So that's sort of one area if we're pushing things in terms of modeling. But the thing I'm most excited to talk with people about here are how do we think about analyzing these behaviors. So something that we were not open to originally was we thought we sort of knew the behavior space. But there are a lot of techniques for how to think about, what are new behaviors, right? So again, I want to really draw the contrast here. If you think you sort of know what behaviors are, you're often looking for a way to sort of say, okay, that behavior has happened, I want to classify or mark that moment. We're now much more excited also to think about, are there ways to sort of map a behavior space and identify new behaviors that we're not able to call out and see ourselves easily. So I'd love to chat about the latest innovations there in this area that I might have missed. Because we're really interested in sort of understanding how do environments change behaviors, right? Again, we can tune this and we can go back and forth and build up new experiments to test predictions, but that's sort of the biggest question we really like to drive with our analysis, as well understanding how do these cells compute together to work together in these situations to drive these new behaviors. So I think I'm getting close to time. So with that, I want to thank the team that drove this work, in particular Dr. Emily Hager, who did all the experiments you saw here, as well as my team back at Boston University that helped support a lot of this work, including Dr. Chiara Katam, some collaborators. We just actually moved, so if you're excited about talking about cellular information encoding and mixing computational work and experiment together, we're looking for postdocs. And since I know a lot of people aren't here, I'm really excited to reach out to chat. I'm here all today. I'm also around basically all the time. Please ping me on the Slack or send me an email. I'd love to have more interactions about this work. So with that, I'll take questions. And I think I can also see the chat. So if people have things in the chat on Zoom, I've got an eye on you also.
So I can ask the question to start us off. I was wondering, so you kind of talked about the cells following different programs at different times. Is this behavior very discrete, like there are these discrete changes, or is it a more continuous space?
So I think the way I would think about it is at different times they're capable of, they don't change their behavior massively. I guess they serve a few different classes of behaviors. When they have access to food, they have an individual foraging program, and they don't really respond. They don't have a way to really respond in terms of behavior that you see to cues you get from other cells, and that persists early on into starvation. In the first phase of starvation, they're very sensitive to chemical cues. They're looking to find each other, and so they have a lot of really directed motility to try and come into contact with one another. You can sort of see this, I think, in some of the videos, but I know that there's a lot of lag, so I'll sort of describe it to you. in the sort of intermediate stage of starvation, they start to get sticky and they want to touch each other. And we get a class of behaviors where they start to stream and their individual, the sort of way that they want to work together as a stream together. And then later they turn off that stickiness, cover themselves up. I didn't show a lot of this here. They actually have a group motility mode called the slug that they'll use to crawl around and actually crawl out of soil. So they've got a few different classes of things that happen over time. But most of them are, again, driven by the amount of individual motility you have. I'd say the main key parameters that change things in cells are stickiness and then your ability to move, how fast you want to move together. I see on Zoom, so for people in the room, in reaction diffusion systems, it's known that the emergence of different kinds of wave patterns depends on the boundary conditions. The question is, are we planning to observe changing wave patterns as the shape of the environment has changed? The answer to that is absolutely. So that's part of what we think is really cool about the different particle sizes. I showed you a sort of a snapshot of a simulation on one of the slides, but I know that probably when we went through it really fast. We still see the wave patterns when you have defects, and those work pretty well. We can see a little bit of waves as we have more channel conditions. We still see the wave patterns, experimentally, and we have this sort of these environments. What we haven't really gotten to in terms of modeling is being able to understand sort of the different kinds of wave patterns that we see as we change the actual boundary conditions and sort of squeeze space down differently. So that is something we're definitely hoping to explore. We sort of see some differences. Something we think is interesting is that oftentimes those wave domains should drive where you get aggregation, and so we know that we must have some interesting effect of boundary conditions on later stage waves and thus how the cells aggregate that we aren't seeing in our movies just yet, and we're hoping some modeling can help us understand how those waves are being affected by the sort of smaller areas, since we do see the big waves as well.
Hopefully keep up the discussion, and we have a break until 11 o'clock.
Do you know how to unpin someone? I think it's good in my video. How pretty close movement? Mm-hmm. How so? In
can you have a bunch of agents that you can just predict their trajectories?
Not really.
Okay.
I guess the key thing is if you have so you can sort of, if you know what the sort of wave pattern is, you can predict where they're going to go. I think there's a couple of things that we don't have. One of them is that the cells have a refractory period, and so we don't have a good idea. For example, I can show you a movie of this, but we don't know how they choose between two different areas. Mm-hmm. For example, a wave comes from here, a wave comes here, and they'll go back and forth and back and forth and back and forth. And eventually they'll commit. Mm-hmm. And I think the thing that dictates that is actually stickiness. At some point they probably stick. Mm-hmm. And people don't have good models that integrate both the signaling and stickiness, so they just lose that. So that's, I think, one. Basically, the answer is yes and no. In the most basic sense, yeah, they'll go where the chemical is, but at some point, since you don't put stickiness and chemicals, then you lose that decision point.
Mm-hmm. And I guess when you have a population of them, if you're trying to simulate all of the agents in the population, then eventually, the collection, it might be hard to simulate. It might amplify the kind of uncertainties.
Yeah, we've been trying to figure out how much you need to simulate to really get something out of it. Okay. For example, 2D is not so bad, right? You can scale it up relatively computationally quickly, but in 3D, it's been hard for us to figure out what is important to put in, and as you rapidly are taking up more and more time and computational power, it's are we learning anything new? Mm-hmm. So we can get it working in 3D and start to see the waves in 3D, but then we don't, we don't have things but we haven't even actually added on cell movement onto this.
Okay.
So people who tend to focus on the signaling part don't have motile agents, and so we finally have the code set up that we can have motile cells moving in the concept of where they're going to be, but we're now trying to figure out it's a sort of how much complexity do we need to get there? So we're also just not there.
It would be fun to work on this if you want to.
I would love to, yeah, very much so. So actually, I think you talked to Emily when she visited. She's the one doing a lot of this.
Yeah.
And we've been talking a lot about what do we need to figure out who to work with on what, because it's just the two of us right now.
Yeah. Did you have a question?
Yeah, actually I do. So it sounds like you can control the way the cells can communicate to each other. Can you, I guess it's kind of like, in real life, do they ever send the wrong signal, and can you force them to send the wrong signal? How does it affect them?
So we can't make them send a signal, like the wrong signal in terms of like, we can't force them to send another chemical or something. What you can do is you can force them to signal at bad frequencies, for example. Like I can show you some videos, like we have optogenetic control over selectivity. So you can force them to signal too fast or too slow. It's interesting, as a group they're relatively robust to signaling too fast, right? It's interesting, like as long as they get some, basically the issue is, right, because they're excitable, if you don't give them enough rest time, you can't make another spike. But if you drive them too fast, they'll make enough of a little bit of a spike that, you know, because they won't make a full spike because of, I don't know much you know about nonlinear dynamics, but okay. Basically, if you're still like unable to signal, you can make a little bit of a stimulus, but you can't make a full one. But it somehow seems enough in a collective that it passes it on. So you can sort of make them miss signal like that. You can drive them too fast. Other ways you can make them miss signal are you can trying to think about how to reframe it this way. You could make everybody not able to make a signal and totally control them yourself. I've done that before. The other way you can think of this signal is actually mentioned as natural isolates. Also there's different kinds of cells, different waves, different wavelengths, and they have different expected patterns, different timings. And so you can think about mixing of these other way of like having this signal. Like, they can talk to each other, but it's at a slightly different speed, and they're expecting different, they're going to do different things at different times. So it still works, but that's a way of creating another form of like, not bad communication, but like not.
Ori, are you there?
Yeah. Okay.
No, go ahead, sorry. I think Ori was.
Yeah, I'm just thinking of like, I think Twitter is
You might be muted, I think, currently. I can hear her, yeah.
Hello. Good morning, everybody.
Hello. Did you want to try sharing your screen?
Yes. Let me do this quickly. Okay, so I'm gonna try to share the slides. And then the question is, will it work? And can you see them progressing and see a movie playing?
Yes, that looks great.
Okay, yeah, great. So, so I think that's what I'll do. And should I unshare it until start? Or
no, what you can keep it shared because I think you're the next speaker. So yeah. So just we can just leave it up there for a couple minutes.
Yeah, perfect.
So we'll start in maybe 3 minutes, something like that, if that sounds okay.
Okay, yeah, I'll be here.
Okay. All right, are you ready?
Yes, I'm ready.
Okay, so go ahead. Okay. All right, so now I'm pleased to introduce Orit Peleg, our next speaker. So Orit is a professor at University of Colorado Boulder, and she's going to be telling us about communication between groups of animals.
Great. Thanks so much for the introduction, and thanks for inviting me. I'm sorry that I'm not able to be there in person, but I really enjoyed the talk so far, so it's great to be able to join remotely, and thanks so much for putting this nice event together. So yes, I'm going to tell you about, as the title suggests, dense firefly swarms. Let me just take a very quick couple of minutes to tell you a little bit more broadly the kind of topics that my lab is interested in. Oops. Let's see. Okay, so as Christine mentioned, I'm interested in animal communication, and I think that as humans, we're kind of used to being able to communicate with each other over very large distances. Now is a perfect example of this. I'm talking to you from Colorado, and we have a bunch of other systems as a backup, like postal mail, satellites, and so on. But animals need to be able to sometimes solve similar problems, communicate over large distances, using only the resources they have at their disposal. So there's a lot of beautiful ways that animals communicate with each other, lots of different modalities, tactile, visual, and so on. My lab is really interested in understanding how these communication signals are generated and interpreted, thinking about physics and computer science terms like detectability and compression and robustness. We use a lot of as tool mathematical modeling and behavioral experiments. The talk today will actually focus mostly on gathering data, but it's very much motivated by thinking about modeling these collective behaviors. And the model organisms that we usually use in my lab are insect swarms with honeybees and fireflies. I think we'll hear about honeybees later in this meeting, but I'm going to focus on fireflies. The reason we work with these two organisms is that very similar to what Alison told us before in the previous lecture, just by looking at them, we can tell something about the internal state, computation that they make, and how they communicate with each other. So I'm going to focus on fireflies that have this really obvious communication signal. There's an organ at their abdomen. It's called the lantern, and produces cold light. As you can tell, this is kind of like a light on, light off signal, so we can almost think about it as an approximately digital signal. It is being produced usually in very large groups during the mating season of fireflies. And it's a very complicated communication setup. So every species of firefly has its own species-specific flash pattern. I'm just showing you a couple of examples here. There's thousands of species of fireflies, actually. Males have a particular signal, females have a particular signal. And somehow they need to detect each other's signals in order to mate with each other. But the point that I want to make here, and why I decided to focus on fireflies for this talk, is that this is, as I already mentioned, an approximately digital signal. It's almost like Morse code, and it might be as close as it gets to computer language as it gets in the animal communication. So that's the problem that fireflies need to solve. This is how they communicate with each other. And as I already mentioned, they do this in very large groups, which poses lots of visual clutter. And for the neuroscientists in the crowd, that's a manifestation of the classical cocktail part problem, where you have to know who is producing the signal and being able to sort of shut out the rest of the room in that sense. So fireflies deal with that in different ways. There are certain species of fireflies where we think what's happening is that they produce flash patterns that become more detectable at the expense of other species. These are non-synchronous fireflies, and this is the movie that is hopefully playing on the left side. And then there's another group of fireflies shown in the movie on the right side that in order to deal with this dense, congested communication, noisy environment, they actually synchronize their flash pattern. And that has obvious implications for increasing signal-to-noise ratio and so on. So for the rest of the talk, I'll focus on these kind of fireflies. And let me just tell you a little bit of the origin story of where this all started from. So synchronization in nature is actually a very common behavior. It occurs in many different non-living systems as well as living systems. And my background is actually in physics, and as an undergrad student, we used this textbook written by Steven Strogatz, thinking about all the different beautiful dynamical systems that occur in nature. Specifically for that physics class, we talked about how electrons synchronize their motion in order to achieve superconductivity. And Steven Strogatz, who is a very good science communicator, actually mentioned the example of firefly synchronization as a relatable, maybe less heavy kind of example that students can relate to, which I really did relate to. And I really remember that moment in undergrad class where I realized that some of the models that explain how electrons synchronize with each other can also eventually explain how fireflies synchronize with each other. So that problem has been dormant at the back of my head, and when I started my own lab, I just had to work on fireflies. So let me tell you a little bit about the classic mathematical models that were discussed in that book, and that till today they are very widely used. The main assumption of these models is that each agent in this multi-agent system is producing some periodic behavior. It can be a clap, it can be a walk of a person, and it can be a flash on, flash off of a firefly. So that periodic behavior can be characterized by a phase, which is plotted here on the y-axis as a function of time. And if we see this periodic function, then we know this agent is behaving periodically. The most basic way to couple the behavior of pairs of agents in order to achieve some global synchronization is shown here in the equation on the left side, where the phase of an individual agent theta I depends on its own frequency, that's omega, and then it is kind of coupled to all the nearest neighbors that this agent is able to interact with, driving the phases of all the individual agents to be similar to each other. And there's an important element here, k, that's the coupling element. It can be a constant, it can be a function of the distance between the individuals. It can be a more complicated function of the distance between individuals. And actually, this is not, to scare anybody, but I just want you to get a sense of how many different ways there are to write these kind of models. And of course, every time you change a little bit the local interaction, that might lead to changes in the global behavior and global synchronization. And the problem is that there was very little quantitative data to actually validate these models with fireflies, which is the gap that me and my lab members were hoping to close it. So to start gathering data about fireflies, we went straight to the source. There's lots of beautiful synchronous fireflies at the Great Smoky Mountains in Tennessee. We worked with Photinus Carolinas. These are fireflies that have a burst structure. So they flash a couple of times on, off, and then they're quiet for a while, and then again, continuing the burst structure. And here is the movie that I hope you can see of one of our camera views showing this swarm of fireflies. So what we just saw was a burst, now it's the quiet part, and here's another burst. And the fireflies basically continue doing this for a couple of hours every night during their mating season. And the point that I want to make here is that this is almost embarrassingly traceable. As you can see, there's very good contrast between the flashes of the fireflies and the background. To contrast, maybe other more complex environments where we don't have that good contrast. So our analysis is actually pretty simple. We kind of threshold the image based on brightness, and then we find the centroids of the flashes, and then this gives us the location along this two-dimensional projection of the three-dimensional system of where the fireflies were flashing. And there's a lot we can learn from this temporal data. For example, here we can kind of see the onset of synchronization. I'm showing you here a plot of the number of flashes as a function of time. At the top, that's in a scenario where there's low density of flashing fireflies. There isn't an obvious pattern. And then as the density of flashes increases, then we can start seeing the burst structure that I mentioned before, where lots or several fireflies are flashing synchronously at the same time. And we can actually characterize this a little bit more properly using some statistical models, assuming a Poisson distribution for a totally random process versus something that is more synchronous, like we're seeing here at the high firefly density, and kind of find the point where the system switches and snaps into the synchronous state. But as I mentioned, in the models of synchronizations, distance between the fireflies can be really important. And that's something we cannot get from just one camera view. So for that, we actually needed to employ stereo vision with 2 cameras. I'm showing here on the left the more traditional approach to set up stereo vision, where we have 2 external cameras kind of looking at the swarm from the outside. We actually used, in some of our experiments, a slight variation of this, where we use GoPro cameras that have 360 field of view, and we place them right in the middle of the swarm. So in a sense, we can kind of get the recording from the perspective of the individual fireflies that is inside the swarm. We had to develop a little bit of new math to be able to solve the triangulation or depth estimation here, because now our spatial variable x, y, and z are actually, or x, y are periodic because we're working with 360 cameras. So I'm not going to go too much into details, but I'll just say that the method is freely available to anybody who wants to use it in the reference here below. And using this method, this is the kind of 3-dimensional reconstruction we can get of firefly swarms. So here again, you can see a burst. Now it's going to be quiet for a little bit, and then the firefly are going to start flashing again. And this is now in 3D instead of 2D. And so we can do this 3-dimensional reconstruction throughout the course of the night, over several nights. And we can start calculating some spatial-temporal correlations between the locations where the flashes occur. Here is an example of such a spatial-temporal correlation function, where on the y-axis we see the distance between the flashes, and on the x-axis we can see the time between flashes. And this is great because now we can actually start feeding all these classical models with data, with empirical data about how the distance between flashing fireflies actually determine the coupling between them. And this is on a fairly short length scale. We can also see interesting spatial correlations on larger length scales. Here, for example, it's on the length scale of about 30 meters. And what we're seeing here is that the first fireflies to flash in these particular situations were actually closer to the camera, and there is this big wave that propagates away from the camera, where fireflies further away from the camera start the burst, and then further and further away, and we can capture that with our recording method. What this tells us about the synchronization process is that when we compare the propagation speed of that wave to the speed of flight of a particular firefly, it's actually an order of magnitude higher. So the wave cannot be explained by movement of fireflies. It has to do with something of a social interaction, visual social interaction between the fireflies. And then something else we can do is actually track individuals as long as they don't move too much between flashes. So here is an example of that, a couple of these trajectories. We can start actually zooming in and asking questions about the interaction between fireflies in smaller groups and how that might lead to global synchronization. And one thing that I would want to point out here is that we noticed that some of the assumptions of the classical models that I mentioned such as that each individual agent is performing periodic behavior, don't always hold these fireflies. And let me show you some data that support that statement. So we're going to focus on the time between the start of one burst to the time of the next burst. If the process was completely periodic and agents were completely periodic, then we should expect to see a characteristic time that is prevalent in large populations. And we see that outside. We see that for large swarms in the swarms outside. However, when we do some more controlled experiments where we bring smaller groups of fireflies into these big tents that are visually occluded, so they're separated from the bigger swarm, we see something different. So let me guide you through these plots. What you see here is a probability distribution function of the periodicity of the flash in bursts. At the bottom here, we see the situation for a large group of fireflies, those are 20 fireflies. And we can see that there's a characteristic peak around 10 seconds, which is more or less also what we're seeing outside for the larger amplitude swarm. But as we decrease the number of fireflies in our system from 5, for example, then this distribution becomes wider, and it even becomes more wide for one firefly. So perhaps thinking about individual fireflies as oscillator is something we need to update, and we're currently thinking about ways to extend these more classical mathematical models to include some of these observations. And then lastly, I want to point out that this is very new work that I'm excited about. We may have found evidence for chimera states. And for those of you who are not familiar with those, these are coexisting populations of synchronous and non-synchronous parts of the large group. It has been explored in lots of different mathematical and computational forms. It's called chimera based on the mythical Greek animal that encompasses two different animals actually. And it has been, there's been several thousand papers on this kind of phenomenon. There's been some lab experiments that are ad hoc in the sense that they were designed to be able to produce these coexisting states, such as mechanical networks of oscillators, for example. But they've been rarely observed in nature until now. And we worked with another species of fireflies. These are Photorus frontalis, and they have a slightly different flash pattern. I'm playing here a movie that shows the flash pattern. So it's a continuous synchronization. They don't have these big breaks between bursts. But if we look at the plots of the number of flashes as a function of time, we can see that there are two populations. There is this population of synchronous agent, or fireflies, and in the background there are these group of fireflies that are actually not part of the same rhythm. So these are chimera states, and we can even get some interesting analysis of how prevalent they are as a function of the time of night and as well as the flash density. Okay. So these are a couple of interesting observations we make with firefly swarms. And then I want to end by pointing out that we think our recording method can also be useful for firefly conservation. This is a relatively recent up-and-coming monitoring techniques that actually show that fireflies populations are struggling. Here are just a few examples from the Surface Society report that came out last year, showing that there are several species of threatened fireflies in the US. And actually, most of them are data deficit. So there's not a lot of data about these species of fireflies. And we are hoping that using the stereo vision setup that I mentioned before, we could employ or crowdsource monitoring of fireflies that would potentially help these conservation efforts. So there's a couple of steps that I'll just very briefly mention, there's the tracking of individual fireflies that we saw before. Our hope is to create a big database that tells us what is the flash pattern of individual fireflies based on their species, and then eventually take these recordings from the field and train machine learning classifiers that would tell us which species it was that was recorded. So just one comment about the tracking. We extended our method a little bit to make it calibration free. For those of you who are familiar with stereo vision, stereo recording, the most annoying part of that process is to calibrate the camera. So you need to do some very careful spatial and temporal calibration, and then pose those as a post-processing step. But since we want to crowdsource data acquisition, we found a way to eliminate those. And we are only able to do this because this behavior of fireflies is so easy to analyze. So we have very good contrast between the flashes and the background. And what we're doing for the temporal alignment or temporal synchronization is we're basically plotting the number of flashes versus time that we get from one camera view, and then do the same for the second camera view. And we basically align them until we get the best fit. So it's kind of an optimization problem. So we're using the firefly flashes to align the two cameras temporally. And then we do the same thing also spatially. So we're doing this brute force calculation where we move one of the cameras in all possible directions until we find the best alignment. The next step would be to create this big database of lots of different species of fireflies. Here I'm just showing you 3, and we're currently compiling data about the number of flashes and flash intervals and the duration of these interflash intervals. And then the next step would be to create a classifier. So this is very much work in progress, but I'll just briefly mention that so we started thinking about this process as using flash patterns from the literature. So this is like the characteristic flash pattern that an observer would see when observing certain species. So here is an example of these patterns for a group of species. And then the output of these classifiers would be these confusion matrices that I'm sure most of you are familiar with, but just in case, the y-axis here represents the true species, and then the x-axis is the predicted species. Wherever we have elements on the diagonal, that means we classify them properly. So it works really well with these theoretical patterns from the literature. However, they don't account for behavioral variability, which we found to be a very important thing. And when we do account for behavioral variability, and in this example with just a very small number of species, it seems to work. But of course, we need to augment our data set with more species. So just a quick shout out to say that we have an amazing group of volunteers who are collecting data for us. And the process is pretty simple. As I mentioned, there's no calibration required. So it's really literally just taking 2 GoPro cameras and putting them in the vicinity of fireflies and pressing record. If anybody's interested and you have fireflies in your backyard, please let me know. I'll send you a kit with 2 cameras and a hard drive and let you know how to do this. And yeah, I think with that, I'll just thank everybody in my lab, especially Rafael Sarfati, who recorded most of the data that I showed in this talk. All our collaborators, and especially the folks from the Park Service, who have been instrumental in helping us find the fireflies at the right time, in the right space, and funding agencies. And thank you all so much for your attention, and happy to take questions.
All right, do we have any questions in person or on Zoom? So I can start with a question. So what you talked about there being kind of a wave of propagation of the signal for one of the species that you looked at, and I was curious how the fireflies could possibly be doing that. Do they have like a different visual signal based on the distance that they are? Because they just kind of look like spots. So if you have a hypothesis of how it might be happening.
Yeah, so one way to explain it is that there's just some behavioral delay. So a firefly perceives a flash, but there's a small delay that gets it to actually respond, and that would explain that wave propagation. But I actually didn't mention this here, and another way to explain it is that maybe there's something interesting or different in the way that these early flashers in the burst move or produce flashes. And we actually noticed that these early flashers move faster, so they produce streaks, these lines of flashes, opposed to the later flashes that tend to be more localized. Maybe they're sitting on a leaf, or they're not flying as fast. But that's exactly the kind of questions that we're hoping to answer with more data and combining these also with agent-based models.
Are there questions on Zoom, or I don't see them? Oh, okay. Yes. Okay. So I think I see it. Okay. So question from YouTube. So wonderful talk. Can you also extract interaction network from video capture of firefly? I suppose you'll be a temporal network since fireflies move as well.
Yeah, very good question that we're really hoping to get this kind of data. So what is the social network? What is the connectivity between different fireflies in this large swarm? At the moment, we're kind of limited because we lose track of the fireflies if they don't flash for a while. But we're trying to solve that problem with using infrared lights, where we can still see the fireflies while they're not flashing. That would hopefully allow us to track individuals over long durations of time and potentially find correlations between flashes of different individuals and try to infer the network structure.
All right. If there are no other questions, we can move on to our last invited speaker. Kirsten, are you there, and can you share your screen?
Yes, I'm here. Can you hear me?
Yes.
Very cool. I'm sorry I missed half your talk, or it looked very, very interesting. You guys seeing this?
Oh, yes, thank you.
All right, excellent. All right, yeah, thank you so much for inviting me.
Go ahead. Just a sec, we're going to pin you as the speaker, and then just to introduce you. So our last invited speaker is Kirsten Peterson. She's a professor at Cornell, and she'll talk to us about, again, collective behavior of colonies of animals.
Thank you for the introduction. Yes, I'm very sorry I couldn't be there in person. Kind of fortunately for me, I see that many people are online, so maybe that's okay. I'm actually too pregnant for any airlines to fly. I just can't tell with this video format. I was also sick the last week, so I'm not quite as well prepared as I would like to be. But hopefully this talk will still get some of the messages across and the ideas across to contribute to your discussion later. And please do feel free to interject at any point if you have any questions at all. I'll monitor the chat as well. But so what I do at Cornell is run the Collective Embodied Intelligence Lab, and our purpose is really to design and build and operate robot collectives that are inspired by swarms in nature. So we look at a variety of mechanisms and materials, from rigid robots to soft robots, and biological systems, because they all also represent different trade-offs when it comes to this type of embodied intelligence in swarms. So given the topic of today's workshop, my plan was to try to give you a flavor of some of the intricacies that sort of comes from studying social organisms, where the behavior really emerges from both explicit and sort of these implicit environmentally based and physics-based interactions that aren't necessarily observable. And then reversely, how does one go about trying to leverage all of those design principles to build these taskable robot collectives? And so, unfortunately, computer vision isn't really central to our research, but it does play a role in pretty much every single study and project that we do. So, this will advance me. Here we go. All right. Just because this term is a little new, right? Collective embodied intelligence. Let me just try to quickly list the different ways in which you can think about injecting intelligence into a system if you're building a robot collective, or if you will, ways in which sort of intelligence comes about in social organisms in nature, right? And please be lenient with my choice of words here. There are many different ways to classify these types of design techniques, so this is just going to be one example. But of course, we know that every individual in a swarm, right, can have some amount of explicit intelligence, right? We're comfortable as roboticists with this type of development. We can do learning, right? I think you've seen a bunch of talks on that this morning. Now, if you have many robots working together, you can also do things like careful pre-planning, or maybe if you have like a dynamic task setting, you can do things like adding a centralized controller with communication to all the agents, or you can go sort of the social bio-inspired route and try to make the system more scalable and error-tolerant by having distributed control with like explicit agent-to-agent communication where all of them just communicate locally. But the point is that we can do more than that, right? That's where the embodied intelligence comes in. Turns out we can also outsource quite a bit of that explicit intelligence to mechanical intelligence or reflex-like behaviors, if you will. I think in nature, you'd often hear this concept under like form and function, so the fact that every part of an individual organism is optimized for the particular environment it exists in. And the same is true, right, if you have many robots working together, right? So it doesn't have to be explicit interactions or even high bandwidth interactions. It could be direct physical interactions or perturbations between agents that that work in the same environment. They can also be sort of environmentally mediated interactions, right? So stigmergy related, right? So one robot can modify the environment, leave a cue, and then that causes a robot that passes by later, or an agent, or an ant, or whatever, to commit a particular action. And so, I'd say as roboticists, at least in my field, very comfortable with these types of design techniques up here, right? With intelligence, with planning, getting much better at large-scale distributed communication networks. But anything based on the techniques over on the right are much more under development still, right? So sort of including them into the planning process becomes kind of interesting. And so that's what we do in my lab. Really look at sort of the interplay between all of these design techniques to try to generate these task-oriented behaviors in robot collectives. And so, part of that means that we're trying to develop very simple robots that can realistically be used in large collectives. So cheap, simple fabrication, low maintenance. And then the other part of the work is trying to find ways that we can generate both scalable and robust coordination among those robots. And as I said, often that basically happens by looking to nature for inspiration, working with biologists, coming up with new ways in which we can study them, and then transferring those ideas back to robotics. And so my hope for today is really to just try to focus sort of mostly on what we've done to explore the concepts on the right half of the plane, so over here, morphology, physically, environment. And hopefully that will complement some of the topics you heard about this morning, which might have been a little more on sort of learning and software oriented control. Okay. So we have 3 big model systems in our lab. We have termites, bees, and slime mold. And I'll focus mostly on the first 2, and we'll see if there's time for the third one. So the termites are really the oldest model system. We started way back in Harvard when I was doing my PhD under Radhika Nagpal. And these are, remarkable builders, right? So each termite is about 1 centimeter long, but it can collaborate with millions of nestmates to build these phenomenal meter scale mounds in Africa that sort of help to provide ventilation to the nest, which is actually located underneath the mound down here. And so, compared to the complex behavior of the colony as a whole, the individuals here are relatively simple, right? They'll crawl around, they'll interact socially, they'll sense social cues and cues like pheromone and moisture levels and geometry specific to construction. They rely heavily on these stigmergic rules, these environmentally mediated rules. They'll coordinate via physical effects like crowding, and they'll even use their own body size as a template for smaller columns and conduits. So you'll see an example here where we put a bunch of termites with mud in a petri dish, and they've started building these columns. Columns are about the height of what a termite can reach, and the columns are about 2 termite lengths apart. And so we studied these systems, right, and they're just like remarkably robust and scalable. So here's an example of a perfectly healthy mound with a tree growing inside of it, right? Perfectly healthy tree, perfectly healthy mound. It's that level of sort of incredible adaptation and robustness that we're trying to strive for in a robotic system. And so I'll give you some brief examples of what we looked at when we looked at the actual termites, and I'll give you some brief examples of like how we translated those ideas to robots. So the first one was talking about these mound-building termites in southern Africa and Namibia. And here we really work with biologists to come up with both sort of in vivo and in vitro studies of their behaviors. These are the Macrotermes mycoceni that build these very large pointy mounds. And then very closely related Natalensis, which have the same morphology, live in the same area, access to the same soil, but build much flatter mounds. So they're kind of an interesting complementary set of termites. And so this first example here is like sort of an ant farm-like structure that we stuck into the side of a mound, so that we could try to visually record the response of the colony over time. Just like how quickly does the alarm pheromone go about, like how quickly do they come up and actually try to plug this hole. Here's another example. So we actually built this portable 3D scanner that we could bring to Namibia, set up, take the termites out of the mound, stick them into petri dishes with soil while they were still engaged in construction, and then sort of runningly record as they were carrying soil around the petri dish. So you see an example of a termite. Here's some dollops of mud. And this was back in the early 2000s. I'm sure you could do much more if you had stereo vision cameras these days, a lot cheaper. Not surprisingly, as we deployed these things and we started looking at termites, we found what everyone else found, which is that they use these sort of geometric cues, right? So a termite that places a dollop of mud or starts an excavation will eventually, like those sites will eventually attract more attention from other termites. But you can also note, so here's a video of termites running around in a petri dish. You'll note these crowding effects, right? Termites are social, they like to work in active construction regions. So that's interesting, termites tend to gather. We also studied just using these simple mechanisms. And you see, like we did sort of tracks and we did behavioral labels as well. We started studying pheromone. So in the past, many people have indicated that you can use sort of pheromone depositions in mud dollops as part of the cue for stigmergy. And we actually found that the pheromone has more of an arrestant effect. And by that I mean it doesn't prompt particular behavior. It just seems to be a way in which termites can identify nest material versus non-nest material, right? And so the majority of the termites are then going to stick to construction on the part that smells like home, which is not surprising. So the lower part down here smells like home, and this is freshness construction up here. Fresh material up here. A more interesting finding, perhaps, is this key individual finding. And so we found that only because we started using computer vision to track and label the behavior of every individual over time in petri dishes. And what we found was that a few termites in every single population we put in is actually attracted to build right at the pheromone gradient. So everyone else, they just stick to home, right? They're safe. But a few of them, they go straight to the gradient and place depositions. And eventually those geometric depositions and cause others to follow suit. And so they're sort of either sort of instigating new construction at the border, trying to build a wall, or perhaps they're actually trying to like expand their nest territory into the perimeter. Whether that comes about because there's some inherent difference from their neighbors, or because they just slowly adapted their internal threshold for where to drop depositions, we don't know yet because our study ended a little early, but it certainly be of interest to study next. I actually wonder if maybe my collaborators ended up looking at this as well. But it was sort of the first time we went from looking at them as these perfectly similar automata to actually looking at sort of the individuals and really seeing sort of which instigate what behavior. And I think it sounds like, a lot of studies are coming to these results as we look at closer and closer individuals, right? So I sort of heard that Orn was just talking about a few of these fireflies, right, that were propagating delays and so on. And so it seems like there's some variation in the populations. But these were sort of the key effects that cause termites to be able to construct together. And it's remarkable that they're capable of creating such phenomenal structures despite of these simple coordination mechanisms. So lately, I've been working with my postdocs, Paul Perdunias and Alexandra Niles, and our collaborator, Dan Goldman at Georgia Tech. looking at local termites, so Coptotermos formosanus. They're very prevalent down in the south, probably where you are right now. These are super termites, so they can digest wood in their guts. They'll eat wood directly, they're subterranean. And what I'm showing in this video here is we've basically taken a colony of them, we've sandwiched them between two glass plates, and then we've given them blue sand to dig in just so we have an easier time optically recording their behavior. And then we can use these custom computer vision scripts that Ali developed to automatically analyze their presence and how their tunnels form over time. And you'll see that their inherent behavior is to just dig. That's what they do, right? They expand their territory, they dig. And in the past, researchers have shown that their tunnels and branching patterns do indeed sort of correlate with these somewhat optimal space covering foraging patterns given both their morphology and the type of wood that they like to digest. So what's interesting again is that, before we're looking at construction, now we're looking at digging, but again, their behavior seems highly dominated just by geometric cues and crowding. In fact, so much so that we're currently doing studies to show that we can largely replicate their behavior with completely innate vibrating discs or robots, if you will. The key really just comes down to how much resistance or memory, if you will, again, is built up in the sand. So again, it's the stigmergic effect, where the environment acts together with the species, with the organism, to cause this behavior to happen. So a pretty complex behavior that results from these very simple local interactions. Okay, so those are my brief pitch on what we've done to actually study how termites use mostly sort of coordination mechanisms on the right half of the circle to generate their behavior. I want to also give you a brief pitch on how we abstracted some of those ideas to work in our robot collectives. And so here's a very brief pitch of that system. Here I asked a number of robots to build a particular structure in 3 dimensions out of a custom material. And so this is work I did back in my PhD at Harvard on the Radiocanach pole. And so inspired by the termites, right, we sort of exploited a number of interesting coordination mechanisms in the system, and it's kind of new because normally you pick one and go with it. So in this case, there's some explicit planning, right? We know the final structure we want to create. Every robot knows it as well. We add a particular docking station in our environment. We sort of compile how robots should be moving through the structures, the general direction that they should be taking, so they don't meet head on. There's some local intelligence that lets the robots bring or add breaks only where they won't become a hindrance to future robots, so it wouldn't try to add a break here because that would create a cliff that other robots couldn't climb over, for instance. The morphology of the robot is also heavily co-designed with their material and the task, so the robot itself is actually rather simple, even though it's performing a rather complex task. But other than that, right, the entire construction progress is guided entirely by these local stigmergic cues, right? So while they do know the target structure, because we want a particular blueprint at the end, their decisions are guided only by what they see in their local environment. There's no exact order to the way the breaks are deposited. There's no way, the robots don't communicate with each other at all. They don't communicate with a central controller. They can only sense their little local footprint. They don't have a memory of what they built before. You can imagine that memory isn't very useful in these kinds of systems where lots of robots are working on the same structure at once. Right, so what they're doing is coordinating entirely through the environment, or through stigmergy, if you will. And so in the past, we looked at a bunch of aspects of these types of coordination strategies. And we really started playing around with where do you best inject intelligence into the system, right? And also sort of looking at, if you are using environmentally mediated interactions, how do errors propagate, right? So if I place a brick wrong, one thing is I placed the brick wrong, the robot walks away, it's happy, but now there's an error that propagates to all other robots that sees that error in the future. Anyway, so that's sort of an exciting system where we took a very abstract view of the termites and said, okay, we can clearly do this without central intelligence. Let's try to do that. But of course, you notice that the biggest difference between this system and the actual termites is that here we're striving for a particular structure, whereas the termites are really just striving for a particular geometry. And so there's no real way, for a particular functionality, right? And so there's, for the termites, there's really no way to fail. They just keep optimizing towards that functionality. Whereas for the robots, there's a very distinct way to fail. And so what we're currently doing is working on a new model platform that lets us work on sort of towards much more organic looking construction. So this is what that robot looks like. It's a very, very minimalistic robot platform. It's called a double-tailed single actuator wave mechanism. So here's a video of that working. Basically, every tail here has a single degree of freedom. It's a rectangular chain that surrounds a helix that's rotating around its own axis to produce a sinusoidal motion that causes propulsion. And these robots have been shown to sort of locomote over a variety of terrains, but the interesting thing is that just by changing the relative frequency of the two tails, we can also interact with the granular material, right? So we can show them leveling terrain, we can show them digging indents, we can transport materials, we can also build mounds and things like that. And of course, you know, being the lab that we are, we're interested in collective behaviors, so we optimize that mechanism so that two robots can climb over each other and then help each other do things that they can't do on their own, right? So climb over each other, they can help push each other under the soil, they can help dig bigger mounds that they would be able to on their own, or climb steeper inclines than they would be able to on their own. And so these videos, granted, are of tethered robots. We actually have robots working in our lab now that are also untethered and to some extent autonomous, so we're working on planners as well. Again, guided sort of by the termite idea. And the key question really is, how can we, just like those termites, use this idea of just crowding and collaborations and stigmergy to arrive at a really, really robust system that still eventually sort of gets at these functional structures that we want. Okay, so that was not about robots. There's really no vision in those. But these were sort of examples where we're studying, you know, how termites coordinate largely through their environment, and then trying to adopt some of those ideas into our robot systems. So let me spend a little more time instead on some of our work that's based on honeybees instead. So this is a model system we use pretty extensively in our group, and it has a bigger role to play with sort of the computer vision that you're discussing in this workshop. So again, if we think of these as sort of a collective system, right? Individual bees are pretty agile. They can fly, they can manipulate items, crawl. As for an indicator of intelligence, I have about 500,000 neurons, but in comparison, we have about 86 billion neurons, right? So you have to think of them, to be fair, right? Bees don't live alone. They live in colonies with thousands of individuals. And so the neat thing is, like, how does an individual bee that has very limited state information still cooperate efficiently with the entire colony, right? That's the big question. And so what I'm going to do is give you a couple of examples where we sort of show you how bees sort of mix and match from these coordination mechanisms. And again, I'm going to try to focus mostly on the right half plane, because I think that's the one that probably poses the more questions for visual observations. Okay, so let me start here. So this is honeycomb construction. And so this is a recent study that came out in PNAS, which started way back in 2017 with a guy who at the time was a grad student here and is now Michael Smith, Professor Michael Smith at Auburn University, and Professor in Knapp here at Cornell University. And so we're really looking at how can bees build honeycomb. And so the interesting thing here is that basically, you know, we all know that bees build honeycomb. They build these really nice hexagonal cells. But the cells are used for a lot of things, right? They're used for storage, they're used for brood rearing. That's of both worker cells, which are fairly small, and drone cells, which are fairly big. They're sort of structural to some extent, especially in nature. And the bees are trying to sort of create or to use minimal material because it's costly for them to produce this wax. Right? And so if you put this in engineering terms, what they're doing is basically trying to create this kind of multi-objective optimization. They're doing it inside of the hive where it's completely dark, and they have access only to sort of local tactile interactions. And so, you know, pretty phenomenal process, again, by these individuals that have fairly limited information. And again, no central point of intelligence. And so what biologists believe is happening, right, is sort of a mix of the fact that they use their own body templates. For example, they'll, you know, so again, the morphology. They'll hang in these bee curtains to guide sort of the orientation of cells. They'll use the size of their own body, sorry, the crowding effect. They'll use the size of their own body to guide the size of the cells. And they believe that there's some, you know, stigmergy, again, sort of environmentally mediated coordination that helps guide how the comb actually grows itself. And so to study that phenomena, we did some pretty extensive field experiments back in 2017. We got a ton of data. But basically what happened was we had 11 colonies work in the field to fill out empty frames of honeycomb. And then we did sort of computer vision analysis and extracted a whole bunch of data on both workers and drone cells. And so a comb pane, for example, looks something like this. And what we're interested in looking at is both transitions and merges. So one thing is that a honeybee can build a perfect hexagonal cells. It's not surprising. What is surprising is what happens when you can't build a perfect hexagonal cells, right? So bees will actually build in multiple different areas at once. So you'll see there's one initiation point here, probably one here and one here. And eventually these have to grow together, right? And what happens right at the intersection point when they're not perfectly aligned? Also, what happens when you have to merge or transition between large cells that are for drones or smaller cells for workers? What happens if cells aren't perfectly oriented, right? All fun things like that. And so those were really what caught our attention. And so transitions, let's see, there's some transitions over here that should be somewhat obvious. So there's some large cells here and some smaller cells here, where the bees sort of started building small and then eventually went to larger cells. So this would be a transition area. And in this one down here, we sort of highlighted cell orientations, which highlight a little better what a merge region looks like. So you'll see that this pane was built at once and then eventually tied together with this pane over here, and eventually tied together with this pane over here. In more or less good ways, right? And so we got a ton of data on all of this. Okay. So what we did then was create automated computer vision-based analysis to extract data like that. And so here's just an example. There's actually a newer interface compared to what we had before. But basically you can load in pictures of these frames, and then in 2D we can sort of analyze, just using either template-based analysis or more sort of traditional computer vision-based methods to try to extract all the vertices and all the sales centers, and then figure out like how long are all the wall lengths, how long is the cell areas, what's the average number of walls, all the fun things that you go about marking in honeycomb. So you'll see it works pretty fast. It's sort of a neat piece of software now. It's based on Python, but has this nice front end that runs in a browser, Java. And so it's pretty user-oriented by now, and we run it across both engineering groups and biology groups, for instance. Okay. As examples, and you'll see that there are both methods to do things in automated ways, but there are also methods afterwards then to go in and sort of, clean up the data so that we can get very accurate data afterwards. Okay. So what do we observe from all of that data? Right, that's really what it comes down to. In terms of cell area, we found that, if we just look at a bunch of samples of worker comb or drone comb, right, they fall in these two very distinct regions, the red and blue regions here. And if we look at transitions, right, where bees start by building worker and slowly advance to building drone, or the other way around, they fall within those two categories as well, never outside. And the same is true for the merging of worker with drone cells. Now, interestingly, if bees are trying to merge two panes of drone cells, then sometimes they'll build cells that are a little bit bigger or a little bit smaller. Smaller is not a problem because you can always rear a worker bee in there. But bigger means that you're kind of wasting a little bit of space. When you're merging worker with worker cells, right, you very rarely get cells that are too small. And that kind of makes sense from a functional point of view, right, because if they're too small, the queen won't even lay an egg. If we start looking at how these sort of seams, oops, let's see here, are gathered together, right, so here's an example of another honeycomb which has a bunch of these merge lines happening here, here, and here. And you'll see that we get these particular motifs of 7 and 4-sided, or sorry, 5 and 7-sided cells together along those merge lines, so we get much higher incidence of those types of motifs. And we also sometimes get motifs of 4 and 7-sided cells together. Sorry, so that's what's shown down here. And finally, what we looked at was the cell orientation. And so we found that, if you're just trying to transition cells, you're not going to really bother changing the orientation of the cell, right? It doesn't make sense. But if you're trying to merge two panes together, it could be that the cell orientation is quite different. And you can see that the number of irregular cells that appear if you're trying to merge ones that have very different orientation get like that proportion goes up considerably, independent of whether you're doing merging worker with drone cells or drone with drone cells or worker with worker cells. Okay. So to really sort of make sense of what we saw with that data, we then developed two models, both sort of a base layer model, a lower bound, and then upper bound and optimal model. So this here is a naive model, and what we did was basically first we try to fit coordinate systems to the actual data, and then we very simply propagate cells forward rigidly in that coordinate system from both sides, and then we pick the one that sort of leaves the least amount of free space. So the blue ones here would be propagated from the left, and the orange ones would be propagated from the right. Whichever one leaves the least amount of space is the one we pick. And then to sort of emulate the fact that these may not care about perfect cells, we also try to vary the sort of amount of overlap that we allow when these cells go together. And so it's a very naive way of dealing with the problem, right? But it's a way to establish this lower bound. What's neat about it is...
Just to break in, you're almost out of time, just to let you know.
Yes, thank you. Okay. I'm always at the end. So what's kind of neat about this is that we can also use this to sort of do parameter sweeps to figure out which types of merges would be harder for bees to fit compared to others. And so this will sort of inform interesting studies in the future. We have this optimal model as well. So very briefly, we look at the amount of spare area, we fit in the number of extra cell centers that we would need, and then we do sort of a spring-based optimization model where we figure out what's the best way to fit in new models. Now what was neat about it was that the proportion of hexagonal cells in both our model and with the bees look very, very similar, and so we're encouraged by the fact that they look similar. Although because in our global model we allow sort of the configuration to be optimized to look across the entire merge line at once, it's able to fit the sort of area distribution much better than the actual bees do. Right, so not surprising, but otherwise they match quite well. And the cool thing was that then when we sort of looked between the bees and the naive model and the global model, the optimal model, we found that bees do almost as good as the global model and much, much better than the naive model, right? So that's encouraging to see. And of course, the interesting thing for us now is to say, okay, we know that they can do it, right? But the question now is, how do they actually, what is the rule set that gets them to go there? Right, and eventually we're hoping to extract those ideas back to our robot systems where we say, like, if I have a large robot system and we're building a big grid-like structure, but we're building in two areas, how do we do what are the coordination mechanisms that will allow us to best merge up those things later on? Okay, so another example, I'm sorry, I ran a little over time. I can end here if you want, or I can finish up in a couple of minutes, it's up to you.
If you want to finish in a couple of minutes, there won't be time for questions, but it's up to you whether you prefer
questions. I'm happy to just take questions instead, that's not a problem.
Okay. Are there questions from the audience? Anyone on YouTube or? Okay. Oh yes, right. Sorry, you're supposed to clap. So I guess I have a question kind of related to putting your talk in context to some of the other talks. I was curious about whether you use some of the simulation environments like Mujoko or things like that for trying to as an in-between kind of the biology and the robots.
I'm sorry, I didn't quite catch that. Can you say
Oh, sorry. If you use the physics-based simulation engines at all as kind of something in between your biology-based theories and robots.
Yes, we do. And unfortunately, I didn't get to it. But we actually have some really recent work on. I'll pull up one slide here. I think, or I might also find it interesting. So maybe that's why. Let's see here. So we have some recent work on swarmulators, which is also work out of Steven Stolgat's lab. And in that work in particular, it's really lagging. Sorry, here we go. we're really interested in, we've shown both parallels to, robotic systems, what we can do and how we can sort of, very, very simple parameters, change the entire coordinated behavior of the collective. But at the same time also model a bunch of interesting phenomena in nature, like metachronal waves and vinegar yields and things like that, and in physics and active matter physics. So really, this should be coming out in a few months, hopefully. Good question.
Any other questions? All right, I think maybe we'll end there. Did you want to give a preview of the afternoon, or we start at what time again? Okay, sorry.
Yeah, thank you everyone for the amazing morning talks. It's been a really interesting mix of topics. I think, oh wait, I have to tab this over. Go away. So for our afternoon talks, let me share my screen. Yeah, just as a preview for the afternoon, we're going to have three submitted talks by speakers who are also going to be presenting their work at our poster session. Then we're going to have a break for a panel discussion between the submitted and invited speakers. And then following that, Jensen is going to be presenting our multi-agent behavior challenge, and we'll see talks by 3 of the winning entries in this year's competition. And after that, we'll have a little more discussion and a poster session out in the lobby. So stay tuned for that. We'll be back at 1:30 CST with the start of the submitted talks. And thank you all for joining us. We'll see you then.
the Zoom is the one, the Zoom is the one.
my postdoc just shared a preprint, so.
No worries. then I'm like, oh, if it doesn't work out, maybe I should work yourself. Yeah.
Oh, no worries. Yeah, yeah, what's up? Really? Okay. Okay. So all the posters are in the same area. I think it's in halls D and E. Is that right?
It's next to a food. Next to the food. Oh, it's like really close to the food. Really close. Yeah, probably back if you can find it. Okay, sounds good. Alright. Yeah, fine. That was just asking the oh, yeah, our poster session should be like pretty full. That's good. Because I want
I
Oh, are you on the Slack?
The Slack?
Yeah.
No.
Okay. I'll email it to you then. Maybe you should enter the email just to make sure. Or, what is it?
It's XAVIER. Oh, it's not here. Anyways,
No.
B of Paris.
T.
UIG.
Oh, just your name.
IG.
I.
Of giraffe, like the last
Oh.
word. Yeah. F@gmail.com.
F?
Yeah. That's what it is.
Okay. Try that. Yeah.
Correct.
Okay.
Awesome, thanks.
Cool.
Nice to meet you.
He's working with me this summer on like extension. Oh, cool. I researched it.
Yes.
Yeah, I was surprised you guys never met when you had to from Northwestern. I was like, oh, is that one?
You're with the mental school, right? Yeah, they're like, there's like they're different families.
Oh, you're in the north?
I'm in, yeah. It's expensive to come.
I remember seeing you in like a big building. That's a bit. Yeah.
There's like a PhD student. He says that he might know Florian Schippers. Florian Schippers. He recognized you. No, he's in Colorado. Oh, okay. Do you work a lot? Do you work with radiology at all? Do you know Todd Parrish? Okay, so I guess you're kind of disconnected. Oh, it's nice to meet you.
I guess virtually.
Yes, in person. Yes. I'll probably have to do like Airbnb or something. Yeah.
That's like housing doesn't cover surf housing after August 20th, which is really bad. I emailed them. I was like, even well, not like talking to students, but they're just like, oh, we can't like.
I think it kind of makes sense. Yeah. Like, you learn like international people like things to make you.
I feel like there's room for like one student. So maybe if they do it for you, they can do it for like 100 other students.
Yeah. September 20th. Yeah, we have like undergrad, we have like the quarter system, so we didn't do
Yeah, like the grad students are the quarter system too. So you're doing the 3D discovery.
Yeah.
Super slow progress. We sent two full-time people.
Yeah, so makes sense. I was actually like chatting with someone and like he gave some like good suggestions. So talk about that.
Yeah, we can meet up somewhere after one day.
Yeah, I saw that you have 2 posters tomorrow, right?
Yeah.
It's like, yeah, I think basically it's
I'll stop by the poster tomorrow.
Yeah, that sounds good.
I'm actually going to
It's like they're specialized.
Oh.
Because like there's 2,000 years of experience. I met Microsoft and like we barely recognize each other. It's like this screen is seeing that the screen is so different.
Wait, so you guys haven't met? Oh, you're doing the virtual.
It's virtual. Yeah. It's really a much too He's hosting the robustness.
Oh, no, yeah, so I'm just gonna
Try to get people to come for like presentation.
What time is your presentation?
It's at 3. Yours is at 2:45.
Okay, I mean, you don't have to push my
Oh, it's on this.
Oh, it's because I'm not like submitted or invited. I just can't.
Oh. I'll be back at 3. That's
Okay, thank you. Just open your copy.
Yeah, thanks. I invited some people, so I'm actually kind of nervous now. Because people will actually be there.
I think it's good to invite people
The one of the guys, I've been chatting with like one of the people that I cite in work, so we've been chatting and he's gonna come see it, so I'm kind of scared.
Yeah, until Friday. So I thought I could do a whole day. And I like the last time I slept was like 9. I feel like experience. So for all the workshops I think I'll be able to be talking about, I sat there and I was like, oh, that sounds around right. I'll just go to the studio.
Okay, so this is from CVPR.
Oh, it's a little slower, it's a little more relaxed. Yes, people actually water. If you go to the Bay Area, you'll see lots of bluegrass. Because if you water your lawn, you'll get $1,000 for wasting water. Yeah, and lots of people like go really hard for their environment.
That works. Get it set up now.
Oh, right. Usually.
Sorry? Yes.
Oh, I guess I'll find out if
Thank you for reading.
I have to for the
Oh, yeah, that's true. I was like, yeah, you're
oh, that's good. So I guess when I did my cert, this was like
Hmm?
This was the summer of like the first summer of COVID, and the reason I did this was and it was like kind of an emergency last minute thing. And I was like, hey, so I've got nothing to do during the summer now.
But we never did it there, right? That was with
No, yeah, I did it with audit. Yes, and then that thing January I kind of turned into this project. So, like, yeah, so I kind of counted it as like the same project because it was
Okay. It's, so what we'll do is once you start, I guess we go to slideshow.
Okay.
And it'll launch. If it doesn't launch there, then we need to tab it over to this window. Yeah, seems okay. Do you need to check any videos or anything?
No. Okay. Awesome. Thank you so much. Do you need details on how to?
Yeah. You can either, if you want to show slides from your own laptop, you can join the Zoom call and share them by sharing your screen. Or you can send me a link via Slack and I'll open it up here and you can share them from this.
Okay, I'll send you a link.
Okay.
Thank you.
By the time it was like 11 a.m., I was like, this makes time like feel like Siberia. It's like, it really feels like 185 or something. It really does not feel like 95 degrees. No, but you go to Taiwan and it says 95 and humid, it feels nicer than this. This is like just roasting in the sun or something. At least when I went to Singapore, that was like also like 100 degrees. I think the reason why is because there's a lot of asphalt.
paid the printer of the first paper, so can I check my PowerPoint?
Give me 1 second to load.
I think I sent you
Yep.
a Google link.
We've got it pulled up here.
Yeah.
So you'll. Let me post this first. Sorry.
So can we check the
the video?
Play the video? Yeah.
Sure. You wanna?
And I think just the slideshows and
Slideshow.
yeah, and yeah.
Oh, so we'll need to tab it over to the screen.
I think it's okay.
Mm-hmm.
Yeah.
Yeah, looks good.
We could discuss it. Also, like project.
Yeah, I think it's okay. Thank you.
Okay. Yeah.
Thank you.
So it's pronounced Peja?
Oh, sorry?
I guess Jen's going to be introducing you if you want to.
Oh, yeah, thank you. I think the meeting will start in 8 minutes.
Mm-hmm.
Thank you.
All right.
Did you get a slide? Sorry, I was. Oh,
So I have dance track.
Can I just make sure that so you're gonna come up every time to do this?
Oh, just at the start. Oh, I'll come up and make sure that the slides get
Oh,
finished,
okay. And then
You
I'll
can-
be doing the bio at the same time?
Yeah.
Okay. Okay, I just wanted to make sure.
And that was Peja up there. You just
Pedro.
tested this.
Okay, cool. Yeah, I saw him come and I was like, I should probably Pedro. Okay, good. I'm not going to use pictures for everybody.
Sorry?
I like to use pictures for everybody.
Yeah.
Oh, we'll have the timer.
Oh, oops. Timer, where'd you go? There.
Thanks a lot for all the setup. It was pretty complicated.
It seems to have worked out.
Wait, so just to confirm, so in 8 minutes, you'll call everyone back and then introduce afternoon sessions. And then I'll come up and give Pedro's introduction. And then I'll come up and then ask if there's any questions. If there's no questions, I'll like
go on to the next.
Oh, I'll have one question.
Okay.
If there's still no questions, I'll come to the next. Okay.
And it's okay if things run a little bit long, because we had the discussion starting at quarter after.
Yeah, I think that'll be fine. And then I assume you're gonna set up the panel.
Yeah.
Probably don't need my help as much.
I think so. I think I'll just have people bring chairs up and then.
Okay. And then maybe we can chat a bit about the challenge during the coffee break between. I think the logistics will be similar, but we should check in.
Yeah.
Probably before we just jump into it. Okay.
Yep. Discussion questions.
I think the morning session was actually like pretty well attended.
Yeah.
Like in the beginning, it was pretty good.
And we had, I think we peaked at like 16 on YouTube concurrently and comparable in Zoom.
Yeah, I think it was around 40 people across YouTube and Zoom, at least earlier on.
Yeah. Yeah, the streaming thing is cute. Where to go? There. So it's concurrent viewers.
I hope you'll come back.
I just tweeted it, so probably.
Hey, did you guys get food?
Yeah.
Okay, is there anything you need help with?
I think we're all set.
I can, I'll try to monitor things on my phone better. I kept trying to watch the...
It's tough.
Yeah.
Oh, so should I, so if I'm asking a question, I might just focus on the talks and, okay, sounds good.
Yeah.
Well,
I'll keep an eye on things. The one thing I sometimes miss is the YouTube.
Oh, okay.
I think I can just get it, make sure that I'm always seeing the most recent.
Yeah, the YouTube is like, if you just check once every 5 minutes, like, it should be pretty chill.
It also shows up here.
Oh.
And then the Slack shows up.
Oh, most people weren't asking questions on Slack.
Yeah.
So I think we'll be okay.
Got some folks joining.
Yeah, I was actually like, one extra thing we can do later is we might do we want a new logo for Slack?
Oh, yeah, at some point.
Okay. I mean, it's really cute, but if it's going to be used for the workshop, which we emphasize multiple domains, we might want to not show favoritism.
totally fair. Let's pull that over here.
Thanks a lot.
Okay. So YouTube, or
So
Zoom, Slack, and YouTube.
I guess I don't have to like touch the computer until it's my turn to present later in the afternoon. It sounds like okay. Because I don't want to miss anything else.
Right. And then I have questions. Oh, I think the discussion would be fun. Did you hear from Yusung?
I messaged him this morning and he said okay.
Okay.
I trust he'll join. If not, I'll just Slack him.
Mm-hmm.
He usually responds instantly on Slack, but I don't know how.
And that's the current, oops.
Okay, that looks really good. Thank you.
Yeah, I think we're set.
Okay. I just have to get ready for my 15-minute at 3.
Yeah.
Which I think will be fine. I guess I can sit down. There's only 2 minutes and a half left.
Yep. I'll hang up here just so there's a face on the
Oh yeah, when people come in. Yeah.
thing. That reminds me, we should bring participants.
Was the YouTube stream just filming this one time, by the way? It was just...
Yeah.
Okay.
How come I don't see the who's talking?
Because no one's talking.
Isn't there usually the little floaty Zoom thing?
Oh yeah, you're right.
Maybe. Oh, it's right there. I'm just blind.
Designed to be dragged.
Yeah.
Okay.
That's what I was about to do.
Okay, I will let you handle it. Thank you.
Okay, I guess we're resuming. The timer has told us we are. Yay! All right, welcome back everyone. Thanks to those who presented and came to the morning sessions. We have 4 segments of this afternoon. We're going to start with some submitted talks, which will also be present in the poster session. We're then going to have a panel discussion that'll run 30-45 minutes between the invited speakers you saw this morning and the submitted speakers, just to kind of compare people's insights from the workshop so far and what we've learned about behavior analysis and multi-agent behavior interactions in different fields. And then after that, Jen Sun is going to lead an overview of the multi-agent behavior challenge, and we'll hear 3 talks from winners of this year's competition. And then after all that, we will have a brief challenge discussion and go on to the poster session. So without further ado, I'll let Jen introduce the first group.
Thanks a lot, Anne. So the first speaker for this afternoon is Pei-Je Sun. He's a second-year PhD student in the Department of Computer Science, the University of Hong Kong, advised by Professor Ping Luo at MM Lab. His research interest is in computer vision, especially object detection, segmentation and tracking, and previously he graduated from Xi'an Jiaotong University. Passing on to Pei-Je.
Okay.
All yours.
Hello, and thank you, Jennifer's introduction. And I'm happy to present our work, Downstrike, multi-object tracking uniform appearance and diverse motion. This work is in collaboration with Jin Kun, Yi, Zehuan, Song, Chris, and Ping. So our work is a new dataset for multi-object tracking. Please first see a demo video. So yeah.
Yep. You have to
so
mouse this over there.
Oh.
And there you go.
Yeah, thank you. So as you can see, our data set has 2 infer-set properties, a uniform appearance and a diverse motion. And for the uniform appearance, humans are in highly similar appearance. For the diverse motion, humans are in complicated motion and interaction patterns, which experience frequent relative position switches. So why do we propose such a data set? Why do we need Downstrike? Our motivation comes from 2 aspects. First is we observe that the appearance feature of different objects are very distinguishable in the feature space on MOT17. MOT17 is existing popular multi-object tracking benchmark. The second motivation is oracle analysis with ground truth detection box on MOT17. We found that the performance is almost perfect in MOT17 with using the ground truth detection box. So in this case, we found that if we can use ground truth detection box, MOT17 will be degenerate to a detection problem instead of a multi-object tracking problem. So this is not what we expect. That's why we propose Downstrike. The uniform appearance and diverse motion is a great combination. For example, only uniform appearance without diverse motion will degenerate the task into a reality problem. At the same time, only diverse motion without uniform appearance, only uniform appearance without diverse motion will degenerate this problem into a pure detection problem. At the same time, only diverse motion without uniform appearance will degenerate this task into a pure reality problem. So that's why we combine uniform appearance and diverse motion.
Just gotta unmute you.
Oh, yeah. Okay, so we briefly introduce Downstrike from 3 folds. First is scene diversity. Downstrike contains multiple types of downs, such as strict downs, pop downs, classical downs, and so on. The second one is very obvious appearance similarity. And the third one is motion complexity. On the one hand, we found downstrack and other datasets have close average LU on adjacent frame. This shows that downstrack does not have unreasonable fast object movement. On the other hand, we found that downstrack have much more frequent relative position switches, which is caused by highly nonlinear motion patterns and inter-object occlusion. So we benchmark current state-of-art multi-object tracking algorithm on MOT17 and downstrack. For the tracking quality metrics, HOTA, IDF1, and ASSA, all algorithms show a significant performance gap from MOT17 to downstrack. For detection quality metrics, MOTA and DETA, all algorithms are in fact higher on downstrack than on MOT17. This suggests that detection is not the bottleneck to have good tracking performance on downstrack, and further highlights the drop of association performance. So I think these two experiment results well correspond to our two motivations. We also do several experiments try to improve the performance of downstrack, and we have three meaningful conclusions. The first conclusion is better motion models brings more benefits. For example, both Kalman filter and LSTM outperform naive LU association by a large margin. The second conclusion is that more modality of mask pose depth makes positive effects. So since downstrack has only box annotation and identification annotation only, we propose to use joint training technology with other datasets to enable the model to output more modality such as mask pose depth, and we could get better performance from that. The third conclusion is multi-object spatial and temporal interaction improve significantly. Here I want to introduce another paper called MOTR. MOTR uses attention modules to perform object interaction and temporal aggregation. So it's due to the attention mechanism, it achieves the state-of-art performance in downstrack, as you can see in the table in the left. So we are organizing multi-object tracking and segmentation in complex environments workshop in ECCV 2022, and we sincerely welcome all you to join our workshop. Thank you for your listening.
Great talk. Thanks a lot. Are there any questions from the audience on Zoom or YouTube? Please let us know. Oh, okay, go ahead. I think it's you.
I was coming up here for the microphone. I was wondering if you tried any individual recognition, like so you said they're virtually indistinguishable, but have you tried training, something that you use for like face recognition to differentiate different dancers, say, and does that work?
Yeah, I think it's, if we want to use some of the face technology, I think it's a great idea, but as we can see in the video, so I guess the resolution of the picture of the can the face be visible is also a severe problem in our data set. So face is a great idea, but I think it's not doable in this situation.
Yeah, I guess I meant the same algorithms used on the whole body of the person, but,
yeah.
But things that have the resolution, the kind of fine-grained.
Yeah, fine-grained, just like the mask pose or the depth. So I think it's a very great future direction, and we have tried several methods, and it indeed improves the performance. Maybe the 1 to 2 points. Yeah. Thank you. Yes, it's a great. I think it's a potential direction for the future, and we need to continue to move on this direction. Yeah. Yeah.
Thank you. Unless there's any other questions, the question time is up. So,
Yeah, okay, thank you.
Yeah, feel free to follow Pedro afterwards. Our next speaker is Xavier Pugh. He is a PhD student at the Computer Science and Artificial Intelligence, so CSAO at MIT, advised by Professor Antonio Torralba. His research combines computer vision, machine learning, and simulation platforms to model human activities to build better helping agents. He organized the Social Intelligence in Humans and Robots workshop at ICRA and RSS 2022. Passing off to Xavier.
Okay, let's get your window up here. Is that you? Presenters. We have like 15 minutes to go over. You want to put it down here?
All right.
Awesome. Let me spotlight you. Oh, the camera's off. That's fine.
Should I turn on the mic?
No. If you want to turn on the camera, then I can spotlight you.
Sounds good.
Okay. Get away.
Okay. Well, hello everyone. Sorry for the technical difficulties. Thanks so much for the introduction and for the invitation. Today I want to talk about our work on neurally guided online probabilistic assistance. There we go. And this is joint work with Tianmin Xu, Josh Tenenbaum, and Antonio Torralba. So on this work, we want to build agents that can assist humans in everyday tasks. And for this, what we want to have is really agents that can essentially infer what are human goals in a fast and accurate way and coordinate with humans in doing these activities. Now, there have been a lot of really great works on multi-agent cooperation, but in many cases, they focus on the cooperation part and they assume that the goal is shared and known across all the parties. Or when they really focus on like inferring human goals, they are generally focused on robotic settings and tasks, which is a realistic and challenging setting, but in pretty constrained scenarios and pretty constrained tasks. And what we would really like to do is to build agents that can assist humans in general scenarios. So in prior work, we basically use VirtualHub, which is a household human simulator, to study these problems in these kind of challenge household activities. And what we did was to basically propose a challenge called Watch and Help, in which we would study how to build agents that could assist humans in those tasks. And so in this challenge, we would have a main agent that will be playing the role of a human, and a helper agent that will be trying to infer what the human was trying to do and help them in performing this task. And so the key here is that we would do this in two stages, called Watch and Help, in which first we will let the helper agent observe the task, the main agent doing the task alone, and then the two of them would be coordinating and doing the task together. Which means that for the helper agent, it could basically do the task in two stages. First, just inferring the goal, and then cooperating with the human in doing the task. Now, this is not a very realistic setting because it requires us to have like human all the time demonstrating the task before going into collaboration. And if the agent gets the task wrong, then you will get the agent really not helping at all. So in this work, what we propose is to do this in an online setting, which we call online watch and help. And so in here, what we would want to do is to have an agent that can concurrently infer what a human is doing in an environment, while at the same time helping the human do the task with what it knows about the task so far. And so this is very challenging because, especially at the beginning of the task, it's impossible to know what the human is trying to do. Like if you see the human walk into a kitchen, how do you know what it's like really trying to do here? And so what we propose to do is, instead of inferring what the goal is, we want them to keep a set of possible goals that the human is trying to do. Maybe it's trying to set up a table, or maybe it's just trying to like go watch TV. And so we will keep these sets of goals that the human is trying to do, and then take an action that is consistent with all these possible goals. In this case, I don't know what the human is trying to do, but probably it will be helpful for me to go to the kitchen to help them. And so as a human takes more actions, my goals will be updated and will be filtered, and I will be taking actions according to these goals. In this case, I don't know if the person is trying to put objects on the dining table or the coffee table, but I can help by bringing more objects to the human so that they can finish the task. And we'll keep doing this until the end of the task, really. So how do we do this? We do this through these neurally guided online probabilistic assistants. And so in this setting, we will have a state of the environment that the human is operating in, and the history of states, like basically all the steps that the human has done so far. We will be representing the environment as a graph, where every node corresponds to an object, and edges correspond to like special relationships between objects. And what we'll do will be to first train a neural network to, given the history of states that the human was in, try to predict what is the final state of the environment, what was the human trying to do. But we will not be predicting one final state, we'll be predicting a set of states that the human may be like, that represent the goal that the human may be trying to do. Based on the current state and the states that we predicted, we will be filtering some, like what are possible goals that the human is trying to do. So we will look at the actions that the human is doing and see like what are the states consistent with that. And so that will give us a set of possible states. We will be planning for these states. We will use a planner to figure out what are the intermediate states that the human was trying to reach in order to do each one of these goals. In the end, these states correspond to like goals that the human was trying to achieve, for instance, grab an apple or put it somewhere. And then we will be selecting one of the goals that the human is trying to do. And the way we will select this goal is by measuring whether it's likely according to what the human did so far, whether by helping in this goal I could help the human, or the human would just do it like faster alone, and finally whether I'm messing the environment. So I don't want to just start doing a lot of actions if I'm not sure about what they're doing. I'm happy to go over more details on the poster session. And finally, once we have like a sub-goal that we want to help the human in, we can run a planner and just figure out how to take an action to help the human in doing this activity. And so this kind of like shows an example of how this like helper agent operates. On the left, we have the helper agent, and on the right, the human doing the activity. And here the goal is to put 3 plates into the table, 3 glasses, and 3 forks. And so on the right, I know it's very small, but you will see how the inference of what the helper agent is doing will be evolving as the agent takes actions. And so first, the helper, like the human, will be going to the dishwasher, and that means for sure that the person is not trying to operate with salmon or anything like this. The moment that the human goes to the table, we know that the task doesn't have to do with like setting up a coffee table, and so on and so forth. So basically, I'm going to skip on the interest of time, but basically as the human takes different actions in the environment, the helper agent can infer what the task is trying to do and help them in performing the task effectively. What is cool though is that this leads to some interesting behavior. So that I was showing some of those, but something that will happen is that in some cases, maybe the helper agent will not know what the task is about, but the best way to help is by maybe giving an object to the human so that they complete the task. So this was one example in which the helper agent inferred that it didn't really need to put an apple in the fridge, and it was enough to just give the apple to the human so that they would like put it into the fridge. And here is another example where the helper agent sees the human doing a task, setting up a table. It grabs 1 fork to help the human, but when it sees that the human is gathering other stuff, it'll just put the fork back because it realizes that it doesn't need to help in like putting the rest of the forks. So we have some interesting behaviors, but we also want to evaluate whether these agents are helpful. And so for that, we will basically measure what is the speed up that we get when the agent is collaborating with the human and when the human is doing the task alone. And we test like an agent that takes just like random goals in the environment, an agent that basically tries to help as much as possible without understanding what the goal of the activity is, an agent that works, as I mentioned, but with a goal proposal that is just random. Not that all of those actually have negative speed up, so they are really not helping. Then a planning-based agent that uses predicted possible goals, and our method that uses predicted goals and tries to infer about like how to help with all those goals. Note that our method is like the best performing one, but the benefits are even higher when we have very few demonstrations, and therefore our network that proposes goals is actually very inaccurate. We finally use like test 3 of our baselines, and we basically try to run these baselines not with another agent, but with real humans doing this task. And what we find is that this is just like an interface on how like people are interacting with the simulator. But essentially what we find is that when helper agents help humans, they actually perform very similarly than when we help our helper agents. So this shows that our framework is like a good setting to study human-AI collaboration. So I really hope that my talk inspires some future work in like building assistive agents in general scenarios, and that we can help move this like field forward. Thank you all for coming, and I'm happy to take any questions. Yeah. I can repeat otherwise. Yeah, so he asked how to define the goals. So in our case, what we do is we define the goals as like logical predicates over the environment, basically saying like there could be like n forks on the table, 1 plate on the dishwasher, and so on. And we basically define the goals so that there's some element of predictability, because otherwise it would be impossible for the helper agent to predict what the task is about given some human behavior. Sorry?
The user has to choose the goals before.
You said, do the users have to choose all the goals beforehand?
No, we design a data set of goals and we like randomly sample from this data. So we basically define a generative process of goals. Let's say, okay, if I want to set up a table for 3 people, I will randomly sample some objects, and that will like limit to a set of like logical predicates that represent the goal.
So like, how does is there anything that the agent uses to distinguish between actions that are like not helpful versus just neutral? Like, how does your system?
No, that's a good question. So the question is how to distinguish between human actions that are helpful versus not useful. So in our case, we just predict, our neural network just predicts the final state of the environment given the states. We don't take into account the human actions. So we just care about whether the environment is evolving in a way that will help us predict the final goal. If a human does actions that are, I mean, in the end, we are assuming that the human will be somewhat rational, which is, I know it's like an over hypothesis, but assuming that the human is doing what it should do, like the agent should be like working on that. I have to say that if the human did something that is not reasonable, because the likelihood of this goal would be very low, the other factors would matter, and so the agent would try not to do that to avoid messing the environment or doing other things.
I was wondering if you thought about doing this in a fully, like in this is a simulation of the real world, but like for things that humans are doing on a computer. Like I could imagine, I was thinking of is like you do a lot of manual annotation of data for labeling data, and I was wondering if you could have like something that's doing helping with that annotation.
Yeah, that's a very good question. So the question is whether we can do this in settings that are not household activities, but in like annotation and other settings. So I think that one key of our approach is that we have a planner that, given a goal, decides what actions to do, and that will inform what the helper should do. I think that if we can like design a planning process for annotation, which I think that we could, or maybe we could like learn it, then I'm pretty sure we could apply this same approach. Thank you.
Thanks a lot.
Thank you.
Oh, thank you again. I'll introduce our next speaker. So Mandy Azabu is a PhD student in machine learning, and a member of the Neuro Data Science, or NERDS lab at Georgia Institute of Technology. He received a MS in engineering from Central Supélec, France in 2019, and an MS in computer science from Georgia Tech in 2020. Mehdi's current research focuses on self-supervised representation learning for neuro and behavioral data. Handing it off.
Okay, let me just get you pinned. Yeah. Un momento. Oops. Sorry about that. Okay, you're good to go. And you'll be on this camera here.
Okay. Thank you for the introduction. Hi, everyone. So today I'm going to talk about our work on doing representation learning on behavior that unfolds over multiple timescales. So the motivation of our work lies on this observation that behavior, or at least the factors that underlie the behavior, unfold over multiple timescales. And so basically at the second level, we might observe short-term behaviors like running or a mouse chasing another mouse. And then at the hour timescale, we're going to find that the motivational state or the time of day might actually impact how the mouse is moving throughout the day. And then even when you go to the even longer timescale of days, we found that age, for example, is another factor, or even there are other factors that differentiate one individual compared to the rest of the population, like strain and morphology. And so it is clear that in order to be able to understand and analyze behavior, we need to be able to capture all these factors. And so we propose a method called bootstrapping across multiple scales, or BAMs. So the idea is to use representation learning to learn representations of this behavior. So typically, in representation learning, at least for sequential data, we would consider neighboring time points and then bring their representation closer to each other. So here we define a positive range over which we're going to take these neighboring points. And then the challenge here is that for multi-time scale data, we don't know how to exactly define this positive range. And so here, for example, we can see that over this short duration, there's going to be a running behavior. But so we would define the positive range based on the fact that the embeddings will be consistent or semantically significant in that small positive range. But then if you're talking about the other factors that over longer timescales, we would need to use a different positive range. And so in our method, we're going to learn short-term embeddings and use the specific positive range, and then also learn long-term embeddings, which will have different amounts of, or a different rule for bootstrapping the representation. To do this, we're going to introduce this architecture, which is basically a series of temporal convolutional networks. So what is cool about temporal convolutional networks is that we can define the receptive fields. And so in this case, we're going to have 3 of them, each with a different receptive field. And this allows us basically to aggregate this information at these different timescales that we're looking at. To train this network, we're going to have 2 different learning objectives. The first one is what I just introduced with the representation learning. So we're going to have 2 different rules for the short-term embedding and the long-term embedding, and use self-supervised losses to learn these embeddings. And then the second learning objective is going to be basically concatenating all these embeddings that we learned at different timescales, and then feed them into a predictor which would predict the future actions in the next 1 second window, for example. And so the idea is that by having the model learn dynamics of the behavior, it should also capture all these factors that are important for it. So we apply our method on the mouse triplets data set from the multi-agent behavior challenge, which we'll hear about later. So the idea is to learn these embeddings from tracking data, so a series of key points from 3 mice. And then what is cool is that there are 13 different tests that allow us to evaluate the representational quality of these embeddings. and these span across different multiple timescales again. So the time of day is like a long-term embedding, and then the chasing behavior is like a frame level one. And so these are our results. So here we can see that by evaluating the qualities of these representation, we find that we get competitive results. But also, what is interesting is that when we look at each embedding space, so the short-term embeddings and the long-term embeddings, and just try to read out these different tasks just from those spaces, what we find is that some tasks actually only reveal, like some factors are only revealed in one or the other space, and for other tasks it actually requires both timescales in order to decode them. And so to conclude, we were able to design an architecture and a set of self-supervised tasks that allow us to learn representation of multi-timescale behavior. But also, we found that in order to understand and analyze this behavior, it is critical to be able to capture these factors at different timescales. To end, I want to thank all the team that is involved in this work, and yeah, thank you. Questions?
Okay, so I'll start actually with a question. So as you're probably aware, we have the second data set of fly groups, which has a variable number of agents, also a larger amount of agents. Mm-hmm. I'm curious if your method can generalize to such situations, and like if there's anything you should change in the framework, or if it can just be applied.
No, definitely. So a thing that I might have not explained here is that when we apply our method to the mouse data set, we actually model each mouse individually, and then on top of that use another network to model the interactions between them, like a simple one. And so definitely in the case of the flies, we can model each fly independently and then capture that interaction the same way. So it would be able to generalize for a dynamic number of agents at the same time.
I have a follow-up question.
Yes.
No, okay. So can you describe that network more? So does the network that produces the final group embedding need to know the number of agents, or can it take in a variable number of agents?
So the current version that we have this is like a simple MLP that captures those interactions based on the embeddings from each mouse. But I also tried, I think, graph neural networks would be the ideal solution for. Yeah, I actually tried this for the mouse data set, and then by modeling those, by having graph network, you don't have to like have a set number of agents to model the behavior in general.
So are the current results with the graph neural network?
No, just with the MLP. Okay. Yeah, the graph neural network is more recent stuff that I'm still working on.
Okay, that would be interesting to see.
Definitely, yeah.
I guess it's time for the panel to set up the panel discussion. So, thank you again.
Thank you so much. Thank you.
Okay, so for the panel discussion, any of the invited or submitted speakers, if you'd like to bring up a chair and hang out around the screen here. We're going to,
Or are you using your laptop?
I think turn around the camera, yeah. That's a good number of chairs. Let's do a gallery view here. Yeah, so the speakers who are in the chat, Alessandro or Yisong, if you can turn on your cameras. Okay, so Are we settled?
I think so. I'm gonna move this way. Oh, we're gonna put a chair there. Are you gonna be in the panel?
I was gonna be here, so I
Okay.
moderate.
This is the microphone. Hopefully it reaches.
Somebody want to say something?
Hello?
Folks on the Zoom, can you hear the panel when they talk?
Hello?
Greetings, hello?
Oh no.
Maybe you guys can move this way a little bit.
Yes. Yeah, you can do this. Can you hear us?
Okay, that's a yes. I think there's just a little bit of lag, so we'll have to take that into account when you folks answer questions. Okay, yes, so for the various audiences we have on Zoom and YouTube and in person. All right, so thank you all for coming to the workshop and for presenting the research. This has been really fun. The goal of this panel discussion was to kind of bring up again some of the topics that we saw in different people's workshop talks and have a chance to try to identify common grounds between the different things that we saw presented today. To start things off, I wanted to just ask each of the speakers who presented one question of how well you feel that you know the behaviors of the agents in your system. Like, how well do you feel that those are cut and dry, identified things? And versus how much do you think that there's something that still needs to be discovered from the data? And how much is this behavior discovery an aspect of the work that you do? So if you want to take turns.
Oh, okay, I can go first. The way I ended up working on behavioral data is actually started from me working on neural data. So the idea of being able to understand what's basically going on in the brain from analyzing the neural activity. And so what became quickly apparent was the fact that behavior was actually more complex, and I had to initially basically manually go through my data, look at the videos, and be able to describe the behavior. And then every single session that I would look at had different different types of behavior that was going on, even though it was the same type of animal. And so that's why I got interested in the way of discovering these behaviors in an unsupervised way and revealing maybe some details that I wouldn't be able to see by myself, especially as someone who's not an expert in those, in identifying those types of behavior. So yeah, definitely the discovery of behavior is a challenging and important aspect of it.
I'd say the same. So actually, similarly, I got into the behaviors that I see in terms of thinking about originally computation. And I'd say the individual agent behaviors are pretty well characterized of what's possible in the cellular systems we work in. But what isn't very well characterized are what are the group behaviors you see in new contexts. And so that's, I think, mainly the main active area of discovery I'm interested in is what's now possible if you have new environments and new scenarios where these cells can work together. What are the things they can possibly accomplish that we haven't seen previously?
Oh, I guess for me, how I started was looking mostly at supervised learning algorithms where experts were defining behaviors and we were training machine learning models to reproduce their annotations. But then it kind of became apparent to me that I guess one is that there's a scalability in that there's so many new behaviors that need to be defined all the time. So that if we keep training supervised models, we are going to have to train a lot of them. The second one is that expert definitions also have variability. So we also want models that can help us figure out somewhat of a consensus amongst the experts or figure out why they disagree. And so I think these unsupervised or behavior discovery methods could be one potential way to help us do that. But I guess they're an area of active work to figure out the best way And I guess that's partially why we have the challenge this year, is to help us discover this.
It looks like Zoom is having trouble hearing me out of my microphones over there.
You want to sit over here too? Oh, your questions are over there.
Okay, I will mosey over this way, closer to the microphone. Okay, sorry about that for the folks on the Zoom call. Yeah, so the question is how well you feel like, or do you feel that the behaviors in your field are kind of cut and dry, well-defined things, and everybody's studying the same thing, versus how much do you think behavior discovery is an important part of research in your area? Can you guys hear me now? Yeah. So if folks on the Zoom call would like to chime in.
Can I add something? Can you hear me?
Oh, sure. Yep.
Okay. I think in our evaluation and our training of behavior, we're thinking of behavior as binary things. It's like it's either this or it's that. But in real life, behavior is not discrete, so to say. So there is also behavior in the middle. It's not flipping from one state to the other. So a lot of things that we panel as wrong are in fact, I don't think that they're wrong, but they're labeled wrong. So I think maybe we should improve our evaluation. And this is the cause of inter-subject variability and intra-subject variability. Because the inter-subject, inter-observer variability is really big. It's about 75% of the frames only that is labeled as the same. So this is something we need to think about, I think.
Yeah, no, that's definitely something that we've encountered with past people.
Yeah, so not all errors are the same, have the same value.
I'm curious, is that a thing for folks working, I guess maybe Alexander, for folks working in self-driving cars or human behavior, where it feels like we don't quite know what the agents are doing? Or do you think it's an easier problem?
I think it's actually, there's an abundance of data for which catching the distribution of behaviors is not too hard, and there are many papers out there from Waymo and other companies that show that actually we can model pretty well the behaviors of agents. What is really hard to catch are the corner cases or the long tails of events. When something really unexpected happens, there it's really hard to model how people would react. So I guess it's probably more like a lack of data rather than an intrinsic hardness of modeling that kind of behavior that we observe on the streets.
If I can add to that, I think there's actually an interesting, maybe more philosophical question of what do you mean by behaviors? But maybe you made the question vague on purpose. So I would say, yeah, it's kind of similar to what I guess Elspeth said, that maybe we don't know, we have this internal representation of what is the discrete set of behaviors we would expect for a particular system, but there can be a lot of things in the middle. It's not necessarily just 0 or 1, it can be a grayscale. And I would say that, at least in my work with fireflies and with honeybees, a lot of what we do is actually finding new behaviors. And sometimes we know what to look for and sometimes not. So this is where machine learning classification can be really helpful in gathering more data.
I'll maybe jump in. Since we seem to be heading in that direction anyway, my next question was going to
Actually, I'm not sure. I think since I work with like deep learning methods, I tend to like think in terms of like continual variables, because it's like at least in the unsupervised case, it's like untractable to define a binary variable for every single one of the behavioral factors that we would want to capture. Obviously, for interpretability, like having those discrete variables makes more sense and makes it more easy to study. But I guess I would tend to go for continual variables, since there are better tools that don't require labeling or defining those variables. So yeah.
Yeah, we haven't really taken the leap to having, currently the way we deal with things is we think about the variables as continuous as a signaling state, and there's an internal and external state, and how well are those states synchronized, but we haven't taken the next step in terms of being able to take those continuous, the continuous internal variable representations and putting those on sort of more binary or discrete behaviors and classifying those behaviors. So that's still, I think, a next step that people in microbes really haven't taken.
I guess for me, since coming from the machine learning side, it feels always to me like it depends on the downstream task that the scientist is studying. So like, I guess for some tasks, like I guess if the mouse did a nose poke or not, something like that could be defined in a discrete way, and it might be like kind of simple to do that. Whereas with naturalistic behavior, I feel like we could discretize it so that we can better understand what the animal is doing. And in fact, like a lot of human action recognition do have discrete labels on when a person is running. But then I guess people here are probably all aware that it's really hard to define start and stop times of these discrete behaviors. So then we often turn to a continuous space. So, I mean, of course, both methods are useful, and I feel like it depends on the downstream analysis.
Hi, can I jump in real quick? Can you hear me? Yep. Great. Hi, everyone. For those of you who don't know me, my name is Yi-Sung Yu. I'm on the advisor panel for this workshop, and I work in various forms of multi-agent systems, from work at being Jennifer's advisor working on animals to I currently am on sabbatical at a self-driving car company at Argo AI, and I also have collaborations with various video game companies that are making massively multiplayer games. And I think the question of discrete variables is a very important one, depending on what your downstream goal is. If you have a very sophisticated, if you will, downstream goal, I think you almost need these discrete variables, right? Like, for example, if I want to publish a scientific study saying that this brain region activity correlates with increased aggression, such as increased number of biting, then you need a discrete variable that says at a binary level of biting or not biting. It's actually very hard to make progress without that. Similarly, for self-driving cars, we need a discrete variable saying whether or not this pedestrian is yielding or not yielding to the autonomous vehicle. And especially in a large team of 20 to 30 people designing a system and trying to understand a system, without these discretizations, the lack of interpretability becomes a problem.
Yeah, I think your output can be discrete, and for downstream tasks to continue it, or for event end evaluation of your system. But you can, for instance, have a more tolerance on the boundaries when you're training. I think that would be a good idea.
I'll have one for everybody who's on the wants to chime in and get things started. I'll say I think that for my work in study of animal behavior, my impression was that the discrete labels was one of the most
And sorry to interrupt, but could you please get closer to the other microphone so we can hear you?
Can you hear me better?
Oh, so much better. Thank you. Yes.
Okay. So I'll remind myself the questions. The question was, out of all of the talks that you've seen at the workshop today, what are the things that you feel are most in common across the different approaches that people are using and the problems that they're trying to solve? And what do you feel is most specific to your particular domain that you're working in?
Can I mention one thing? Yeah. I don't know if you, the thing that struck me the most actually in the workshop was kind of the dichotomy between the computer scientists and the engineers. We're kind of looking at small numbers of agents, maybe like 3-ish, and then the people working on biology who were looking at thousands of agents, and like things that seemed like a big disconnect between the two areas.
I guess I can jump in with something that I thought was really cool, is that, for example, in Allison's talk, there was communication between the cells with like the propagation, and then in Or's talk, there was like communication between the fireflies with like flashing, and the methods of communication seems different, but like also connected in that way, like as well as self-driving cars when they're observing pedestrians. So I guess there's some sense of this like communication and coordination between agents that, I mean, of course, it's a multi-agent behavior workshop, but it seems like to come up in multiple themes, despite the difference in domain and how the agents actually do that specific coordination.
So actually building off that point, I think somewhere there's a nice similarity and nice difference is this idea of timescales and temporality. So something I really enjoyed in the workshop was the role of how sort of different behaviors have different timescales and how far back you have to go to understand what this behavior is. It was sort of a theme that I saw come out in a few different talks. But I think partly what you're calling out there is actually there's different kinds of communication. Your ability to coordinate it actually sometimes brings in different timescales. So a nice thing about both mine and Orit's work is there's actually communication can't be instantaneous. There's actually a time scale of a delay of how you get to another cell or another agent in the system, and that can actually, that effect changes how behaviors are coordinated and has different downstream effects in how you model those systems. But in some senses, you could actually think of communication as effectively instantaneous, like in neural systems, and so you lose that extra time scale and the different kinds of effects it has on how they're able to coordinate their behavior. So for me, that sort of sense of different things that time scale brings into the problem is really key at both how they're the same and different.
And if I can add a small thing on top of that, I think there's an interesting physical delay in how the signal is being propagated. For example, if it's chemical, then it's limited by the diffusion coefficient. To the behavioral delay, maybe there is some, internal processing that is being done on the cellular level or on the just a full organism level, and that causes some delay as well. And it's interesting to distinguish between the two, I think.
Any other thoughts from the non-biologists? I don't know, Alessandra or Nicholas, if you had thoughts on what overlapped between the biology talks and yours and what felt very foreign and not important. Well, maybe don't say that, but felt different.
Well, I think most of the things were actually mentioned already by other panelists. I think there's the sort of what I had in my talk was a small number of agents that had relatively rich behavior individually that were interacting, whereas I guess what featured in many of the other tasks, especially the more biological tasks, was over agents where a large number of agents where each individual element of the system had much simpler behavior, and the intelligence that arose really from the large group. But then maybe what's shared is that the interest in thinking about different scales for a system, the different scales at which a system can be considered, which is at an abstract level true for what I've been looking at, and also what appeared in other talks.
Can I actually ask a question to build up of that? I'm really curious if that you think that in order to, so that's a really interesting distinction that I think the agents of biological systems have a equally rich individual repertoire of behaviors. It's just that you don't need them to get the emergence of, of the sort of larger scale emergent behaviors. Maybe the details of both are important, but to get the little, sort of dynamical features correct, you might need those little bit of details, but you actually don't need them to get the big scale emergent phenomena. Is there an analogous sort of thing you can think about where you actually, you don't need those sort of, you don't need to model those behaviors as richly in order to see sort of the sort of larger group dynamics emerge in games? I'm actually not sure. Do you really need all the details, or is it?
Well, I mean, I think the work that I was presenting was perhaps quite particular in that I think, so yeah, it was quite particular in the sense that it was sort of aiming at building up the behavior across all scales, right, and really modeling it on a relatively fine-grained level. But I think then the work that I didn't get to present, which is sort of, which focuses more on describing human behavior in soccer games, I think there already a lot of the details are being abstracted away simply because you can't get the measurements. Right? You can get measurements of players on the pitch, but you can't get like necessarily all their poses and other variables. And it's perhaps also not necessary for some of the goals of this work, which is sort of more situated in the world of sports analytics. So in that sense, just superficially, I would say yes. Otherwise, I think it's probably something to do also with the size of the system that's being considered. If you have a small number of interacting agents, then probably the details are more relevant than when you really think about sort of large-scale systems with many interacting agents. If I think about crowd dynamics, I mean, I'm not an expert at this, but then I think many of the details are not relevant so much, and you can describe the behavior of individual agents at a much simpler level.
Just building off of that, do you think there is a phase transition that happens in behavior, or is it just that once you get to a bunch of agents, you can't capture the individual choices that they're making? Is it just different kinds of behavior that you're describing? One is the moment-to-moment actions of an agent with its surroundings, and one is the action of the whole population. Maybe that's the difference.
Was this a question for me? I couldn't really understand.
That was a general comment, I guess. Is it that the behavior changes when there's many agents, or is it that what we're calling behavior changes because you can see emergent things that you wouldn't see if there were just two agents interacting with each other? I'm honestly not sure.
I feel like I
agree
with that there's kind of like, the same way that there's like short time scales and long time scales, maybe there's like
local interactions,
and then like you have to aggregate when you get to a big crowd, say, or something like that.
And you can't possibly store enough information about all possible states of all possible other things. You have to coarse grain even in the system in order to have that group behavior. Yeah.
But I wonder how many soccer players you need to have to represent them as a crowd versus individual players.
Well, I guess behavior strategies of your soccer players can change if there are like 500 people on a team.
And I guess it goes to, I don't know if Alessandra can hear me, sorry, I'm kind of far from the mic, but it goes to kind of the type of thing that you were talking about of how do you scale these things to many interacting agents.
Were you guys able to hear that?
I only heard soccer players, and that was about it.
Do you want to summarize it close to the mic.
Oh, I was just saying that, you know, kind of what hit on the theme, I think, of Alessandra's talk of how you scale to larger numbers of agents. You know, whether you try to do that exactly with kind of dynamic programming types of solutions, or if, you know, you mentioned, for instance, that, you know, a more heuristic way would be to just consider the closest neighbors, for instance.
Another question I had that came out of a couple of the talks today, let's see, Allison's talk and Alessandra's talk, was the difference between an agent that is sensing and inferring the actions of other agents in its environment versus the effort that an agent puts into signaling its state and its intentions to other agents. How do you see the balance between inferring versus communicating in both how agents actually behave and in how you might design virtual agents? Do you focus all on one or all on the other? Are there both of those things present in the system? Do you treat them differently? All of those things.
Can I ask a question to sort of get a distinction here? So you say inferring versus communicating. Is the assumption that a communication is a perfect representation of what one state of the agent state to another agent?
Right.
Like, or is all communication not an inference?
I guess it's there's still an inference part of it, but how much does an agent attempt to communicate its state to other agents versus how much is it just focused on inferring their states?
Oh, I see what you're saying. It's how much is transmitted versus just assuming it's
Yeah. So in your case, I know that this signaling was a big part of the collective behavior, and I was wondering, like, how much do they change their kind of signaling based on their objectives, or the range of their signaling based on their environment?
Yeah. So something I find interesting is that, you know, I used to think intuitively that these agents can easily communicate with many different agents over long distances because it seems diffusive mediated, right? This is a molecule that they can excrete. It actually turns out that the ability to communicate rapidly drops off much faster than you would expect with distance because they're actively shaping the diffusion of this molecule with other molecules they excrete. And so you actually can't really rapidly communicate very far unless you have many different agents. So you're actually really only able to be closely coupled to those around you and get a pretty local readout of your state. If the other agents aren't really nearby, you actually infer your environment that there's very little around you, and you sort of continue a random walk. You have to be very close together. So I would say agents are not, I think of agents as sort of just like broadcasting I'm here, but other agents' ability to actually sense that they're around drops off pretty rapidly, so that they're doing a lot of just trying to basically they're inferring a lot about the state of the system, and actually they're doing it pretty badly, is the way I think about it. It actually is pretty remarkable to me that they manage in this random walk scenario to eventually get everybody close enough together to do this. And the way they do this is through being really robust and allowing themselves a lot of time to do it. Everything I showed today is a relatively short time scale, and I'm putting that in air quotes because, like, a day is actually not short, but if you space them out so they're even like half as dense, they'll do the same process over 3 days. Like, they give themselves a lot of time to have really bad information about the system around them and just find each other and get closer and stick to each other. Because it is a life or death situation, but apparently they didn't come up with a better solution.
From a learning perspective, it's not so clear to me that there is sort of a strict distinction to be made. So at least for the system that are presented in my talk, the agents have the ability to implicitly, and they do to some extent, infer what their teammate or their opponent is currently doing. It's not made. There's no explicit mechanism built into the system, but they observe the history of the state of the system, and can from that draw conclusions, I guess, and that there's a sort of incentive to learn to do this is incentivized by the reward. And then I guess agents could in principle learn to communicate, even though there's no explicit communication channel, through their body, for instance, and performing actions that maybe only serve for the purpose of communication. I guess this is hard to detect. I think this becomes more relevant in a more fully partially observed setting. The setting, I guess, was for simplified was relatively fully observed, not fully, but pretty well observable. And then one can obviously introduce extra sort of channels for communication, right? That maybe are easier to use than to use the body, or that have a longer distance or something like this. But it's, so at least in this setting, I'm not sure whether this distinction is sort of very clear, or whether one can draw hard boundaries.
I guess one distinction I saw between Nicholas, your setup, and the slime molds was that, well, there are a couple differences between the two, but one of them was that you have competition in your setup versus in the slime molds, it's, at least in the data you showed, it was purely cooperative. So maybe when you have agents that are competing, there's less of a reason to signal your intentions because the other agents that you're competing against will see them. Is that?
That's I think one of those questions that comes up a lot in the field. So actually, there is in some sense a competition, like define competition. About 20% of the cells are going to die, and there's something about how that process unfolds that dictates who's going to die and who's going to live. In the system I showed, everyone is actually identical, or at least genetically identical originally. But you can actually mix them together, and they appear to be able to coerce one another to take on different fates, and there is a competitive aspect. There's something known about the sort of times at which those competitive parts tend to come into play, but it's not entirely known. So it appears to be cooperative, and it is cooperative in the sense that some agents of almost all types will end up being successful to some extent. You can find complete cheaters or complete altruistic agents, but actually largely in natural systems there is a mix. But it is actually unclear that we have, even at any given stage, entirely altruistic, non-competitive behaviors. So, we can certainly model it as completely altruistic and cooperative, and it works out, and it seems to reproduce the general results you see. But if you start to dig into various, nuances of it, it does appear that there is some sort of coercion and competition, and that those could potentially change. I would say not qualitative outcomes, but quantitative outcomes.
Maybe the closest we had to cooperation was the self-driving cars.
I think that's
like there you want to communicate clearly what your plan is. So do you see, is there any evidence that agents are making actions that make it easier for their future intentions to be read out in these settings? Or is this something that self-driving car companies have thought about?
Is that a question asked to me?
Either you or Alessandro, yeah.
Okay, sure. I'll go first real quick. At Argo AI, where I'm working, this is not explicitly modeled. Part of that is due to the asymmetry of specifically autonomy, and the asymmetry is that if something bad happens to the car, we lose some money. If something bad happens to another actor on the road, it costs a lot more, both in terms of the possibly loss of life, financial damage to other people, reputational damages to the industry. And because of that asymmetry, the companies, at least Argo, I imagine the other ones as well, treats this very differently. And so, we're very complacent, and we're very, permissive. So, we basically do the most conservative thing possible. And so this type of communication you're suggesting is not something that is very sophisticated on the vehicles right now. Although in principle they could be, but it's just that the asymmetry, it's not really a competition. It's not even a cooperation. We just don't want to cause trouble,
You're muted.
We can't hear you, Alessandro. Maybe your microphone?
Can you hear me now?
Yep.
Oh, okay, sorry. I switched to the other microphone. So I was saying that, I'm not in the industry. I don't work for a self-driving vehicle company, but I know that there have been, let's say, funny studies around also simply on whether you should signal whether the vehicle is autonomous or not. And it turns out that it's often the case that people would start taking advantage of it because they know that it's autonomous, right? So there are even people that would just jump in the middle of the road because they know it will stop, things like that. So for sure, in general, especially if I think about my niche or the examples that I've shown in the talk where you have these microscopic interactions, when you add communication or signaling, which is fruitful, it simplifies a lot the problem, because once you commit to an homotopy class where we decide who's going to go first and what's the sequence of with which we take the shared resource of the road, that simplifies a lot the problem. On the other hand, there are this evidence coming from real world that it might not be always the case, that you wanna unveil what you're about to do, or what's your nature, let's say.
Maybe I'll just add one more thing. It can go both ways. So, one of the most interesting scenarios that I encountered was, the autonomous vehicle is trying to make a left turn against oncoming traffic, and so it does not have the right of way. This other driver decides that they want to be nice to the autonomous vehicle, so they slow down and wait for us and lets us take a left turn. And the logic on the AV says, I don't have the right of way, so I'm just going to sit there and wait. And then the other car says, just waiting, and then eventually there's some logic on the AV that says maybe this other car is stuck, and so I should just go. And then both cars start going at the same time, and so it was unintentional bad cooperation, if you will.
Now I want to see these cars in a rotary.
Yeah, that's awesome. I guess, let's see. Changing topics slightly, we've talked a little bit about representation learning and understanding the representation that an agent has learned. What are some ways you see of evaluating those representations in terms of their quality or the utility? How do you think about that? If you are interested in this topic of learning representations of agents' behaviors.
I feel like I should pass on that because I work on the challenge. I guess I'll just start.
We had our own approach that Jen's going to talk about in a little bit, which was based on human-defined labels. But are there other things that you think are important for evaluating the quality of a learned representation?
I think I can say something. I think that's a really challenging question, like in representation learning in general, and the reason actually I started working on behavior is because I didn't have ways of evaluating representation quality for neural representations, and so if we cannot understand, like in that case, we don't even have the capacity to have humans label neural activity because we don't understand what's happening in the brain, and so for behavior, the advantage that we have there is that it's possible to get those human annotations and be able to say based on those features whether the representation is good or not. Obviously, there's that first way of performing evaluating the representation, but yeah, there's always obviously the bias that we introduce with defining the labels or what we think is the representation should encode. The other alternative is defining downstream tasks, maybe by having a way of evaluating representations through maybe how predictable they are in the future, or how they're maybe distinguishing between populations. So I think, for example, if you know that two different populations should have distinct behaviors, you can verify whether those representations might reveal those differences. So in that sense, we're not manually annotating the data ourselves, and that could be another way of evaluating the representation.
maybe I have a very particular view on the topic, in the sense that I'm interested in representations that can actually be translated into behavior ultimately, rather than just, I mean, I'm also interested in representations for the analysis of behavior, or to characterize behavior, and sometimes the distinction isn't entirely, isn't a hard one, but for many purposes I'm interested, when I talk about representations of behavior, I mean something like a skill in some sense, right, that can be translated directly or indirectly into something that an agent ultimately executes, or something that can inform learning of executable behavior in the end. And then I guess there's sort of maybe different desiderata depending, I only lightly touched on this in my talk, for instance, the extent to which such a representation actually constrains the learning outcome, or maybe just regularizes, shapes it in a soft way, and also what the computational effort is to translate the representation into something that can ultimately be executed by the agent. And so I had two different examples in my talk, the sort of the skill module, where you can send a command in and then it does something, and then sort of the behavior prior, which is more like a cost term that you need to kind of copy into your learning agent through a learning process itself. So there are very different costs involved in translating the representation into something that's going to be executed.
I'd like to add something. I also think there's many different types of behaviors, and in the beginning of human activity recognition, like 10 years ago, there was data sets only with mapping and running, and jumping also. So those were kind of easy behaviors to distinguish between each other. And while in reality we have a much richer, much more subtle palette of behaviors. And I think that if you use a downstream task for evaluating and embedding, you should really take care that the classes that are in downstream tasks are representative for what you find in the real world. So there should be rare behaviors in it, and there should be short behaviors in it. Because if you keep using those obvious behaviors, like, sorry to say that chasing is to be, and that's very important to know, but there's a lot more subtle social behavior between mice, for instance. And I think for evaluating the heading, you should have evaluated full palette of behaviors.
Can I ask a question? Maybe because it's a field that I don't know so well, is there some form of taxonomy, for instance? Because I'm sure there's not a single level of description, right? Because there's different levels of abstraction, which you may want to characterize the behavior, and then maybe even different dimensions. Has to do work on characterizing just the ontology.
Sure, there's a lot of intergrams that are used by biologists that study rodent behavior, social behavior, and there's for instance pinning and bouncing of animals where they have play behaviors and it goes really fast. Or boxing behaviors when we box very shortly to each other and then separate again. They also have social behavior when they're not in contact with each other because they can see each other, and for rodents, smell is a little more important. So when an intruder is placed in a cage with another animal, the social interaction starts already without the animals being close to each other. So it's a long way, but I think if we want to evaluate embeddings, we should not look only at the things that we can, that computer vision experts can easily see. it's kind of, of course, I mean, it's always more difficult than you think, but I think that's important for everything.
Yeah, there's been a big drive from NIH and from researchers to try to increase the granularity and resolution with which people describe animal behaviors. We're at the point where we can record thousands of neurons at once or tens of thousands, but then we're correlating this big high-dimensional data set against, like, is the mouse sniffing or not, or is the mouse turning or not, and it's very coarse. So the hope is that an unsupervised, a data-driven method would give you a more detailed description of the animal's actions, but you're right, it's also helpful to have these human-defined behaviors, I think. And there are things that you might not necessarily be able to pull out of the data in an unsupervised way, or something that you still try.
Because that's, if you do an unsupervised dimension reduction, it's not the scarce data that come out. They come out most of the time. It's the bigger groups that you because you lose some of the variability in the PCA, for instance, you always lose 2% variability or more. But it could be that a certain behavior is exactly in those 2%. Yeah. So I think there's a limit to what we can do unsupervised. I think in the end we would like to have a system that works together with the humans, so you have both the unsupervised and you have the human who is interactively helping out, deciding what to focus on. But that's hard.
Yeah. I guess in the last 7 minutes or so, one question I wanted to ask all of the speakers again is what they considered a success in their field. Like, what is the ultimate goal, and what would, if you were to publish it, what would make you feel like you had kind of accomplished something and made like the next big step in your area of research as it relates to multi-agent behavior?
Should I just jump in on something? Oh, I don't know if it's like the ultimate thing.
It doesn't have to be the ultimate
thing.
But
I don't know if it's like, okay. I think one thing that would be really cool for me is if we had a hierarchical, temporal, and spatial model of behavior that is also interpretable for human domain experts, so that you can zoom in on various temporal and spatial scales and get interpretable descriptions of the agent's behavior at the individual level, at different time scales, and at the group level. I don't know if this solves every single problem, but to me, this model would be, it seems like it would be helpful based on some of the biologists I talked to from various labs. It would be useful to have something like that.
Yeah, if I can say something about that as well, I think I agree in terms of like what would be ideal in that situation, or at least my perspective of it is like having that interpretability and also decomposing basically the behavior into these multiple factors, and being able to for sure like learn it in an unsupervised way from a lot of data which is available, but being able to like present those explanations and describe it in like in a hierarchical way or like a decomposable way where you're describing different factors that are impacting the behavior, also definitely also describing the how different populations are or like even within a population how different individuals are different or like heterogeneous, and also describing those factors as well. Having all those layers, I think, would definitely help a lot of people with their analysis of behavior and like correlating it with other things.
So it's largely a better description of the behavior that's happening and that spans time scales, gives you just a richer explanation of things. What about things like forecasting or knowing what's causing an animal to do what it's doing? Is that something that you care about, or sort of secondary to descriptions?
I think, no, definitely. I think that's a really interesting aspect of it, and especially in the case of like multi-agent interactions, like being able to explain that, oh, this behavior can be explained by this interaction either with another agent or even with the environment, and being able to describe that, or like find that causality link between different elements is also super important part of that modeling, I think.
I'll try and take things in a little bit of a different direction. One of the big questions that keeps popping up in my field is how important is heterogeneity and where does it come from? Right? If you have a bunch of agents that are in theory basically identical, how do they break symmetry and drive these new behaviors? And is that heterogeneity is it driven by completely stochastically driven, or is it actually there is some internal state that some kind of cell has changed and that is required for the agents to break symmetry and get these kinds of behaviors? So people can you can model this where you have cells that are different and are pacemakers and act as leaders for the whole process. You can also model this where it is in fact purely stochastic and see the same situations. And so some finer way of observing different states and the behaviors and identifying if you be able to, I think I put a nail in the coffin of that question and say you can only get the behaviors we see, that richness, if you have one or the other, I think would be huge in the field. And we haven't quite figured out, I think, how to model and analyze behavior at that level of detail. And we also don't quite have the ability to get the data sets we need to uncover different biological differences that could be the drivers of those. So, but that would be huge if we could do that.
Any differences of opinions from the more engineering-focused folks?
Sure, I'll say something real. Oh, someone else can go if you want.
Please go ahead.
Okay, I was just going to say something very generic, which is agreeing with what everyone's saying, but saying it differently. As an engineer, I would say what I really care about is a level of understanding that admits an intervention that I have confidence in, which is basically agree with everyone else is saying, but just framing it along a different axis. What I mean by that is, I've developed a model that can capture something interesting about behavior, and I feel confident that I can run a follow-up experiment that tests a concrete falsifiable hypothesis, and I have confidence that this will generate an interesting outcome. In the context of engineering artificial systems in a multi-agent domain, I have confidence that I can quantify certain behavior that I can design a policy around, whether it be in a virtual world for gaming or for autonomous vehicles that I have confidence will improve the interaction with the other agents in the system. And another way of this characterizing in broad strokes what people have mentioned in the last 5 minutes is there's this tension between this global hierarchical view of behavior, which tends to also be macroscopic and coarse and perhaps not actionable in certain ways, and these sort of very targeted, fine-grained behaviors, which may not be the modeling which may not be able to scale to every possible scenario, but can allow very specific models in a very limited data distribution, both of which can be interesting in terms of the types of interventions you want to produce afterwards.
Yeah, I agree with what the speaker has said so far. I think in particular in the field where I'm working, I would say that one of the challenges is to properly incorporate the uncertainty that you have about the other's behavior. And in particular, really incorporate it, not just in academic way where you just put a Gaussian on possible things that the other can do, but really understanding all the consequences that can happen, and that there's a tail of events that would lead you to be at fault, a tail of events that would lead the other to be at fault, and all these, let's say, effects that are not properly understood, or it's hard to model them properly without oversimplifying, and to incorporate this kind of uncertainty into the decision making.
I have maybe another sort of comment that I think also broadly agrees. I think the difficulty in characterizing behavior is also reflected also appears when you try to synthesize it. Because as soon as you move beyond sort of end-to-end learning, where everything emerges in this one monolithic learning process, and you don't actually have to worry about
We lost him.
Are you still there? We might have lost you. Oh no. Okay.
Well, I'll fill time by just saying that I thought Yisam's idea might be something we could make a challenge out of. If you had like we as biologists that could maybe do experiments. If we had machine learning people come up with hypotheses and ideas of what they might, what the outcomes would be, and we all agreed, like, okay, we will do those experiments that you come up with, machine learning people, and see whether your predictions are correct. We had something that would tell us how unexpected those results would be to a biologist. That would be really fun to try. I don't know.
Yeah, that's something I'm wondering about. Is I realize we're running into the start of the challenge discussion, so I don't want to push this too much longer, but whether these models can create testable hypotheses or whether you still need a human to interpret the model is something that's really interesting to me. Yeah, since it's 3:02, I'll say if anybody has any concluding comments they want to make on that. Otherwise, we might jump to the multi-agent behavior challenge. Nicholas, did you want to wrap up your thought?
I think my browser didn't like what I was saying and decided to restart. So I think my point was simply that as soon as you have to modularize the learning process in some way, this notion of the taxonomy of behavior or how you break it apart sort of suddenly becomes important, because you have to think about either how you structure your architecture or how you structure the learning process. And I think that's actually something that has held the field back from shaping agents that exhibit complicated behavior for a long time, because we tend to think of all these particular interfaces between different modules, and that's simply not appropriate for the richness of behavior that sort of biological systems produce in the real world. Like, how do I communicate to my motor skill module, whatever that may be, what should be done? Is it a gait pattern? Is it a target to move to? Is it a pose? Depending on the circumstances, different semantics will be appropriate, and it's very hard, I think, for us to sort of to think about this. And the same, and this becomes just more complicated as we move to more complex behaviors.
Yeah, no, I think that's a really good point. I'm going to wrap up and thank the speakers now. Oh, sorry, if you want to, did you have another comment?
Yeah, I'd like to say one more thing, since I'm not sure I'll get a chance to say this later. I'd like to take this opportunity to thank the organizers and Jen and Kristen for putting together a great workshop. I'm sorry I wasn't not here to be there in person, but I think we should all just appreciate the hard work that they've done in putting this workshop together.
Couldn't thank you more.
I agree.
Yeah, and thank everybody for taking part in the panel. We're going to jump now to the multi-agent behavior challenge, which Jen is going to introduce, and then we'll see talks from the winning submissions.
Sorry, everyone. Slides. This should be it.
Oh, can you go back there?
Yep. There you go. And yeah, you're good.
I think it's good. Oh, so thanks a lot, Anne, for the panel discussion. I'm here to introduce the challenge before we dive into the challenge talks later this afternoon from our challenge winners, of course. So I'm excited to be here to talk to you guys about our data set this year, which you can see consists of 4 data sets, 2 of which are video and 2 of which are trajectory data. So the video data, as you can see, consists of triplets of interacting mice, as well as ant and beetle interactions. And the trajectory data consists as well as triplets of mice, as well as fruit fly group interactions. And for this, for the talks this afternoon, we'll be focusing on the winners from the trajectory challenge, as the video challenges are still wrapping up currently. And a quick recap from this morning. So Anne gave some really great examples of multi-agent behavior, and we heard some great talks from many different domains like robot collectives, self-driving cars, sports analytics, animal behavior. And as you guys all saw, multi-agent behavior is crucial in all of these fields. And one very important technique that a lot of people are looking into right now is this idea of how do we learn a good behavioral representation. So if we have behavior data in the form of pose or video, how do we know the representation we are learning on top of this data is good? And of course, as we also talked about, there is so many downstream analysis tasks we could do. For example, like classification or categorization, which assigns frame-by-frame labels of behavior into the video. Oh, Elspeth says we don't see a screen. Oh, I'll keep going for the meantime. Another important task is, of course, behavior discovery, which is becoming more and more important.
Sorry.
Oh, no worries. Which is becoming more and more important. As manual annotations of these behaviors is very hard to scale when we get to a super large amount of fine-grained behaviors. And so discovery is becoming more and more important these days, as we've discussed. The last box, which will pop up next, is generation. And this is something that, oh.
I think you're good.
Oh, it doesn't go to the next
Oh,
thing.
you gotta click on the...
Oh, okay. But, and the other task I want to talk about was generation. Oh, thanks a lot, Anne. Okay, and then so the one question is then, what is a good representation of behavior that takes into account potentially diverse input agents, diverse representation types, as well as diverse downstream tasks? And to give you a sense of how many different models there could be, I'm not going to go through all of it, but for example, your behavior representation could be discrete or continuous. It could capture individual or groups. It could be hierarchical. It could consist of many different timescales. Input modalities, video or trajectory, or both. Many different architectures, and as you guys may know, transformers are like the hottest new thing, so now they're in consideration as well. There's also learning objectives, whether it's supervised, semi-supervised, or self-supervised. So in order to evaluate all of these diverse representation learning models, we've developed an evaluation protocol for the multi-agent behavior challenge, which consists of a wide range of downstream tasks, like the strain or environmental conditions, like the light cycle, or human annotated behaviors like chasing, for example. And once we have these range of downstream tasks, we can use to evaluate our models. We can then compare performance of our models on these standardized evaluation procedures. And these tasks are hidden to the representation learning model during training. And another important note is that we really want these evaluation procedures to capture the performance of diverse agents as well as diverse hidden tasks. And so how we started thinking about these challenges, I guess, just 1 year ago, was when we first proposed the Caltech mouse social interactions data set, where the theme was on supervised learning. And our collaborators was the Anderson Lab at Caltech. We presented this challenge at CVPR last year, as well as with the NERPS 2021 paper. So as you can see, our data set last year was kind of standard supervised learning, where we have a data set of video or trajectory data. And our task was to predict frame-by-frame behavior labels of the animals' interactions by reproducing expert annotations. So we had a set of annotations that matched the trajectory data. And for evaluation, we wanted the model to be able to predict these same behaviors on a test set. Moving on now to this year, we are collaborating with more labs. So we are expanding our benchmarks with more diverse types of agents, as well as tasks. And of course, the goal this year is on evaluating representation learning. Before I jump into that, I just want to comment for next year, we are planning on developing a behavior generalization benchmark in order to learn how behaviors generalize across, say, different labs or in the wild conditions, as well as many different domains and agent types. So if you're interested in helping us develop this challenge, or if you have data recommendations, contact us at maybe.workshop@gmail.com. Okay, so now jumping more into our challenge for this year. So again, our goal was to quantify what good behavior representations are. And one way that we've looked into this is by both discriminating between experimental conditions, which are not annotated by humans, but rather there are different conditions that cause differences in the animal's behavior that should ideally be captured by the representation learning model. The other task that we looked at is recapitulating observations of biology experts by asking them to annotate for a set of behaviors that is of interest to the particular data set, and asking the representation learning model to be able to discriminate between these annotations using a simple linear classifier. So our challenge this year consists of the data set has several thousand short clips of animal poses or video. The task is to produce an informative embedding of the animal's actions without seeing the downstream tasks. And so these hidden tasks that I mentioned earlier, which we use for evaluation, is not seen during the representation learning training itself, but is rather used to evaluate the representation. And our trajectory data consists of both mice and fly data, as you can see here, where the mice data has 12 key points that corresponds to different body parts on the mouse's body, as well as tracking the tail. On the fly data set, there are 19 key points tracking the legs, the body, as well as the wings of the fly. And when we visualize the mouse and fly data sets, they look like this as they behave over time. And in total, we have around 9.6 million frames from the mouse data set and around 4.4 million frames of the fly data set. And we would really like to thank the dataset contributors, which is the Kumar Lab from Jack's Laboratories and the Branson Lab at Genelia, respectively, for contributing these data sets for our challenge. How the evaluation protocol worked for the challenge was that during training, so this is what participants of the challenge had access to publicly, they were able to download raw data sets of the trajectories of the animals, of mice and fly, and they wanted to they would upload a learning embedding without seeing the downstream tasks. So from the participants' training side, there was no labels available, and they want to train this model in a self-supervised or unsupervised way. And then once they have trained the learning embedding, we then ask them to upload the learning embedding, and then we train a series of linear classifiers and evaluate how well their representations can discriminate between a series of hidden downstream tasks using the representation itself. So how we actually did the final leadership board had one more thing beyond evaluating F1 score, which was we did this method called a board account rank, where if you're in nth place, you are awarded n points, and you want the lowest number of points. I'll just give a really brief illustration of 3 tasks to what that means. So here there are 2 methods, and I'm showing the F1 score of each method across these 3 tasks. If I compute average F1 score, method 2 is better, like it's 0.5, and method 1 is only 0.467. So method 1 would actually lose if we compute F1 score. Using board account rank, where I now assign each method a rank based on how well it does relative to other methods. In this case, method 1 would do better because it does better on a larger number of tasks compared to method 2. And the idea this year of using board account instead of average F1 score was because we wanted methods that can perform well on a wider range of tasks versus a method that gets really high F1 score on a small subset. And this is something that we're considering for the future, and would love to hear feedback from you guys once we move on to the challenge discussions. For the challenge itself, we had around 300 participants with over 1,000 submissions across the video and trajectory data sets, and we really appreciate all the participants for entering our challenge. When we look at the distribution of participants, the darker colors here correspond to countries with a greater number of participants. And there was a lot of participation from US and India, But in general, there was people entering from every continent, so we were really happy about that. And one thing to note is that if you're interested in testing your representations after the challenge is over, our evaluator will be staying up possibly indefinitely. So if you want to test out your trajectory or video representation learning methods, you can check our evaluator. And once our video representation challenge is done in a few weeks, we'll update the Slack with those results. So if you're interested, feel free to check our Slack as well. I'll briefly conclude with the challenge takeaways and then move on to the challenge winners. So some of the takeaways we have from this year is that we did observe a significant performance improvement on the hidden tasks, despite the representations not having seen those tasks ahead of time. And this is relative to baseline, which was a simple PCA, a frame PCA, as well as a trajectory variation autoencoder. A lot of the top participants, not everybody, but a lot of people used transformers for behavior this year, which ended up performing the best. So I think that's one thing that we might want to consider for behavior modeling moving forward, since I have not seen a lot of papers using transformers for behavior, at least at this current moment. Some of the challenges we saw was that some of the models did not generalize very well to a larger number or a variable number of agents. So the mouse data set had 3 mice, and the fly data set had 9 to 11 flies, depending on the frame. And some of the models, I think almost all of the models could work on the mouse data set, but only a subset could generalize to flies. So I think generalizing to larger number of agents, like tens of thousands of agents, is still something that we should work on across a lot of models. One final comment is that the rare labels we found during evaluation was difficult for almost all of the models. So if a class occurred, like say 0.1% of the time, I think almost all of the models have very low F1 score for those behaviors. And so learning a general representation that can capture rare behaviors at the same time is something still to be explored. But given that, I'm really thankful to our challenge participants and our mouse challenge winner, Param, runner-up Ed, and fly challenge winner Zach, who will be presenting shortly. And I will conclude my presentation again by we are developing this behavior generalization benchmark, and we're happy to have collaborators. So if you're interested in discussing or having inputs on the challenge for next year, please let us know at the link. And I will pass down to Param if he is here. Thank you.
So I think Param is here. We just need to stop sharing so he can share.
Oh, okay. I also will pull up his introduction.
And stop share.
Can I share my screen?
Oh,
yeah, we can hear you. Yeah, can you share your screen?
I'll do his introduction really quick. So Param Uttarwar is a vision enthusiast with a background in machine learning and image processing. He is currently living in Germany, working on aspects of digital fashion like virtual try-on at a Dusseldorf-based startup. He used to build FPGA-based hardware subsystems for satellites before moving to computer vision. And he also studies music theory and jazz piano in his spare time. I'm passing on to Param.
All right.
Cool. Thanks, Anne, for the introduction, and thank you for inviting me.
Can you share your screen?
Yeah.
Okay. Let's get you over here.
Is my screen visible to you all?
Yeah, you're good to go.
Okay, cool. Yeah, so as I was saying, thanks for the introduction. I am joining you from India. I actually work in Germany, but I'm in India for a couple of weeks. So yeah, I'm here to present my solution for the multi-agent behavior challenge this year for the mouse key point data. So yeah, let's begin. Just a brief recap about the problem statement, which Jennifer already said. So the task for this challenge was to learn a per frame embedding that encodes the mouse triplet interactions. So as you can see in the figure here, the data set provided is a lot of sequences of 3 mice is interacting with each other, and the data is in such a way that the key points are annotated, so it's only the key points for each frame is available as 2D coordinates, and the task is to create an embedding from each frame as a per frame embedding, which is later then used to train a two-layer network, I believe, for their tasks, and that's how the classification scores are calculated. And the tasks are anonymized because, of course, if you know the task beforehand, let's say if you know that the task is chasing, for example, then you can easily encode or calculate a manual feature like velocity and get a classification, and then you get the bias in the embedding. So the anonymization was, I believe, was kept to make the embedding as representative as possible. And these are the mouse key points which were available and the data, so the 12 key points each representing different aspects or different parts of the mouse. So this was the problem statement which I found pretty interesting. So when I went to the website, and it's a bit, I don't have a formal background in mice and biology and neuroscience and behavior encoding, but I found it interesting because it's not like other challenges, vision challenges, which you just take like a off-the-shelf object detection or semantic segmentation models and do a lot of hyperparameter tuning. I thought it required a lot of, it was a creative problem to begin with, so that was kind of a motivation to begin with the challenge. And also I'm a bit of a competitive person, so I like participating in these challenges, and it gives me the kicks, yeah. So my observation of on first preliminary analysis of how the data was and what is required from the task was, and also doing a bit of background research about how these mouse behaviors are handled and how they are evaluated and quantified is. So in the data set, I found that the mice were unordered, in the sense that there was no specific order of the mouse, and the label is denoted from the behavior. So if the order changes, you still get the same. You should expect it to get the same label, but then if you have let's say a function which takes in these 3 mice's key points data, and then you want to include an embedding, the embedding should remain the same regardless of the order of the mouse, and that was kind of my main motivation to start with a permutation invariant embedding, so that it's easier for the network to it makes the tasks easier. So regardless of the order of the mouse, you get the same embedding and hence the same label. So my philosophy of solving any of the machine learning problem is to make the task as easy for the network to solve, because these networks are inherently dumb. So yeah, so my approach was mostly centered around creating and generating this permutation invariant embeddings. So using this operators like sum, mean, and max, for example, these are commutative operators. So if even if you change the order of the operands, you get the same result. And also I came across this. I know from my existing method with PointNet is one of the networks which does inference on this unordered point cloud data. So now the task was also I wanted to use this, so somehow use this, and this is a supervised method, this PointNet. It has labels and this point cloud data, but then the task also then was to how to formulate this particular mouse problem into a problem which PointNet can solve and understand, because that was the benefit of this network. It has permutation invariant. It's a network, so it's the same result regardless of the order of the point cloud here, and to the extension the order of the mouse. So this is a brief overview of the method. I will go in each part in detail in further slides. I will just discuss this as a big picture. So this orange part is the frame data, which is 3 cross 12 cross 2, where 12 is the key points, and 3 is, of course, the number of mouse triplet, and 2 is the XY coordinate. And then, of course, you have past and future data, which is also required to calculate certain features like velocities and trajectory. So the first head is basically you take these three mice, you generate this pairwise mouse features. So you have three mice, you get 6 pairs, and 10 are the features. So these features are primarily like velocities and distances, which then are processed through PointNet. So you get basically 6 points of 10 feature. Each 10 dimension is the feature, 10 is the feature dimension, and then you get a 15 dimension embedding. And the second head is basically creating these handcrafted features, which I came across the existing research papers, which are important for these behavior encoding, and then also sort each feature individually visually so that the computation means if you have like say 3 mouse speed, and then you always sort it so that regardless of the order of the speeds, in the output embedding you get the same result. And the third one is this pose normalized PCA. So you take all the mouse in the data set, you rotate the mice in the principal direction, and then calculate the PCL, the key points, and as a final thing you get in the runtime, you take these 3 mice to do the PCA, and then calculate the mean PCA pose, which contributes to the final embedding that is created. So the mean kind of takes care of the computation invariance. So it doesn't matter where or which order the mice is. Yeah, so the first head is this manual handcrafted features. It's basically velocities and angles and distances between a pair of mice. So these are the features that I extracted for creating the embedding. So one is this distance between the mouse pair, and angle formed between the neck and the body of the other mouse, distance from the nearest wall, distance of the nose, angular speed and absolute speed of the mouse, and also this distance of the nose from one mouse to other nose of other mouse. Some of the features are found from existing research paper, and some of the features, just looking at the data, I thought it was relevant to distinguish the different kind of frames and different kind of positions apart. And to get this invariance, a couple of strategies. One is this sort feature individually, first, and also other is taking the max, min, mean slash either one to obtain computation invariance. This was the manual when you do calculation of manual handcrafted features. Second one is the mean PCA pose of the mice. So as you can see in the figure, there are a lot of, you extract mice from all the frames, different directions, you rotate it into a upward direction, have the key points, calculate the PCA, and then I believe I said 15 dimensions, which almost covers 85% of the variance. And also I don't select tail tip because it was just too much variation. I just did not intuitively not assume that it would contribute much to the behavior per se. And yeah, so the final embedding is kind of the average PCA pose of all the 3 mice. So this is the second head, and then the third is basically using the PointNet data. So the PointNet network, PointNet uses this commutative operators and share them to infer from unbounded point clouds. So how I use it is basically can create these handcrafted features as I discussed in the earlier section from pairwise mouse interaction. So there are 3 mice, so you get 6 pairs of 10 dimensions each, and these 6 pairs are equivalent to 6 points. And this PointNet head gives this permutation invariant embedding. And for the data and the loss, it's chosen that we assume that the nearby frames have a similar embedding, and then some randomly chosen frames from different sequences have negative pair embeddings, and then use the cosine similarity loss to make these nearby frames closer, and then the randomly chosen frames farther apart. So as you can also see in the figure here, you have this data set. You have the anchor point, nearby frame, and then random frame. You from the frame data, you create these pair-wide features for each point, pass it to the point net, you get the embedding, and then backpropagate the cosine similarity loss based on if you want it to be closer and then farther apart. Yeah, so the results from the method that I discussed above was, of course, it was calculated from the mean F1 score was visible as part of the challenge, and the F1 for each task was, some of the tasks were visible, but not all of them. So this was the first place position at the time when the deadline ended. And I also have how individual features affect like the three heads for like three streams of embedding. So just using the manual features, you get an F1 scope of 0.218, and then you add the mean PCA pose, then it increases to 0.42, and then finally after adding this point net embedding, you get the final 0.284, which was the highest at the time when the challenge ended. So yeah, this was about the method, results, and yeah, thank you. If you have any questions, just feel free to ask.
Are there any questions on Zoom, on YouTube, or here for Param on his method? I'll actually get started with the method. So I know, our question. I know that in your method, you had 3 different like representations as your input. So from PointNet, PCA, and handcrafted features. Did you ever, or do you have any sense, or did you do an ablation study on like which features were the most important?
Yeah, so I did not put it in the presentation here, but yeah, I did, and then the mean PCA pose was the most important, followed by the point net, and followed by the manual features. That's how it went in terms of importance of each of them. When I individually submitted it as submissions into the leaderboard challenge.
I see. So just to confirm, the individual, so if you use PointNet by itself, it would work worse than using PCA by itself. It's when they're combined that when they, okay, I see. Are there any other, do you?
Maybe I couldn't hear much about how PointNet works. I'm not familiar with what PointNet is. Can you reiterate that?
Yeah, so the question was, I guess, like, can you reiterate again PointNet and the setup of it? So I guess the understanding is that I think PointNet is a point cloud based method. So like every input point in PointNet is a point cloud, but it's also permutation invariant because in point clouds it doesn't matter which point comes first, but it would be good if you can, I guess, explain that in a little more detail, the PointNet setup itself.
Sure. Should I also share again my presentation? Would be easier.
You can share again if it's helpful for your explanation. Thanks.
Yeah. So yeah, as Jennifer said, the PointNet uses this. There is input point cloud, which is unordered, and then you have, of course, it's supervised, so you have labels, and then you train a car into it. But the good thing of the network is that it gives the same output regardless of whatever order that you put in, like whatever order of the point cloud. And so here, how to translate that into this mouse problem is you have, let's say, 3 mice, and then from 3 mice you get 3 C2 pairs. So let's say we take 1 pair, and then we calculate handcrafted features for each pair. So if you take 1 pair like mouse 1 and mouse 2, we calculate features like distance between them, the angle of in which they are, whether they are facing towards or opposite, the relative speed between them, and all these features are kind of the dimension of the feature. So these are the 10 features that we calculate just manually from what we think is important as features, and that's how you get from 3 mice you get these 6 pairs and then 10 features each. And in point cloud, you have like n cross 3 as input, where 3 is x, y, z, and n is the number of points, usually in order of 1,000 or 10,000. And how we translate it here is basically we have 6 points and of 10 features each. And I also changed the network in terms of the capacity of the network so that it handles, because the number of points here are less, and in that way. And that's once we get from each frame, we get these pairwise features. We pass it through the point net and get an embedding. So first the network is not learned, and then we learn it via this positive by giving this cosine similarity loss between two embeddings. So you said that the nearby frame has a similar embedding, and then the pairwise features would also be similar kind of in a way, and then the point net results in a positive embedding, which we try to make it closer to the anchor point, and then the negative embedding far from it, and that's how it's backpropagate, and then it learns the whole thing.
Thank you. I think that's actually the end of the question time. So thanks again, Param, and congrats on placing first on the mouse challenge. Thank you.
Thank you.
So next I'll introduce Ed Hayes. He works at TikTok as a machine learning engineer within the trust and safety team. Before starting his current role, he graduated in AI and ML from Imperial College London. His interests include self-supervised learning, natural language processing, and multimodal modeling. Passing it on to Ed. Thanks for joining us today.
Let's get you set up. And I'll remember to share the screen this time.
Is there any chance there's
Presenter view?
Yeah, presenter view. I can probably...
I'm not sure. There ought to be a way to do that within Google Slides.
Yeah, you can. Is it, where's the...
Let's quit out of this. One moment. My computer is terrible. Sorry, one moment. As my computer finds itself.
Is it crashed completely?
I hope not. I wouldn't be sad. Maybe I can close some of these things. Technical difficulties. Oh, that goes back. Too many windows and not enough RAM.
It's really stuck, isn't it?
Yeah, it unstuck for a moment. My hope was I could quit out of PowerPoint and free enough memory for it to do something.
You are using a lot of memory.
Yeah.
next to the button that says slideshare is a little drop down, and hopefully it should give you.
Yeah. I can get these guys to respond. Let's see what else I can end. One second as I free up some RAM. It's reacting at least. Presenter view. And where's the actual slides?
So behind it, yeah. Cool.
Okay, and now we need to share the screen. One thing at a time. And before I forget, let's pop out chat again. Okay, I think you're good to go.
Great, thank you. Testing? Okay. So yeah, thank you for the introduction. Before I start, I just need to say that I'm here on my own volition and not on behalf of TikTok. So yeah. There we go, thanks. So yeah, the problem's been quite well described so far, so I think I'm safe to skip over this. But yeah, just talking about reducing the burden for collecting data for analysis. So we can do what's very common in machine learning now, which is just a pre-training step and then fine-tuning to try and leverage some unsupervised or self-supervised representation learning first. So some things I considered when coming up with my approach was that representation should be as general as possible, although it's difficult to define exactly what that means. We should also aim to model behavior not only at a single mouse level, but also interactions between pairs of mice and also the group as a whole. In terms of the third point there, the most recent state of the art looked at kind of an autoregressive type method, whereas for this competition I thought we've got a frame level representation, so if you have an autoregressive style framework, then at the beginning of your sequence you won't have any context, yeah, the representations starting earlier won't be quite as good. So one way to address that, and going on to my approach, was a transformer encoder based on BERT, and one of the key aspects of it was to separate out the input key points for each agent. And I don't know how familiar we are with NLP, but that's a very common thing as well, where you have multiple sentences and you separate them with a special learned embedding. So it's very similar to that. And then have a multitask learning framework based on various different tasks, which I'll go into in more detail. So here's just an overview in terms of the inputs to the model. So just to make it more crystal clear that how I've separated out each agent sequence. you can ignore the purple, yellow, and red boxes for now, but you also so at each input you also have a output embedding, which is a per mouse output embedding, which we then combine in different ways to get group and also paired representations. So for masked mouse modeling, that's what I've dubbed it. We mask about 70% of the input, and that's quite a high proportion of the input compared to most transformer self-supervised methods. The reason is because the sampling frequency is very high in the data set, so obviously not much happens between adjacent frames. I decided to mask spans of the input key points instead of randomly masking them, because it may just interpolate between nearby key points. But another reason, one of the reasons why I decided to separate out the agent key points again is because it allows the model to attend over the actions of what other mice are doing in order to solve masked modeling. So if you've masked a portion of the input of one mouse, then the model can use the actions of the other two mice to try and infer what it's doing, which is kind of treating interactions as a first-class citizen. Whereas if you concatenated all of the key points in together and passed it through as one, then obviously you don't get that kind of interaction. So second set of tasks were feature predictions. So you've got individual mouse features, which is just speed, distance from the center, that kind of thing. And we can predict it directly from the output embedding. Inter-mouse features, which is just these mouse pairs that Param was talking about earlier. It's a very similar idea. I decided to take the embeddings for each mouse in the pair and subtract them. There are other ways of combining them, but you're just aiming to get a representation for the pair as a whole. And then in terms of group features, you're concatenating the embeddings for each of the agents separately. So in terms of contrastive learning, yet another task. I tried two different types. One worked and one didn't, but I thought I'd cover them both because I thought the other one should work, but I'm not sure why it didn't. So I tried one in terms of very similar to Param, where the positive pair is two input subsequences sampled from the same 1 minute clip, and the negative pair are just randomly sampled from the data set. And again, you pool using a weighted average to get a single representation for the entire input subsequence. And then just a normalized temperature scaled cross entropy loss. Say that 5 times fast. Yeah, that's pretty bog standard. And then I also tried random mouse replacement, which is where you perturb, you replace one of the trajectories in the set with a randomly sampled one, and you try and get the model to predict whether it's being perturbed or not. That really didn't work quite catastrophically. I think perhaps it introduced some extra noise that it wasn't expecting. Perhaps from a feature perspective, it didn't really make sense, maybe if one is overlapping or something like that. But I think it should work in theory, and there could be some future research on that. So yeah, this is the total overview of the model. Starting from the bottom, you've got again the separated key points, and then you provide you embed and add all of the features, and then you pass them through the transformer encoder, And then the output embeddings of green are per mouse. And then starting from kind of the left-ish, you do your masked modeling, so you predict the masked key point for an individual mouse. And then the yellow box at the top is the intermouse feature, where you're predicting the pair features by subtracting the two embeddings of the pair. And then I've got the group feature as well. And the group feature is what I ended up submitting. So the group embedding is what I ended up submitting. It's just a concatenation. So one or two extra things about this architecture is that it should be quite flexible. So if you've got data sets with different numbers of mice, it's quite amenable to being trained on different ones, and also should be able to perform inference on unseen numbers of mice. So if you train on 3 and 2, you should be able to do inference on 4, for example, which is more difficult if you've concatenated all of the input representations at the start. Another thing is if you have labels per mouse, so let's say one of the mice is exhibiting a particular behavior, but the others aren't, and you have annotations for that, then you can go back and find out which mouse it was in the original data, whereas if you, again, just have a single group representation, that's much more difficult to do. And actually, yeah, no, good. Go on to the next slide. Just some extra details. Some augmentations, rotation, reflection, random noise. I took one of the binary classification I took one of the public annotations for a binary classification task as well. I only chose chasers because I found lights didn't actually help. And yeah, concatenate the embeddings for the final embedding for the group. And I encoded overlapping windows. So the input length is 80, and then I encoded overlapping windows to make sure that at every frame it's got as much context as possible. And yeah, that's it. I don't have an ablation for you. I didn't have time, I'm afraid. But I've kind of plotted over time what happened to my score. The big spike in the middle is when the evaluation method changed, so I can't take credit for that. But yeah, I think introducing the separated model at the start was quite helpful, and the mouse pair features were also very important. And yeah, that's all. Thank you.
Are there any questions from Zoom or YouTube or in person? Oh.
When you introduce the augmentation that you described, the rotation and reflection, do they have how much of the features that you have bubble, like velocity and angles, how much of the features are actually affected? Like changing?
How much are they changing?
Yeah, since like angles won't change if you rotate it.
Sure. Yeah. So they shouldn't change at all, most of them, because all of the positions of each mouse relative to the others are the same. So there was, I took the augmentations from, I believe it was your paper, Shaber. Yeah. So I took most of them, and there's an extra augmentation where you randomly randomly shift one of the trajectories, and I didn't take that for no particular reason other than I thought it might change the features, and I wasn't sure.
I wasn't sure about that one either when we did it. It's good you removed it.
Well, no, not sure. But yeah, I hope that answers your question. Cool.
Yeah, I guess just adding a little more, those augmentations were designed not to be perturbed by handcrafted features, because we thought the features, at least for task programming, we thought the features that we chose were important for behavior. And then we didn't want the augmentations to actually change the behavior the animal was exhibiting. So they're designed to be like attribute preserving. I guess that was the term we used, I think.
Makes sense.
I have a really quick question. I know you didn't have time to do a full ablation study. I guess I'm just always very curious. Do you have any sense? I know your loss function at 4 terms. Wait, now I'm like, remember. So one was a chasing loss you added, contrastive loss,
Masked modeling,
mouse modeling, very difficult to say.
And then feature prediction.
Feature prediction. Do you have any sense which one is the most important? I know you didn't have time to do an ablation study, but as you did model development, surely you saw some values.
I got a so the features were the most important, to be honest. You can see there's a small increase when I separated out the key points, but I spent quite a long time on features because I thought they were probably the most important aspect.
Okay, that's nice to know. Thanks a lot.
Thank you. Thank you.
Yeah, I think we were actually running behind. I know Zach is in Australia, but let me introduce him really quick. Hopefully it's not too late. So our next speaker is Zach, who is a computer scientist and machine learning enthusiast. He graduated from the University of New South Wales, or UNSW, Sydney, in late 2021 with a Bachelor of Computer Science. Currently, he is tutoring 2 machine learning courses at UNSW before starting as a software engineer at Google. Zach regularly competes in machine learning competitions across a wide range of topics, but primarily within computer vision. Hello, Zach, passing it on.
Hi, thank you very much. I'll just share my screen as well. Okay. So my model could work in both the fly challenge and the mouse challenge. We've kind of had a bit of an introduction on both of those already, so I'll kind of jump straight into it. So briefly, the model, I take the original key point features, and I am doing similar to some of the other people, I am creating some of my own handcrafted features from that, which is of a high dimension. And then on the frame level, I downsize these high dimensional features into the target size. So for example, for the mouse data set, we needed to reduce that down to 128 dimensions and 256 for the fly data set. And so with these downsized features, then that goes into my transformer, and so that combines it at the sequence level, and then through masked language modeling as well. And I'm trying to predict back the original the high dimensional features, which is earlier in the sequence, as well as a couple of the class labels that we were given, which was just there were just 2 of them that I was using, and so for example in the mice, that was whether or not there was chasing happening in that frame, or if it was day or nighttime, were the only 2 labels we were provided with. So to talk about some of these handcrafted features, I think I may have been using more than, like this, I was only using these features and doing masked language modeling, or didn't have, that was like my primary input that I was using, and I think I might have had more features in total than some of the others. So this included angles, distances, and velocities between body parts within an agent. So like between the feet and the head, or the wing tips and the abdomen or something like that, as well as also the angles and distances between all pairs of agents. And this meant that when there were a lot more agents, I had more a higher dimension. For the flies, like, it's not the exact same features because there's obviously different key points on mice and flies, so they were custom to the data set as well. And then I also included a few features like angles and distances to fixed points in the within the scene as well. I was just guessing that there might be things in the scene that they were interacting with as well. In the cases where there were less, like there were a maximum of 11 flies, and I was just zero padding it out so that the model kind of could take in a standard size for each, for every frame. It doesn't matter how many agents there are. And yeah, so all of these features aren't, are all kind of concatenated together per frame as the input. So I've got all of the pairwise interactions as well as the within a single agent body parts, distances, features, I guess. They're all together in the one frame features. And yeah, I also thought I'd mention the way that I was encoding the angles. Rather than just using an angle like between going from, I could say that it went from like 0 round to 360, but then if it kind of just suddenly jumps from one side from like 0 to 360, it's hard for the model to learn. It thinks that those are completely opposite, whereas they're actually kind of quite similar. So I instead embedded them using two different numbers, what I kind of refer to as like a forwardness and a rightness. So with these diagrams, the top one is, these are kind of aligned with the direction that this fly is facing. And so this angle here would be about 0.5 kind of in forwardness, because it's mostly in front of the origin, and it would be approximately a negative 0.5 in the other dimension as well, because it's like over towards the left. Okay, so then comes the part where I'm doing the sequence modeling, and I am trying to combine all of these features across frames so that you can learn the whole sequence features. So the handcrafted features that I had already created are downsized with shallow neural network, and these compressed features are what's used as the inputs to the transformer, and in particular I'm using a perceiver transformer, and this was because it means that I can use a longer sequence than like the BERT models that were mentioned earlier, because it's got a lower complexity. It's only scales with it scales linearly with the size of the sequence input rather than quadratically. And yeah, so that means that I had long sequences of 512 frames in at once that was that were getting masked and re-predicted on the other side. And also for the fly data set, I was just emitting every second fly due to the, every second frame, I mean, due to the high original frame rate. And this is just a diagram of what the perceiver model from the perceived paper originally looked like. So my kind of objectives primarily was masked language modeling, but it's modeling the agents, but rather than a natural language like English or so on. And I was using mean square error as my loss function to predict those original unmasked high dimensional features. And I was also masking out a variable number of frames during training, and they are up to 8% in total, and they were often masked out in regions. So many frames at once could be masked all together. And then my second training objective was to predict a couple of the class labels that we were provided with, and I was just aiming to do that with cross-entropy loss. Yeah, so just coming back to that original diagram so that we can kind of see how it all kind of fits together. So the original key point features, so this was just like the X and Y coordinates of where every key body part was in each frame. That gets converted into the higher dimensional handcrafted features, gets downsized with a 2-layer neural network on the frame level. Then 512 of these get combined together in the transformer encoder. And something I should have mentioned, just between the high dimensional features and the downsized feature vector is where I am doing the masking. So I might mask out this frame altogether, and so the shallow neural network will kind of learn to create a token for what is masked as well, what a masked input should look like, and yeah, then combine all of these frames together to hopefully recreate the missing masked out information and yeah, predict the features back again. All right, and yeah, that's it. So are there any questions?
Are there any questions for Zach, either in person, on the Zoom chat, or on YouTube? I think I can start off with a question, because I was putting together the paper that was describing your methods at some point, and I noticed that, Zach, your features were much higher dimensional, like your handcrafted features were much higher dimensional than some of the other top participants. I was wondering, how did you maybe come up with a set of features, and also what motivated you to write the 2000? I think in your feature dimension was around 2000, I want to say.
So I did have 2000 for the flies, but it's not like I'm kind of individually finding all of those myself. So they're kind of, I was doing for every fly, there's kind of a feature to all the other flies in there, which so you can kind of do that in a loop easily. And I kind of wanted, I didn't look at existing research to see what other people had done. I just kind of guessed what seemed useful to me, actually. So, for example, there's within the flies, the features, within one fly, the features that I had was the distances and angles between the head and most of the other body parts, on the top half of the fly, and then I had the distances and angles between the the abdomen and all of the other kind of body parts on the lower half of the fly. And then between the flies, I also had the angles and the distances, not just between like the center and the center, but I was doing to some of the other things. So between like the head of one and the tail of the other, as well as the wing towards the wing tips and so on as well. So I just tried to add in as many as I kind of thought would be useful for something.
Oh, thanks. Are there any other questions for Zach? Oh, yep.
I think I go here. I was just wondering what your impression was, what you spent the most time on during the competition. Was it feature engineering? Was it adding terms to your loss function? What did you spend the most time on, and what improved your score the most?
Yeah, okay. So I think the features I didn't actually spend that long on. I just kind of was like, all right, let's just try and get everything down. So some of them probably aren't very useful, but there's enough of them that I've got a lot of useful ones as well. And so I didn't kind of think about that too long. So there were different things within the model about like what it should be, like the length of the sequence, for example. How should I be when masking it out? Should I be like replacing it with a just zeros, or should it be some special token? Is there yeah, and I also with like the labels, I was kind of experimenting with should I kind of only train, because for example the chasing doesn't happen during very many frames, so I was learning only on where chasing was actually observed and nearby frames, and just randomly a few other frames every now and then, but not on all of it, not on all of the non-chasing frames. Yeah, so I guess model architecture is where I was spending most of the time, changing the kind of parameters of the model.
Oh, thanks a lot. Since we are really close to poster session time, maybe I'll just ask the challenge competitors one question. So Zach, if you could stay on, or Param, if you're still there. So maybe one question I'd like to ask the three top participants is, for our challenge next year, is there any feedback you would want to give us back to us as organizers, so that we can make your experience, like future participants' experience, like more smooth for next year? Like, is there anything we could have done differently that would have like helped you out when you're developing your solution, for example?
I don't know, it seemed pretty good to me.
Thank you.
Not something else that I can think of at the moment, no?
Just that it wasn't clear that we could use all of the data. So in the last week, I had to adapt everything to use the solution that you want. But in terms of the way it was set up and everything, I thought it was really well.
Oh, thanks. I guess for the Zoom people, Ed was commenting that maybe the way that we communicated what data could be used for like train versus test wasn't super clear. That's something we can do better on for next year. For example, I know we had a few publicly available labels that participants could actually use, so we'll be more clear about that for future years. But if you guys think of anything else, of course, we'll be on Slack. And I'll pass it back to Anne. Thanks again, you guys.
Okay. So that, let me bring us into a more normal looking view here. So that brings us to the end of the talks component of our workshop. From now we're going to go to downstairs gallery to the poster session to see some of the other submitted work. Thank you again so much to the invited speakers, the submitted speakers, and to the participants in the challenge for contributing to this. It's been really interesting hearing about all of your work, and I think there's going to be a lot of mouse researchers downloading your code repositories and trying them out on their data sets now. So thank you all, and thanks to the folks on YouTube and Zoom and Slack and everything else who tuned in over the course of the day and participated as well. All right.